Before there were bones, there were builders. Before there were brains, there were decisions. Before anything we'd recognize as alive walked, swam, or crawled across this planet, something else was already engineering. Not with tools, not with blueprints, but with chemistry, geometry, and something we still don't fully understand: the ability to build itself. Right now, inside you, trillions of molecular machines are working. They're copying DNA, transporting cargo, generating energy, repairing damage. They move with purpose. They solve problems. They coordinate across distances. And every single one of them built itself from nothing but atoms following rules we're only beginning to decode. We call it life. But maybe we should call it what it really is: the universe's first architecture, the first machinery, the first computation, all happening at a scale so small we forgot it was engineering at all. But here's what we missed for so long. These molecular machines didn't arrive fully formed. They emerged step by impossible step from simple chemistry. Somehow molecules learned to fold into shapes that could do work, shapes that could copy themselves, shapes that could sense, respond, and adapt. The question isn't just how life began, it's how matter learned to move with intention, how structure learned to store information, how the non-living became the first thing that could build, maintain, and repair itself without an architect. Tonight, we're going back to the moment chemistry became machinery, to the origin of movement, memory, and making, to the first machines that built themselves, because if we can understand how a molecule becomes a machine, we might finally understand what life actually is, and why the universe seems so determined to create it. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a single molecule drifting in ancient water. It's not alive, not yet. Just a chain of atoms, maybe 100 units long, folded into a particular shape by forces we'd call mundane. Electromagnetic attraction, hydrogen bonds, the simple geometry of angles and distances. Nothing about it suggests purpose. Nothing about it says machine. But then something happens. Another molecule drifts close and the first one grabs it. Not randomly, not by accident. The shape of the first molecule has a pocket, a groove, a surface that fits the second molecule perfectly. When they touch, something changes. A chemical bond forms, energy releases, and the first molecule, having done its work, lets go and drifts away, ready to do it again. This is a catalyst, the first machine. It doesn't think, doesn't plan, doesn't know what it's doing, but it does something. It makes a reaction happen faster, more efficiently than random chance would allow. It performs work. And the stunning thing, the thing that changes everything, it wasn't designed by anything outside itself. The shape that allows it to work emerged from the properties of the atoms themselves. The machine built itself. Now multiply that by billions, by trillions. Imagine an ocean full of these molecules, each one folding into shapes dictated by their atomic composition, each shape enabling a different kind of work. Some molecules speed up reactions, others slow them down. Some bind to specific targets with lock and key precision, others flex and change shape in response to their environment. Without intention, without intelligence, chemistry had invented engineering. This is where life begins, not in the spark of consciousness or the complexity of cells, but in the fundamental discovery that shape equals function, that structure can perform work, that molecules can be machines. But calling them machines almost feels wrong, doesn't it? When we think of machines, we think of human creations, pistons and gears, motors and circuits, things designed with purpose and built from the outside. These molecular machines are different. They're not designed. They're discovered, or more accurately, they discover themselves. The right combination of atoms folds into the right shape, and suddenly, that shape can do something useful. It can make things, break things, move things. It can work. And here's where it gets strange. Once these molecular machines existed, once chemistry stumbled onto shapes that could perform work, something else became possible. The machines could start building other machines. A molecule that catalyzes one reaction could help create a molecule that catalyzes another reaction. which could help create a third, which could circle back and help create the first. Feedback loops, networks, systems of machines building systems of machines. This is metabolism, the web of chemical reactions that powers all life. But it started before life, in that chemical space where simple molecules were beginning to coordinate, beginning to form the first proto-networks of mutual construction. The key insight, the thing that makes this so profound, is that none of this required a designer. The machines emerged from the laws of physics and chemistry. The right atoms in the right arrangement produce the right shape, and the right shape performs the right function. It's automatic, inevitable even, given enough time and the right conditions. But that raises a deeper question. If molecular machines can build themselves, if structure and function emerge automatically from chemistry, then what separates the non-living from the living? Where's the line? Most people would say, life is what reproduces, what evolves, what responds to its environment with purpose, and that's true. But all of those properties rest on something more fundamental. They rest on molecular machines, on structures that can perform work without being told, that can build and maintain themselves using nothing but the materials and energy around them. A virus has molecular machines but can't reproduce without hijacking a cell. A cell has molecular machines and can reproduce, but only because its machines work together in coordinated networks. An organism has trillions of cells working together, each one running its own molecular machinery, all of it coordinating across scales from nanometers to meters. The boundary between living and non-living isn't a wall, it's a gradient, and somewhere on that gradient molecular machines crossed a threshold. They became complex enough, coordinated enough, that they could maintain themselves, repair themselves, copy themselves. They became autonomous. That moment, that threshold, is what we're searching for tonight. Not the moment life suddenly appeared, but the slow, stepwise process by which chemistry became architecture, by which molecules became machines, by which machines became systems, and systems became the first thing that could truly be called alive. We're going to trace that path from simple chemistry to cellular complexity, from random folding to purposeful construction, from the first catalyst to the molecular motors that drive every living thing today. And what we'll find is that the machines that built themselves didn't just create life, they created the possibility of everything that came after. Movement, sensing, thinking, evolving. Every innovation in the history of biology rests on the foundation of molecular machinery, on the fact that atoms can arrange themselves into shapes that work, that build, that maintain order in a universe that constantly slides toward chaos. So let's begin where all engineering begins, with the simplest question a molecule can answer. What shape am I, and what can I do? Let's start with the most basic truth about how machines work. Shape matters. A key fits a lock because the peaks and valleys of the metal align with the pins inside the mechanism. A wrench turns a bolt because the hexagonal opening matches the hexagonal head. Human engineering has always understood this. Form follows function, and function depends on form. But we invented that relationship. We designed the key to fit the lock, the wrench to fit the bolt. Molecules don't have that luxury. They can't design anything. They can only be what their atomic composition allows them to be, and yet somehow they end up with shapes that work, that perform specific functions with precision that rivals anything humans have engineered. The secret is in the forces. When a protein folds, and proteins are the workhorses of molecular machinery, the molecules that do most of the actual work in living systems, it's not following instructions in the way we usually think of them. Yes, the sequence of amino acids is determined by DNA, and that sequence matters enormously, but the sequence doesn't tell the protein what shape to become. It tells the protein what forces to experience. Each amino acid has chemical properties. Some are hydrophobic, repelled by water, wanting to hide in the interior of the structure. Others are hydrophilic, attracted to water, wanting to face outward. Some are positively charged, others negative, and opposite charges attract while like charges repel. Some amino acids form strong bonds with each other. Others are flexible, allowing the chain to bend and twist. String hundreds of these amino acids together in a particular order, drop that chain into water, and something remarkable happens. The chain begins to fold. The hydrophobic amino acids cluster together, away from the surrounding water. The charged amino acids arrange themselves to minimize repulsion and maximize attraction. Hydrogen bonds form between different parts of the chain, pulling distant segments close together. The whole thing collapses, twists, bends, and settles into a compact three-dimensional shape. This happens in milliseconds. No thought, no guidance, just physics and chemistry playing out at molecular scale. And the shape that emerges isn't random. It's the shape with the lowest energy, the most stable configuration given that particular sequence of amino acids and the environment around it. According to Christian Anfinsen's groundbreaking work in the 1960s, which earned him the Nobel Prize, a protein's shape is entirely determined by its amino acid sequence. Unfold it, and it will fold right back into the same shape, every single time. That's astonishing. It means the information for the shape isn't stored separately somewhere. It's encoded in the sequence itself, in the properties of the amino acids and how they interact. The machine's blueprint is the machine. But here's what makes this even more profound. The shape isn't just stable, it's functional. The way the protein folds creates pockets, grooves, surfaces, clefts, three-dimensional features that can interact with other molecules in specific ways. A pocket might be exactly the right size and chemical character to bind a particular small molecule. A groove might fit along the surface of DNA. A cleft might bring two molecules close together, lowering the energy barrier for a reaction. The protein becomes a tool, not designed but discovered through the blind process of chemical self-organization. And not all proteins are static. Some of them move. They change shape in response to their environment, and that shape change performs work. This is where chemistry really becomes machinery, where structure transitions into motion and motion into function. Consider myocia, one of the motor proteins responsible for muscle contraction. It's a long molecule with a head that can bind to another protein called actin. When myosin binds to actin, it's in one shape. Then ATP, adenosine triphosphate, the energy currency of cells, binds to myosin. The addition of that small molecule triggers a conformational change, a shift in shape. The myosin head tilts, pulling on the actin filament. Then the ATP gets hydrolyzed, broken down, releasing energy, and myosin returns to its original shape, ready to do it again. One cycle of this process moves the actin filament by about 10 nanometers, 10 billionths of a meter. Individually, it's nothing. But millions of myosin molecules working in concert, all of them pulling in coordinated waves, generate the force that lets your muscles contract, that lets you move your hand, blink your eyes, pump your heart. Molecular machines, cycling through shapes, converting chemical energy into mechanical work. And every motion, every shape change, is driven by the same forces that made the protein fold in the first place. Electromagnetic attraction, hydrogen bonding, hydrophobic effects, the geometry of atoms. This is what we mean when we say life is machinery. Not metaphorically, literally. These are machines, mechanical systems that perform work, that have moving parts, that convert energy from one form to another. They just happen to be made of proteins instead of metal, powered by chemistry instead of combustion. But calling them machines also reveals something we missed for a long time. Machines can be engineered, they can be optimized, they can evolve. Because here's the crucial point. Not every sequence of amino acids folds into a useful shape. Most random sequences would fold into useless blobs, or wouldn't fold stably at all. The proteins that exist today, the ones performing all the work in every living cell, are the survivors. They're the sequences that happen to fold into shapes that could do something useful, shapes that gave the organisms carrying them some advantage, some edge in survival and reproduction. Natural selection has been engineering molecular machines for nearly 4 billion years, not with intention, not with foresight, but through the relentless filter of what works. Mutations change amino acid sequences. Most changes make things worse. The protein doesn't fold right, or folds into a shape that doesn't work as well. Those variants get eliminated, but occasionally a mutation creates a protein that works better, or works differently in a way that opens new possibilities. Those variants spread. Over deep time, this process has produced molecular machines of staggering sophistication. Proteins that can copy DNA with error rates of less than one mistake per billion bases. proteins that can pump specific ions across membranes against concentration gradients, proteins that can sense single photons of light, proteins that can generate rotational motion at thousands of revolutions per minute, and all of it emerges from the same basic principle. Shape equals function, and shape emerges from sequence, and sequence changes through mutation and selection. But proteins alone aren't enough to make life. They're tools, machines, engines. But tools need organization. Machines need to be arranged into systems. Engines need to drive something. The next level of complexity, the next step from chemistry to life, is when molecular machines start coordinating with each other. When they form networks. When one machine's output becomes another machine's input. When feedback loops emerge and systems begin to regulate themselves. This is metabolism, and it's where things get truly interesting. Metabolism sounds like a single thing, but it's not. It's thousands of chemical reactions happening simultaneously, each one catalyzed by a specific enzyme, each one feeding into others, creating a web of transformations that spans the entire cell. Glucose comes in. Through a series of 10 reactions, it gets broken down into pyruvate, releasing energy that gets captured in ATP and electron carriers. The pyruvate enters the mitochondria, gets converted into acetyl-CoA, feeds into the citric acid cycle, 8 more reactions producing more ATP, more electron carriers, carbon dioxide as waste. The electron carriers feed into the respiratory chain, a sequence of protein complexes embedded in the mitochondrial membrane that pump protons, create a gradient, drive a molecular turbine called ATP synthase that cranks out ATP molecules by the thousands. That's just one pathway. There are hundreds more. Pathways for building amino acids, for synthesizing lipids, for making nucleotides, for breaking down toxins, for storing energy, for mobilizing it. Each pathway is a series of reactions. Each reaction is catalyzed by an enzyme. Each enzyme is a molecular machine that had to fold into exactly the right shape to do its particular job, and all of these pathways are connected. The product of one reaction becomes the substrate for another. Energy released in one place gets consumed somewhere else. Molecules flow through the network like goods through an economy, constantly being built up, broken down, modified, redirected. What makes this remarkable isn't just the complexity, it's the coordination. The cell doesn't run all pathways at full speed all the time. That would be wasteful, even destructive. Instead, metabolism is regulated. When energy is abundant, pathways that consume energy to build complex molecules ramp up. When energy is scarce, pathways that break down molecules to release energy take over. When a particular amino acid is plentiful, the cell stops making it. When it's needed, production resumes. This regulation happens through feedback. Many enzymes don't just catalyze reactions, they respond to the presence or absence of specific molecules. An enzyme at the beginning of a pathway might have a binding site for the end product of that pathway. When the product accumulates, it binds to the enzyme and changes its shape, reducing its activity. The pathway slows down. When the product gets consumed and its concentration drops, the enzyme returns to its active form. The pathway speeds up again. Negative feedback, the same principle that governs thermostats and cruise control, operating at molecular scale through nothing but shape changes and binding events. Other enzymes are activated by their products, creating positive feedback loops. Some are regulated by molecules from completely different pathways, creating crosstalk between systems. The result is a network that self-adjusts, that responds to changing conditions, that maintains balance without any central controller telling it what to do. This is what researchers in the mid-20th century began calling homeostasis, the ability of a system to maintain stable internal conditions despite external fluctuations. But the term almost undersells what's happening. This isn't just stability, it's dynamic equilibrium. The cell is constantly consuming energy and materials, constantly building and breaking down molecules, constantly running reactions in both directions. But the overall state remains stable because the rates of all these processes are balanced. It's like a fountain where water constantly flows but the shape stays the same. Except in a cell, the shape isn't determined by gravity and plumbing. It's determined by the properties of thousands of enzymes, the availability of substrates, the concentrations of regulators, all of it interacting in networks so complex that we're still working out the details. And here's what makes this relevant to the origin of life. Metabolism had to come before anything we'd recognize as a cell. Before membranes, before DNA, before reproduction, there had to be networks of reactions that could sustain themselves, that could extract energy from the environment and use it to build complexity. The earliest metabolic networks were probably tiny compared to what exists today. Maybe just a handful of reactions, catalyzed by simple molecules that weren't even proteins yet. Perhaps RNA molecules with catalytic activity, or small peptides, or even mineral surfaces that could speed up certain reactions. But once you have even a small network of reactions, where the product of one reaction feeds into another, and some of those reactions generate useful products or energy, you have something that can persist. If one of the reactions produces a molecule that helps catalyze another reaction in the network, you have autocatalysis, the network promoting its own continued existence. This is the bridge between chemistry and life. Not a single moment, not a lightning bolt animating dead matter, but a gradual transition where networks of reactions became self-sustaining, where chemistry organized itself into systems that could maintain their own complexity against the constant pull of entropy. Stuart Kaufman, a theoretical biologist, has called these autocatalytic sets, collections of molecules where each molecule's formation is catalyzed by other molecules in the set. In principle, if you have enough different kinds of molecules and enough possible reactions, you might spontaneously get sets that close in on themselves, that become self-sustaining. Mathematical models suggest this kind of organization might be inevitable once chemical diversity crosses a certain threshold. Whether that's exactly how life started, we don't know. The early Earth was a messy place, full of competing chemistries, different energy sources, countless possible pathways. But somewhere in that mess, molecular machines began coordinating. Reactions began forming networks. Networks began sustaining themselves. And once that happened, once chemistry became a self-maintaining system, everything else became possible. Because self-maintaining systems can grow. If the network produces molecules faster than they decay, the system expands. It can spread. It can colonize new spaces. And if the network occasionally makes errors, if molecules get copied imperfectly, or if new molecules get incorporated, then the system can change. It can evolve. But there's a problem. A network of reactions floating freely in the ocean is vulnerable. Products diffuse away. Useful catalysts get diluted. The system can't concentrate its own components. It can't separate itself from the environment. This is where enclosure becomes essential. This is where chemistry needs to invent boundaries. Not walls in the sense we usually think of them, rigid, impermeable barriers, but membranes, selective borders that keep some things in and let other things through. And remarkably, membranes are another thing that builds itself. Certain molecules, particularly lipids, have a split personality. One end is hydrophilic, attracted to water. The other end is hydrophobic, repelled by water. Drop these molecules into water and they spontaneously organize. The hydrophobic ends cluster together, hiding from the water. The hydrophilic ends face outward, interacting with the aqueous environment. The most stable arrangement is a bilayer, 2 layers of molecules with their hydrophobic tails sandwiched in the middle and their hydrophilic heads facing out on both sides. And if you have enough of these molecules, the bilayer curves, closes in on itself, forms a sphere, a vesicle, a bubble with an inside and an outside. No instructions required, no assembly process, just molecules following their chemical nature, and a boundary spontaneously appears. Suddenly, the inside is different from the outside. Molecules trapped inside can interact with each other at higher concentrations. Reactions that produce useful products don't immediately lose those products to the surrounding environment. The system can maintain gradients, accumulate specific molecules, create conditions that favor certain reactions over others. This is a protocell, not alive, not yet, but closer. A self-maintaining chemical network enclosed in a self-assembled membrane. A system that can concentrate its own components, that can evolve independently of its surroundings, that has an inside distinct from the outside. And once you have that distinction, once you have a boundary between self and environment, you have the beginning of autonomy. Autonomy is a strange concept when you're talking about molecules. We usually reserve that word for things with agency, with decision-making capability. But in the context of early life, autonomy means something simpler and more fundamental. The ability to maintain your own identity, to persist as a distinct system separate from your environment. A membrane gives you that. It creates a pocket of chemistry that operates by different rules than the chemistry outside. Inside the vesicle, concentrations can be different, pH can be different, the mix of molecules can be completely unique. The membrane doesn't just separate, it defines. Everything inside is part of the system. Everything outside is environment. But a membrane alone isn't enough. If the boundary is perfectly sealed, completely impermeable, the system inside stagnates. It can't bring in new materials, it can't export waste, it can't harvest energy from outside. The protocell might maintain its distinct chemistry for a while, but eventually it runs out of resources and stops. Life needs membranes that are selective, not absolute. Barriers that can regulate what crosses them. And again, chemistry provides this automatically through nothing but the properties of the molecules involved. Lipid bilayers aren't completely impermeable. Small uncharged molecules can slip through, water, oxygen, carbon dioxide. They dissolve into the hydrophobic interior of the membrane, diffuse across and emerge on the other side. Larger molecules, or anything with a charge, can't cross easily. They're blocked. But early cells, or proteaels, would have needed more than passive diffusion. They would have needed ways to actively transport specific molecules, to pump things in or out against concentration gradients, to control their internal chemistry with precision. And this is where molecular machines really earn their name, because transport proteins are engines. They use energy to do work, to move molecules from where they're abundant to where they're scarce, fighting against the natural tendency of everything to spread out and equalize. Consider the sodium-potassium pump, one of the most important proteins in animal cells. It sits in the membrane and does something that seems impossible from a thermodynamic perspective. It pumps sodium ions out of the cell and potassium ions in, both against their concentration gradients. Sodium is more concentrated outside than inside. Potassium is more concentrated inside than outside. The pump makes both gradients steeper. How? By consuming ATP. The pump binds 3 sodium ions on the inside of the cell. ATP binds and gets hydrolyzed, breaking one of its phosphate bonds. The energy from that reaction causes a conformational change. The pump changes shape, opens a pathway to the outside and releases the sodium ions. Then 2 potassium ions bind from outside, triggering another shape change. The pump releases them inside and the cycle repeats. One molecule, cycling through shapes, moving ions uphill against their concentration gradients, powered by chemical energy. It's a pump in the truest sense, mechanical in its operation, just built from proteins instead of pistons. And the gradients it creates aren't just for housekeeping, they're functional. Nerve signals depend on those gradients. When a neuron fires, sodium channels open and sodium rushes in, driven by the gradient the pump created. That influx changes the voltage across the membrane, opening more channels, creating a wave of electrical activity that propagates down the axon. Thought, sensation, movement, all of it ultimately powered by molecular pumps maintaining concentration gradients. But we're getting ahead of ourselves. Early cells wouldn't have had voltage-gated channels or action potentials. They would have needed something much simpler, ways to bring in nutrients and export waste. And even that requires machinery. The earliest transport systems were probably just channels, proteins that form pores through the membrane, allowing specific molecules to pass through. Some channels are always open, others are gated, opening in response to particular signals, a change in voltage, the binding of a molecule, a mechanical stretch. The sophistication of these systems is startling. Some channels are so selective they can distinguish between potassium and sodium ions, which differ in size by only a fraction of an angstrom. The channel has a narrow region, a selectivity filter, where the geometry is so precise that only potassium fits. Sodium is too small, it doesn't make the right contacts with the protein, doesn't get stabilized, doesn't pass through. Potassium fits perfectly, gets briefly coordinated by specific atoms in the protein, and then continues through. It's lock and key again, but at atomic resolution. The protein's shape determines what passes and what doesn't, and that shape emerged through evolution, through random mutations being filtered by natural selection for functionality. So now we have a picture: vesicles enclosed by membranes, filled with networks of metabolic reactions, with proteins embedded in those membranes controlling what goes in and out. A system that maintains its own distinct chemistry, that harvests energy from the environment, that builds complexity internally while remaining separate from the outside world. This is starting to look like life, but it's still missing something crucial. It's not hereditary. There's no way for the system to copy itself, to pass on its organization to descendants, and without heredity there's no evolution. Without evolution there's no way for the system to improve, to adapt, to become more sophisticated over time. This is where information enters the story, because the molecular machines we've been discussing, proteins, don't just exist, they have to be built, and building them requires instructions. In modern cells, those instructions are stored in DNA. The sequence of nucleotide bases, adenine, thymine, guanine, cytosine, encodes the sequence of amino acids in proteins. The genetic code is read by molecular machines called ribosomes, which link amino acids together in the order specified by the DNA sequence, producing proteins that fold into their functional shapes. It's an incredibly elegant system. Information stored in a stable molecule, read out by machinery, translated into functional structures. But it's also almost certainly not how things started. DNA is too stable, too inert. It's great for long-term storage of information, but it doesn't do anything on its own. It needs enzymes to replicate it, to transcribe it, to repair it. And those enzymes are proteins coded for by DNA. It's circular. You need proteins to handle DNA, but you need DNA to make proteins. The likely solution to this chicken and egg problem is RNA. RNA can do both jobs. It can store information, like DNA, and it can catalyze reactions, like proteins. It's not as good at either job as the specialized molecules that eventually replaced it, but it's good enough. And critically, RNA can copy itself. In the 1980s, Thomas Keck and Sidney Altman independently discovered that some RNA molecules have catalytic activity. They called them ribozymes, RNA enzymes. The discovery earned them the Nobel Prize and revolutionized our understanding of life's origins, because it meant RNA could potentially have existed in a world before proteins, before DNA, serving as both information storage and catalytic machinery. The idea, now called the RNA world hypothesis, is that early life was based entirely on RNA. RNA molecules that could catalyze reactions, including the reactions needed to copy other RNA molecules. A network of interacting RNAs, some catalyzing the synthesis of others, some binding to small molecules and speeding up metabolic reactions, all of it encoded in sequences that could be copied and passed on. It's not as far-fetched as it sounds. Researchers have created RNA molecules in the lab that can catalyze their own replication, at least partially. They found RNAs that can splice themselves, cut and paste genetic material, even build peptide bonds between amino acids. RNA is versatile in a way that DNA and proteins individually are not. And RNA is still central to modern biology. The ribosome, the molecular machine that builds proteins, is mostly RNA. Transfer RNAs carry amino acids to the ribosome. Messenger RNA carries genetic information from DNA to the ribosome. Even some regulatory systems use small RNA molecules to control gene expression. It's as if modern life still carries traces of its RNA-based origins, like an old foundation beneath a renovated building. But whether or not there was an RNA world, the key point is this. Once you have molecules that can store information and that information can be copied, you have heredity. And once you have heredity, variation becomes meaningful. If all protocells were identical, it wouldn't matter which ones survived and which ones didn't. But if each protocell has a slightly different set of RNA sequences, a slightly different collection of catalytic molecules, then some will be more efficient than others, some will replicate faster, some will be better at scavenging resources, some will be more stable, more resistant to damage. The more successful ones will produce more descendants, the less successful ones will fade away. And if copying isn't perfect, if mutations occur, if sequences occasionally change, then new variants appear. Most changes make things worse, but occasionally one makes things better. That's natural selection, And once it kicks in, once you have hereditary variation and differential survival, evolution becomes inevitable. The system starts optimizing itself, not through design, but through the relentless filtering of what works. Evolution doesn't have foresight. It can't plan ahead, can't see where it's going. It only responds to immediate pressures, immediate advantages. A mutation that helps right now gets preserved. One that doesn't, or that actively harms, gets eliminated. The system is entirely reactive, entirely blind. And yet, over time, it produces outcomes that look designed. Molecular machines of staggering complexity. Systems so intricately coordinated they seem intentional. This is the paradox at the heart of biology. The appearance of design without a designer. The emergence of purpose from purposeless processes. Take the bacterial flagellum, one of the most sophisticated molecular machines we know. It's a rotary motor, literally a spinning propeller that drives bacterial movement. There's a long filament, the propeller itself, attached to a hook, which connects to a motor embedded in the cell membrane. The motor is made of multiple protein, rings. Protons flow through channels in the motor, and that flow causes the rings to rotate. The rotation spins the hook, which spins the filament, which pushes against the surrounding fluid, propelling the bacterium forward. It can spin at hundreds of revolutions per second. It can reverse direction almost instantly. It can respond to chemical gradients, steering the bacterium toward nutrients or away from toxins. It's a fully functional rotary engine, built entirely from proteins that self-assemble, powered by proton gradients, controlled by sensory systems that feed back into the motor. For decades, this was held up as an example of irreducible complexity, a system so intricate that removing any part would make it non-functional, supposedly proof that it couldn't have evolved gradually. But that argument misunderstands how evolution works. You don't need to build the flagellum all at once. You just need each intermediate step to be useful for something. And when researchers looked closely at the proteins that make up the flagellum, they found many of them have homologs, similar proteins used in other systems. Some of the motor proteins are related to proteins in secretion systems, molecular pumps that export proteins across membranes. The filament proteins have similarities to pili, hair-like structures some bacteria use to adhere to surfaces or exchange DNA. What this suggests is that the flagellum wasn't invented from scratch. It was cobbled together from parts that already existed, that were already being used for other purposes. A secretion pump got repurposed into a motor. Structural proteins got elongated and modified into a filament. Regulatory systems got co-opted to control rotation. Each change, each addition, provided some benefit. Maybe just a little more efficiency, a little more motility, a slightly better ability to navigate. Over millions of generations, small improvements accumulated. Proteins duplicated, mutated, took on new roles. Systems that started simple became complex, and eventually you end up with a rotary motor built from dozens of different proteins, all of them working together with precision that looks engineered but emerged through the blind, iterative process of mutation and selection. This is how all molecular machines evolved, not through sudden invention, but through gradual modification of existing systems. Evolution is a tinkerer, not an engineer. It doesn't start with a blueprint. It works with whatever's already there, making small changes, keeping what works, discarding what doesn't. And because evolution works this way, molecular machines often carry traces of their history. Proteins that do similar jobs in different organisms are usually related. They share a common ancestral protein that duplicated and diverged. Sometimes you can trace entire pathways back through evolutionary time, seeing how a simple two-step reaction got expanded into a 10-step pathway as new enzymes were added, as intermediate steps became more efficient. The result is a living archive. Every cell carries within it the molecular history of life on Earth, written in protein sequences, metabolic pathways, genetic codes. The machinery that runs life today is built on machinery that ran life billions of years ago, modified, refined, but fundamentally connected. But here's what's easy to miss when you focus on individual machines. None of them work alone. The bacterial flagellum only functions because the cell produces the right proteins, assembles them in the right order, generates the proton gradient that powers the motor, integrates sensory information to control rotation. It's embedded in a network of dependencies, a web of interacting systems. This is true at every level. Proteins depend on other proteins to fold correctly, to be transported to the right location, to be activated or degraded at the right time. Metabolic pathways depend on membrane transporters to bring in substrates and remove products. Transporters depend on energy generated by metabolism. It's circular, interconnected, a network where every component depends on others. And this raises a profound question. How do you evolve a network? How do you change one part without breaking the whole system? The answer is redundancy and modularity. Living systems aren't optimized to the point of fragility. They have backup pathways, alternative routes, multiple ways to accomplish the same task. If one enzyme gets damaged, sometimes another enzyme with overlapping specificity can compensate. If one metabolic pathway is blocked, sometimes another pathway can take over. This redundancy provides a buffer. It means mutations don't immediately crash the system. A change to one protein might reduce its efficiency, but if there are backup systems, the organism survives. And if the change also happens to improve something else, maybe the protein now catalyzes a slightly different reaction, or works better under certain conditions, then natural selection can favour it. Modularity helps too. Molecular machines are often built from domains, semi-independent functional units that can be mixed and matched. A protein might have a domain that binds DNA, another domain that catalyzes a chemical reaction, and a third domain that responds to regulatory signals. These domains can be shuffled, duplicated, combined in new ways. Evolution doesn't have to invent entirely new proteins from scratch. It can recombine existing parts. According to research on protein evolution, many modern proteins are mosaics, patchworks of domains that originated separately and got fused together over evolutionary time. This modular architecture makes innovation easier. A new combination of domains can produce a protein with a new function, built from parts that already worked individually. So evolution proceeds by tinkering, duplicating genes, shuffling domains, tweaking sequences, keeping beneficial changes, discarding harmful ones. And, because the system is interconnected, changes in one place can have ripple effects elsewhere. A mutation that improves one enzyme might increase the flux through a metabolic pathway, which might create a selective pressure to improve the next enzyme in the chain, which might open up opportunities for further modifications. The network evolves as a whole, not just as isolated components. And over time, the network becomes more sophisticated, more pathways, more regulation, more integration, more capability. But there's a limit to how complex a single cell can become. There's only so much you can pack into a microscopic volume, only so many reactions you can run simultaneously before you start getting interference, conflicts, unwanted cross-reactions. The next major transition in the history of life, the jump from simple cells to complex cells, happened when one cell engulfed another and instead of digesting it, kept it. Endosymbiosis. The origin of mitochondria and chloroplasts, the organelles that generate energy in modern eukaryotic cells. Mitochondria still carry their own DNA, a small circular chromosome that encodes some of the proteins needed for energy production. It's a remnant of their bacterial ancestry. About 2 billion years ago, an ancestor of modern bacteria capable of aerobic respiration got internalized by another cell. Maybe it was captured. Maybe it was a symbiotic relationship, but instead of being broken down, the internalized bacterium survived, continued generating energy, and provided that energy to the host cell. The host benefited from the extra energy. The internalized bacterium benefited from a stable environment and access to nutrients. Over time, most of the bacterium's genes got transferred to the host cell's nucleus. The bacterium became integrated, became an organelle, became mitochondria. It's one of the most consequential events in the history of life. With mitochondria producing energy efficiently, cells could afford to be larger, more complex, more metabolically active. They could support bigger genomes, more proteins, more sophisticated regulatory systems. The jump from prokaryotes, simple cells without internal compartments, to eukaryotes, complex cells with nuclei and organelles, was enabled by this merger, this incorporation of a molecular machine into a larger system. And once you have complex cells, once you have the energy budget to support larger size and greater complexity, entirely new possibilities open up. Cells can specialize. They can differentiate into different types, each with its own set of molecular machines, each performing different functions. They can form tissues, organs, bodies. They can become multicellular. But even in a multicellular organism, even in something as complex as a human body, the fundamental units of life are still cells, and the fundamental workers inside those cells are still molecular machines. Proteins folding into shapes that perform work, enzymes catalyzing, reactions, transporters moving molecules across membranes, motors generating motion. The same machinery that started billions of years ago in the first protocells, now elaborated, diversified, specialized, but still operating on the same principles. Shape determines function, chemistry drives motion, information gets copied, inherited, modified. Networks of machines coordinate, regulate, sustain themselves, and all of it emerges from the properties of atoms and molecules, from the laws of physics and chemistry playing out over deep time. There's something almost paradoxical about molecular machines. They operate at a scale where the rules seem different, where intuition breaks down, where the distinction between deterministic mechanics and random chance blurs. At the macroscopic scale, the scale of cars and clocks and conveyor belts, machines are predictable. Push a lever and it moves, turn a gear and it rotates. The motion is smooth, continuous, governed by forces large enough that thermal noise, the random jiggling of atoms, doesn't matter. But shrink down to the nanoscale, to the size of proteins, and everything changes. Thermal noise is no longer negligible, it's dominant. Molecules aren't gliding smoothly through space. They're buffeted constantly by collisions with surrounding water molecules, knocked this way and that in a chaotic dance called Brownian motion. A myosin motor pulling on an actin filament isn't pushing with steady force. It's jerking, stuttering, fighting against random kicks from the surrounding fluid. The motion only looks smooth when you average over thousands of cycles, thousands of motors. Individual events are stochastic, probabilistic, governed as much by chance as by chemistry. And yet, somehow, it works. The machines function despite the noise, or perhaps because of it. Thermal fluctuations aren't just obstacles to overcome, they're part of the mechanism. Proteins don't have to supply all the energy for conformational changes. Thermal energy is constantly pushing them into different shapes. The protein just has to bias the probabilities, make certain states more stable than others, guide the random walk in a particular direction. This is a fundamentally different kind of engineering than anything humans have built. We design machines to resist noise, to operate deterministically. Molecular machines embrace noise, harness it, use it as an energy source. They're probabilistic engines, ratchets that convert random motion into directed work. The classic example is the Brownian ratchet, a thought experiment that helped clarify how molecular motors can move in one direction, despite being constantly battered by thermal fluctuations. Imagine a gear with asymmetric teeth connected to a pole, a little lever that can catch the teeth and prevent backward motion. If the gear is jiggling randomly due to thermal noise, you might think the pole would let it ratchet forward but not backward, producing net motion in one direction. But that doesn't work in equilibrium. If everything's at the same temperature, the pal is also jiggling thermally, sometimes allowing backward motion. There's no net movement, no violation of the second law of thermodynamics. Molecular motors get around this by operating out of equilibrium. They're coupled to energy sources, usually ATP hydrolysis, that break the symmetry. The energy input biases the probabilities, makes forward steps more likely than backward steps. The motor still jitters, still experiences thermal noise, but on average it moves forward. According to work by physicists like Dina Stumian and Peter Henge in the 1990s and 2000s, this kind of Brownian motor is exactly how many molecular machines operate. They don't fight against thermal fluctuations, they ride them, using chemical energy to bias random motion into productive work. This has profound implications. It means molecular machines don't need to be perfectly rigid. They can be floppy, flexible, constantly shifting between different conformations. The important thing isn't the structure at any given instant, but the statistical ensemble of structures, the probabilities of being in different states. It also means molecular machines can be incredibly efficient. Because they're small and operate in a regime where thermal energy is significant, they can convert chemical energy into work with efficiencies that approach thermodynamic limits. Macroscopic engines, car engines, power plants, waste most of their energy as heat. Molecular motors can operate at 70%, 80%, even 90% efficiency, extracting nearly every available joule of useful work from the chemical reactions that power them. And they do it at room temperature, in water, using reactions that are gentle enough not to destroy the machines themselves. No combustion, no high-pressure steam, no moving parts that wear out and need replacement. Just proteins cycling through conformational changes, driven by chemistry, stabilized by the same forces that made them fold in the first place. But efficiency isn't the only remarkable thing. There's also speed and precision. Some molecular machines operate astonishingly fast. DNA polymerase, the enzyme that copies DNA, can add 1,000 nucleotides per second, with error rates of about 1 mistake per billion bases. That's not just fast, that's fast and accurate. The enzyme has to select the correct nucleotide from a pool of 4 different types, match it to the template strand, form the chemical bond to add it to the growing chain, and move on to the next position, all in a millisecond. All while maintaining fidelity high enough that genetic information stays intact across generations. How does it achieve that accuracy? Through a combination of mechanisms. Base pairing, adenine with thymine, guanine with cytosine, provides initial selectivity because the shapes of correct pairs fit better in the enzyme's active site than incorrect pairs. But that alone isn't enough. The enzyme also has proofreading. If a wrong nucleotide gets incorporated, the enzyme can detect the mismatch, reverse direction, cut out the incorrect nucleotide, and try again. Some polymerases even have a separate proofreading domain, a 3:5 exonuclease activity that checks behind the growing chain, excising errors. The enzyme toggles between polymerization mode and exonuclease mode, building and checking, ensuring high fidelity through redundant quality control. It's a molecular assembly line with built-in quality assurance, operating at speeds that would make any human manufacturing process jealous, with error rates so low that replication can proceed for billions of bases without a mistake. And DNA polymerase isn't unique. Ribosomes translate genetic information into proteins with comparable speed and accuracy. They read messenger RNA 3 bases at a time, select the corresponding transfer RNA carrying the right amino acid, catalyze peptide bond formation, and move to the next codon. 20 amino acids per second in bacteria, a bit slower in eukaryotes, but still impressively fast given the complexity of the task. Like DNA polymerase, ribosomes have proofreading mechanisms. Initial selection of the correct tRNA is partly based on base pairing between the tRNA anticodon and the mRNA codon. But the ribosome also checks the match, and if it's incorrect, the tRNA is more likely to dissociate before peptide bond formation. Even after the bond forms, the ribosome can detect mismatches and eject incorrect tRNAs, though at that point the amino acid is already incorporated into the growing chain, so the error can't be reversed, only reduced in frequency. The result is translation accuracy on the order of 1 mistake per 10,000 amino acids. Not as precise as DNA replication, but still high enough that most proteins fold correctly most of the time. This combination of speed and accuracy is what allows living systems to be so reliable. Your cells are constantly synthesizing thousands of different proteins, copying DNA during cell division, transcribing genes, translating mRNA. Any of these processes could go wrong, mutations could accumulate, proteins could misfold. But the machinery is good enough, fast enough, accurate enough, that errors are rare, and when they do occur, there are often backup systems, repair mechanisms, quality control checkpoints. The molecular machines aren't perfect. Evolution doesn't produce perfection. It produces good enough, and then refines toward better. But the machines that exist today are the result of billions of years of refinement, billions of generations where variants that worked a little better outcompeted variants that worked a little worse. And the refinement continues. Every organism alive today carries molecular machines that are slightly different from the machines in its ancestors, and slightly different from the machines in other organisms. The bacterial flagellum isn't the same in all bacteria. Some spin clockwise, some counterclockwise. Some have multiple filaments, Some have different numbers of proteins in the motor. The diversity reflects different evolutionary paths, different solutions to the same basic problem of how to move through fluid. Even within a single organism, molecular machines aren't uniform. Human cells express thousands of different proteins, many of them variants of the same basic machine, adapted for different contexts. There are multiple types of myosin, each specialized for different kinds of movement. Multiple types of kinesin, molecular motors that walk along microtubules, transporting cargo within the cell. Multiple types of ATP synthase, adapted for different energetic demands. This functional diversity, this proliferation of variations on common themes, is one of the signatures of life. It's what allows organisms to adapt to different environments, to occupy different niches, to specialize and exploit opportunities. And it all traces back to the same source, the ability of molecular machines to evolve, to change through mutation and selection, to diversify and refine over time. But evolution doesn't just modify individual machines, it modifies the relationships between them, the networks, the systems, the ways machines coordinate and regulate each other, and that's where things get truly complex. Because a cell isn't just a collection of molecular machines, it's a society of them, interacting, communicating, organizing themselves into functional hierarchies. Think about what a cell actually is. It's not a factory with a central office issuing commands. There's no CEO nucleus telling the cytoplasm what to do. There's no top-down control, no master plan being executed. And yet, the cell functions with extraordinary coordination. Thousands of different molecular machines working simultaneously, somehow all contributing to the same overall goal: keeping the cell alive, building what needs to be built, breaking down what needs to be broken down, responding to changes, adapting. This coordination emerges from the bottom up, from local interactions between molecules, from feedback loops and regulatory networks, from the fact that molecular machines don't just perform tasks, they respond to context. An enzyme doesn't catalyze reactions at a constant rate regardless of conditions. Its activity depends on substrate concentration, on the presence or absence of regulatory molecules, on pH, on temperature, on the availability of cofactors. The enzyme is reading its environment, and its behavior changes accordingly. Multiply that across thousands of enzymes, and you get a system where activity self-regulates. High concentrations of a product inhibit the pathway that produces it. Low concentrations remove that inhibition. The system automatically adjusts to maintain balance, not because anything is monitoring the whole system, but because each component is responding to local conditions in ways that happen to stabilize the whole. This is distributed intelligence. Not intelligence in the sense of consciousness or thought, but in the sense of adaptive problem solving, of a system that responds appropriately to challenges without centralized coordination. And the more you look at cellular systems, the more you see this pattern. Gene expression is regulated by transcription factors, proteins that bind to DNA and control whether genes get transcribed. But what controls the transcription factors? Other proteins. And what controls those? Signals from outside the cell, metabolic states, feedback from the very pathways the genes encode. It's a web of mutual regulation, where every component is both controller and controlled. Even cell division, one of the most carefully orchestrated events in biology, operates through distributed control. There's a checkpoint system that prevents the cell from dividing, until DNA has been replicated, until chromosomes are properly aligned, until everything is ready. But the checkpoint isn't a single watchdog protein. It's a network of proteins monitoring different aspects of the process, all feeding into the decision of whether to proceed. If DNA is damaged, checkpoint proteins halt the cycle and activate repair systems. If chromosomes aren't properly attached to the spindle, different checkpoint proteins prevent separation. The cell moves forward only when multiple independent checks all give the green light. It's consensus decision-making at the molecular level, and what's striking is how robust this is. You can knock out individual proteins, disrupt specific pathways, and often the cell compensates. Backup systems kick in, alternative routes get activated, the network rewires itself on the fly, maintaining function even when components fail. This robustness isn't accidental, it's been selected for. Organisms whose cellular machinery was fragile, where a single mutation or a single environmental stress could crash the system, didn't survive. The ones that did survive were the ones with redundancy, with flexibility, with networks resilient enough to tolerate perturbation. But resilience has limits. Push too hard, disrupt too many systems, and the network can't compensate. The cell dies. And in multicellular organisms, dead cells are a problem. They need to be removed, recycled, replaced. So cells evolved mechanisms for self-destruction. Apoptosis, programmed cell death. A cascade of molecular events where the cell systematically dismantles itself, breaking down its own DNA, its own proteins, packaging everything into neat little vesicles that neighbouring cells can engulf and digest. It sounds grim, but it's essential. During development, programmed cell death sculpts tissues, removes temporary structures, carves out spaces. The webbing between embryonic fingers disappears through apoptosis. Excess neurons in a developing brain get pruned. The system builds more than it needs, then removes what's unnecessary, like a sculptor chipping away stone. Even in adults, apoptosis maintains balance. Cells that accumulate too much damage, that fail the quality control checks, that show signs of becoming cancerous, they're supposed to trigger their own death, preventing potential harm to the organism. When that system fails, when cells stop responding to apoptosis signals, you get uncontrolled growth, you get cancer. So even death, even destruction, is part of the machinery. It's regulated, controlled, integrated into the larger system. Molecular machines not just for building and maintaining, but for dismantling when necessary. This is the full picture of what molecular machines are, what they do, how they function in the context of living systems. They're not isolated tools, they're components of networks, embedded in feedback loops, responsive to context, coordinated through distributed control. They build, they maintain, they regulate, they repair, and when necessary they dismantle. And all of this emerges from chemistry, from atoms arranging themselves into shapes that perform work, from shapes that change in response to their environment, from networks of reactions that self-organize, that evolve, that persist despite the constant pull of entropy. Life is molecular machinery, not metaphorically, not as an analogy, literally. The things happening inside every cell are mechanical processes, molecules moving, binding, catalyzing, transporting, signaling. The fact that the machines are built from proteins instead of metal, that they operate in water instead of oil, that they're powered by ATP instead of electricity, doesn't make them less mechanical. It makes them a different kind of mechanical, adapted for the nanoscale environment, where thermal noise is significant, where precision is measured in angstroms, where speed is measured in microseconds. And the profound thing, the thing worth sitting with, is that these machines built themselves. No designer drew up the plans, no engineer assembled the parts. The machines emerged from the properties of matter, from the way atoms interact, from the way molecules fold and bind and react. Evolution discovered them. Not through foresight, not through intention, but through the blind, iterative process of variation and selection. Molecules that happened to fold into useful shapes survived. Networks that happened to self-sustain persisted. Systems that happened to copy themselves with variation evolved into more sophisticated systems, which evolved into cells, which evolved into organisms, which evolved into the entire biosphere. The machines that built themselves didn't just create life, they created the conditions for complexity, for diversity, for intelligence, for consciousness. Every thought you've ever had, every movement you've ever made, every sensation you've ever felt, all of it rests on molecular machinery, on proteins cycling through conformational changes, on ion gradients maintained by pumps, on signals propagating through networks of neurons built from trillions of cells, each one running its own set of molecular machines. We're not separate from this machinery. We're made of it. Our existence is entirely dependent on proteins folding correctly, on enzymes catalyzing reactions, on transporters moving molecules, on motors generating motion. Take away the molecular machines, and there's nothing left. No metabolism, no structure, no life. So when we ask what life is, when we try to define the boundary between living and non-living, maybe the answer is simpler than we thought. Life is what happens when molecular machines coordinate, when chemistry organizes itself into self-maintaining, self-replicating, evolving systems. when matter learns to build, to maintain, to adapt, using nothing but the rules physics and chemistry already provided. The machines built themselves, and in building themselves they built everything. What if the strangest, most alien landscapes in the universe aren't out there among the stars, but right here, in a single drop of seawater? Magnify it, and suddenly you're in another world. Shapes of glass, lattices of impossible geometry, shimmering bodies that look like spacecraft adrift in the black. These are diatoms, plankton, comb jellies. Creatures so translucent and strange they look like something that should be floating between galaxies, not swimming off the coast of Earth. Their bodies are mostly water, their outlines sculpted by rules they never chose. Rules that somehow make them look like the cosmos itself. And here's where it gets strange. The patterns you see under the microscope don't stay there. Zoom out and you start noticing the same forms everywhere. Lightning forks like the branches of a tree. A river delta mirrors the veins in a leaf. Your lungs branch in the same fractal rhythm as coral reefs. Even galaxies spiral with the same curve you can find on a seashell you pick up at the beach. Coincidence? Or is the universe hinting at something bigger? Scientists have long noticed this echo, this family resemblance between things that, at first glance, seem unrelated. The mathematician Benoit Mandelbrot gave us the word fractal to describe shapes that repeat at different scales, from the outline of a coastline to the folds of a brain. The biologist Darcy Thompson once argued that everything is the way it is because it became that way, meaning that natural law, not design, is what sculpts the geometry of life. And physicist Adrian Baden has proposed the constructal law, that flow systems, whether rivers, trees or blood vessels, naturally evolve to find the most efficient paths. It's as if the universe is economical. It keeps arriving at the same solutions, the same motifs: branch, spiral, network repeat. Which raises a question with enormous stakes. If these shapes are everywhere, from the tiniest cells to the largest galaxies, is life itself inevitable? Could the universe be set up in such a way that complexity, order, and maybe even consciousness are not accidents, but habits of matter, written into the equations from the start? That's what this journey's about. The blueprint. Not DNA, though we'll get there, but a deeper code, a physics of form, a set of patterns that recur across scale after scale. We'll start small, where the patterns first reveal themselves, inside the microscopic worlds that hide in every raindrop and tide pool. We'll watch how simple rules of chemistry generate stripes and spots, how spirals emerge from steady growth, and how branching keeps showing up whenever something needs to spread or deliver. Then we'll step back. We'll look at deserts that self-organize into waves of marching dunes, snowflakes that sculpt themselves in mid-air, and galaxies that arrange themselves into a cosmic web that looks eerily like the neurons firing inside your head. And from there, we'll stretch outward, to the alien. Deep-sea creatures that glow like living glass will become mirrors for what life might look like on Europa or Enceladus. Hydrothermal vents on Earth will guide us to plumes bursting from icy moons. We'll ask, if physics is truly universal, should we expect alien life to follow the same blueprint we see here? Finally, we'll come home to you, to the fractal networks inside your lungs, the patterns of your heartbeat, the trillions of microbes that make up your second self, to the fact that your body is not an exception to the blueprint, it is one of its most elaborate expressions. This isn't a story about trivia, it's about something deeper. Why the universe keeps writing the same shapes into everything, everywhere, at every scale. And whether those shapes can tell us something about why we're here, what life is, and how much of it might be waiting for us beyond the sky. So look again at that drop of seawater. alien creatures drifting in the dark, carrying secrets older than DNA. If you can learn to read their shapes, you're not just learning about biology, you're glimpsing the handwriting of the cosmos itself. Take a drop of seawater. To the human eye it looks empty, clear, unremarkable, but magnify it and you enter a universe. Inside, translucent bodies drift like spaceships, latticed shells spin like glass ornaments, and shimmering forms pulse with a life that looks more alien than anything in science fiction. These are plankton and diatoms, comb jellies and salps, creatures so otherworldly that if you saw them in a film about deep space you wouldn't question it. Many are almost entirely transparent, their tissues little more than scaffolds for the faintest traces of colour and light. Some can refract rainbows through their gelatinous ribs, others glow with bioluminescence like living lanterns. A chain of salps can stretch for meters, yet each individual animal is almost invisible. These delicate architectures are as real, as essential, as the fish we eat or the trees that give us shade, and yet most people never realise they exist. Then there are diatoms, single-celled algae that build their bodies out of glass. Their silica shells are among the most intricate natural designs on Earth. Hexagons and spirals, lattices and filigrees, patterns repeated with microscopic precision. Each one looks manufactured, as if sculpted by an artist with infinite patience. And yet, diatoms are not art. They are survival. Their ornate glass houses strengthen them against predators and give them a way to thrive in oceans, lakes and rivers. Diatoms alone may produce up to 20% of the oxygen you breathe, every fifth breath, courtesy of living glass. And then there are the neurons inside your head. Under the microscope they weave into glowing networks, electrical storms flashing across their branches. Place an image of neurons next to a map of galaxies, and the resemblance is uncanny. In both, filaments connect dense clusters, forming webs of energy and information. They operate at scales so different, micrometers versus millions of light years, that the comparison seems absurd, and yet the geometry is there. Neurons and galaxies both solve the same problem: how to connect many nodes without wasting energy. This is the first sign that biology and physics may be rhyming with each other. Patterns echo across scales, not because of coincidence, but because the same rules, efficiency, flow, distribution, keep arriving at the same solutions. The brain doesn't look like a galaxy because it was designed to. It looks like a galaxy because both are following a blueprint written in physics. Even within a single cell, the architecture feels cosmic. Mitochondria busily churn out energy, like little stars. Microtubules stretch and contract like highways. Vesicles move cargo along molecular rails, each protein folding into machines precise enough to rival any human factory. None of these parts know the whole picture. None of them intend to make a cell work. And yet together, they form an emergent order, a city in miniature, as alive and intricate as anything we see at human scale. Scientists call this self-organization, the ability for complex systems to arise from simple interactions. Birds flock without a leader, fish school without a plan, sand dunes align themselves without a designer. Likewise, cells coalesce into tissues, tissues into organs, and organs into organisms. Complexity, once thought improbable, turns out to be natural. When conditions are right, matter doesn't just sit idle, it organizes. This is why microscopic life often feels alien. We are not used to thinking of complexity as inevitable, but under the microscope, you can watch it happening in real time. Order emerging from randomness, patterns arising from chaos. A single-celled organism with no brain and no awareness can create beauty that looks like art, but is only physics playing itself out. And these organisms are not rare. A litre of seawater may hold billions of them. Together they form the base of the food chain, the lungs of the planet, and the invisible foundation of life. Without them, oxygen would vanish, ecosystems would collapse, and Earth would be unrecognizable. The alien-looking world inside a drop of water is not exotic, it's essential. What makes this more profound is that the same visual motifs repeat elsewhere. Look at coral reefs, colonies of tiny polyps building calcium skeletons that resemble branching trees. Look at slime molds, which expand across surfaces in networks that eerily resemble both roadmaps and neural networks. At every scale, life converges on the same themes: branching, spiraling, connecting. And so the microscope becomes a telescope turned inward. It reveals that the blueprints we associate with galaxies and planets are already here, already at work in creatures too small to see. The alien is not distant, it's everywhere. By beginning here with the microscopic, we establish the first principle of the blueprint. When energy flows and matter is allowed to move, patterns emerge. Cells look like cities, neurons look like galaxies, plankton look like spacecraft, not because they are copies of one another, but because they are all following the same underlying rules. And that is the real marvel. The fact that life, from its smallest scales, already resembles the universe it inhabits. Stand in a forest and look up. Branches split into branches which split again into finer twigs, tracing lines into the sky. Now look down. Roots mirror the canopy, forking their way through the soil in search of water and nutrients. Then leave the forest and look at a river delta from above. Countless veins carry water to the sea in the same repeating rhythm. Look at lightning crackling across the sky. It follows the same script. This is nature's obsession with branching. It's not just a coincidence, and it's not mere beauty. It's efficiency. Whenever something needs to distribute or collect, blood, water, electricity, sap, the universe defaults to branching. It's the fastest way to cover ground while minimizing effort. Engineers call this the constructal law, the idea that flow systems evolve to move energy and material with greater ease. Nature discovered it first, but branching is only one line in the blueprint. Another is the spiral. Spirals are everywhere once you notice them. The shell of a nautilus curls according to a logarithmic spiral, each new chamber expanding proportionally to the last. Sunflower seeds pack themselves into spirals that follow Fibonacci numbers, maximizing space and light. Hurricanes swirl with the same geometry, as do spiral galaxies stretching their arms across millions of light years. Different scales, different materials, the same curve. The spiral is mathematics embodied. Growth that is steady and turning creates this shape again and again. Plants don't need to know math. Physics does the work for them. By growing at angles that minimize overlap, seeds and leaves capture the most sunlight, and storms distribute force evenly. The golden ratio and Fibonacci sequences are not mystical. They're practical solutions written into the fabric of growth. Then there are patterns that seem to appear from nowhere, as if matter is painting itself. Spots on a leopard, stripes on a zebra, ripples on a tropical fish. In 1952, mathematician Alan Turing proposed a simple explanation: reaction diffusion. When two chemicals spread through tissue, one activating and the other inhibiting, the interplay can generate repeating motifs. It doesn't take a painter to draw the stripes, just chemistry running its equations. Today, computer simulations of Turing's model can produce everything from leopard spots to zebra stripes, even the mottling on seashells. His idea, once speculative, has since been validated in biology. Fish patterns and mammalian coats often follow the very logic Turing imagined. But the script isn't confined to living things. Look at the desert. Sand dunes arrange themselves into ridges and waves, marching slowly under the force of the wind. From the ground, they seem chaotic, but from the air, patterns emerge. Parallel lines, crescents, stars. Each dune follows simple rules of grain movement and airflow, yet together they create order across landscapes. Snowflakes tell a similar story. A snow crystal begins as a tiny hexagon of ice. As water vapor attaches, branches sprout. Temperature, humidity, and airflow dictate how those branches grow, and because no two paths are identical, every snowflake is unique. Yet the six-fold symmetry never changes. It's an emergent geometry, sculpted not by design, but by physics. And then there are fractals, the mathematics of roughness, the geometry of repetition. Coastlines look jagged no matter how closely you zoom in. Clouds billow with repeating edges. Mountains from a distance look like triangles, but up close they break into smaller ridges, and those into finer crags. Benoit Mandelbrot coined the term fractal. to describe these shapes that repeat across scales. What once seemed irregular and messy was suddenly quantifiable. Nature, it turns out, has a preference for fractals because they maximize surface area and interaction. Lungs, roots, river networks, all fractals, because they need to exchange as much as possible with their environment. What all these examples show is that nature doesn't improvise, it reuses. It discovers efficient answers and applies them across domains, whether it's moving water through soil, distributing nutrients through veins, or scattering lightning across the sky. And here lies the deeper point. These patterns aren't just beautiful accidents. They're consequences of physics. Branching happens because it moves things faster. Spirals appear because growth under rotation follows mathematics. Stripes emerge because diffusion under constraints produces regular intervals. Snowflakes crystallize because water obeys symmetry at freezing temperatures. The universe isn't copying itself. It's following rules. Once you begin to see these patterns, the world becomes uncanny. You notice how your own veins echo the branches of a tree, how a river delta mirrors the structure of your lungs. You realize that the nautilus shell and the hurricane are distant cousins in geometry, that a snowflake falling on your hand has something in common with the stripes of a tiger and the ripples of sand in a desert. Patterns in nature are not decoration. They are signatures of the blueprint. And once you can read them, you begin to suspect that life, our lives, are part of a much wider story. Up to this point, we've looked at patterns as if they were curiosities, beautiful shapes the universe seems to favour. But here's the truth. They're not optional. They are demanded by physics. They are the inevitable outcomes of matter and energy in motion. Let's return to branching. Why does lightning split and fork rather than strike in a straight line? Because electricity, like water, seeks the easiest route through resistance. The moment one path starts forming, the electric field reshapes itself and new branches sprout to find alternate routes. It's not art, it's optimization, solving the same problem millions of times in fractions of a second. The same principle governs the branching of your lungs. To get oxygen into your blood efficiently, airways must reach as much surface area as possible in a limited volume. Branching is the answer nature keeps circling back to, whether it's moving water, air, or electrons. Engineers like Adrian Bajan call this the constructal law. Flow systems evolve so that they can flow more easily. In other words, designs are not just lucky, they're forced by the physics of efficiency. Order can also rise in places we'd least expect. In the mid-20th century, chemist Ilya Prigogine studied what he called dissipative structures, systems that create order by exporting chaos. A cell, for example, isn't a little miracle of order-defying entropy. It survives precisely because it feeds entropy into its environment. By burning energy and releasing waste, it buys itself a pocket of organization. The greater the imbalance, the more opportunity for order to appear. In this sense, life isn't opposed to the second law of thermodynamics. It's a direct consequence of it. Think about a pot of water on a stove. Heat it from below and at first the molecules just jiggle faster, but push it harder and suddenly the system flips. Convection cells form. Columns of hot water rise, cooler water sinks, and hexagonal patterns appear on the surface. No one designed them. They're the system's way of handling the flow of energy. The same thing happens in the atmosphere as air currents create storms. It's physics insisting on order as a way of balancing imbalance. This is self-organization, and it's everywhere. Sand dunes align under the push of the wind. Bacteria colonies expand in fractal patterns. Starlings flock into coordinated swarms without a leader. Each bird follows simple rules. Keep close, don't crash, copy direction, and the result is an airborne ballet. Complexity arises not from command, but from repetition and feedback. What makes this profound is that it implies complexity is not fragile. It's inevitable. Given energy, given imbalance, given time, matter organizes, not always into life, but often into forms that echo it. Now stretch this principle outward. Galaxies themselves spin because gravity collapses clouds of gas into disks. Within those disks, spiral arms form not because they are structures in stone, but because waves of density sweep through, compressing gas, triggering star formation, moving on like ripples in a pond. A galaxy's spiral arms are traffic jams of stars, patterns emerging from gravity's relentless pull. Or look inward again, the rhythmic beat of your heart. At its core is an electrical pacemaker, cells firing in repeating cycles. The rhythm is not imposed. It emerges from the interplay of ion channels and feedback loops. Your heartbeat is physics at work, repeating itself with just enough irregularity to stay alive. Too perfect, and it signals danger. Too chaotic, and it collapses. Life balances on the edge of order and disorder. So the blueprint of life isn't a set of instructions etched in stone. It's a tendency, a bias in the universe towards certain shapes whenever energy moves. Branches to spread, spirals to grow, waves to oscillate, webs to connect. These patterns are the solutions physics keeps rediscovering, and that raises a deeper thought. If physics makes complexity likely, then maybe life itself, what we think of as improbable, might actually be common. Not common in its details, not common in its DNA, but common in its forms. Wherever there is flow, wherever there is imbalance, wherever matter is nudged from stillness, the blueprint may be waiting to write itself again. This is where awe becomes unsettling. If life isn't a cosmic accident, then we may not be rare at all. The universe may be filled with different versions of the same habits, branching organisms, spiraling growths, networks exchanging energy and information. Not copies of Earth's life, but siblings in physics family, which leaves us with a haunting possibility. Maybe the universe doesn't just allow life, it leans toward it. Maybe the blueprint doesn't only describe what is, but predicts what will be. When you look at the night sky, it feels infinite, scattered, random. Stars pinned against blackness, galaxies swirling without order. But zoom out far enough and the chaos resolves into structure. Astronomers mapping the universe discovered something astonishing. Galaxies are not sprinkled evenly like dust, but strung along filaments, woven into a vast web that spans billions of light years. This cosmic web is the universe's skeleton. Galaxies cluster at the nodes, connected by glowing threads of gas and dark matter. Between them lie vast voids, deserts of space almost entirely empty. On the grandest scales, the universe looks less like a spray of stars and more like a neural network. And here the comparison becomes uncanny. Place an image of the cosmic web beside a map of the human brain, and the resemblance is undeniable. Both are webs of filaments connecting dense hubs. Both rely on the same balance. Too few connections and the network collapses. Too many, and it becomes inefficient. Statistically, the distribution of links in both systems follows similar laws. One is made of stars, the other of neurons separated by 27 orders of magnitude in scale. And yet the blueprint rhymes. Is it just our pattern-hungry minds drawing connections? Maybe. A galaxy cluster is not a neuron. Gravity and dark matter are not synapses. But when systems separated by incomprehensible scales converge on similar architectures, it begs the question, are they solving the same kind of problem? How to connect, how to distribute, how to balance efficiency with resilience. The cosmic web formed from the physics of gravity and expansion. Tiny fluctuations in the early universe, ripples in the primordial plasma, grew over billions of years. Regions with slightly more matter pulled in more, forming filaments, while emptier regions drained away, leaving voids. The result? A foam-like structure, a universe that looks less like a void and more like a living network. On smaller scales, galaxies themselves display familiar patterns. Spiral arms curl like seashells and hurricanes. Star clusters resemble the scattering of pollen or the pattern of raindrops on a window. Planetary rings ripple with the same wave mechanics that shape ripples in a pond. Physics keeps reaching for the same toolkit no matter the scale, and yet the resemblance to the brain lingers. Both brains and the cosmic web must move information. In your head, electrical signals fire across neurons. In the universe, gravity transmits information about mass and energy, shaping where galaxies form. Both systems are modular, clusters within clusters, hubs within hubs, connected by bridges. Both balance local specialization with long-distance integration. In neuroscience, That's what allows thought. In cosmology, it's what allows galaxies to form communities, not just islands. No one is saying the universe is conscious in the way you are conscious, but the echo is there, structures that emerge from simple rules, scaling up until they resemble one another, and once you've seen the resemblance, it's hard to ignore. This is the moment in our journey when scale ceases to matter. A neuron and a galaxy cluster, a brain and the universe, they're all following variations of the same rules. The blueprint is not bound to biology or to physics alone. It is more fundamental, an architecture that reappears whenever matter is given freedom to move, connect, and grow. For some, this realization is comforting. It suggests we belong, that we're not an exception carved into a hostile cosmos. For others, it's unsettling because it raises the possibility that what we call life may simply be the universe's way of drawing filaments and spirals over and over on every canvas it can find. And so we stand at a crossroads of interpretation. Either these patterns are coincidences, shapes our minds can't help but connect, or they are hints that life and cosmos share a common logic. Maybe the reason neurons and galaxies look alike is because there are only so many ways to build a resilient, efficient network. Maybe the universe is not imitating life. Maybe life is imitating the universe. If that's true, then the boundary between biology and cosmology starts to blur, and the blueprint we've been tracing so far, through diatoms, rivers, shells, and snowflakes, turns out to be the same blueprint etched into the largest structure ever discovered. If the blueprint repeats itself across scales, then perhaps it repeats across worlds. To imagine alien life, we don't need to leave Earth. We just need to go deeper, into the ocean's twilight, where sunlight fades and strange bodies drift in silence. Here, you meet creatures that seem conjured from imagination. Comb jellies with rainbow light running down their transparent ribs. Chains of salps, ghostly tubes linked together like beads, stretching for meters through the water. Jellyfish that vanish in daylight but glow at night as if filled with stars. These forms are so delicate, so unfamiliar, they might as well be emissaries from another planet. And yet, they are here, abundant, essential to the ocean's ecosystem. Life in the deep ocean shows us that biology can thrive under extreme pressures, near freezing temperatures, and in total darkness. In fact, Earth's most alien-seeming creatures may also be its most adaptable. They are proof that once physics grants the right flows of energy, life bends itself into shapes that look alien to us, but are perfectly natural responses to their environment. Some of the most astonishing ecosystems are not powered by sunlight at all, but by heat and chemistry. At hydrothermal vents along mid-ocean ridges, black smokers belch superheated, mineral-rich water into the abyss. Around these vents, life blooms. Giant tube worms, shrimp, and microbes that eat hydrogen sulfide instead of sunlight. Entire food webs are powered by chemistry alone. This discovery overturned the assumption that life needs the sun. It doesn't. It needs energy and gradients, differences to exploit. The blueprint doesn't care if that energy comes from photons or from boiling water, rising through cracks in the seafloor. Now take that insight and look skyward. On Saturn's moon, Enceladus, jets of water vapour erupt from cracks in its icy surface. When NASA's Cassini spacecraft flew through those plumes, it detected organic molecules and molecular hydrogen, the very fuel that Earth's vent microbes thrive on. Europa, one of Jupiter's moons, hides an even larger ocean beneath its frozen crust. Tidal forces from Jupiter's gravity churn that ocean, keeping it liquid and restless. If hydrothermal vents exist there, the same blueprint that powers Earth's darkest ecosystems might be running beneath Europa's ice. This is where speculation becomes grounded. We don't have to dream up crystal-based life or silicon forest to imagine aliens. We just need to look at our own planet's most extreme habitats. Physics says energy gradients must be used. Chemistry says molecules will organize when nudged. The outcome is not guaranteed, but the odds are better than chance. If the blueprint is real, it should show up wherever energy flows and matter can move. And consider this. Many of the creatures we think of as alien-looking are actually among the oldest lineages on Earth. Jellies, sponges, microbes, they've been here for hundreds of millions of years, outlasting mass extinctions. evolving in forms that seem strange to us only because we are latecomers. Perhaps alien life too will not look humanoid or mammalian, but gelatinous, microbial, luminous. The kind of life that doesn't need bones or eyes, only patterns that physics keeps rewarding. Even beyond our solar system, the idea holds. Exoplanets orbiting distant stars may host conditions wildly different from Earth. But if physics pushes matter into the same recurring motifs, then spirals, branches and networks might appear again and again, sculpted by necessity. Alien life may be stranger than anything we imagine, but also familiar in its logic, efficient, patterned, organised. Which leads to a provocative thought. When we finally meet life elsewhere, whether it's microbes beneath Europa's ice, plankton drifting in Enceladus's ocean, or something entirely new on a distant exoplanet, it may not shock us with how different it is. It may shock us with how much it resembles what we already know. Alien life may not be alien at all. It may be another verse in the same cosmic poem, written with the same recurring words: flow, pattern, shape, efficiency. And if that's true, then the blueprint of life isn't Earth's private code, it's the universe's habit. We've zoomed through oceans and across galaxies, but perhaps the most astonishing place to see the blueprint is here, inside ourselves. Start with your lungs, two balloon-like organs, soft and pink, but within them lies a forest. The airways branch and branch again, from trachea to bronchi to bronchioles, until they terminate in alveoli, tiny sacs that exchange oxygen for carbon dioxide. If you unravelled them all, their combined surface area would stretch to the size of a tennis court. This is not random design, it's fractal logic. Branching maximises surface area while minimising volume. It's the same strategy rivers use to reach every corner of a watershed, the same strategy trees use to feed their leaves with water. Your lungs breathe because physics demands efficiency. Next, look at your circulatory system. Arteries split into smaller arteries, then into capillaries so fine they thread between single cells. This branching web ensures no cell in your body is more than a hair's breadth away from nutrients and oxygen. If you laid all those vessels end to end, they'd stretch for about 60,000 miles, more than twice the circumference of the Earth. Again, fractals at work. What lightning does in the sky, your blood does in silence, every moment of your life. Then there's the brain. 86 billion neurons, each branching like trees, each firing across synapses to neighbours, forming a network so vast that its structure defies imagination. But it isn't chaos. Neuroscience has shown that the brain is a small world network, highly connected locally, but with long-range bridges that allow information to travel quickly. That same architecture shows up in social networks, the internet, even power grids. And it rhymes with the cosmic web itself. Clusters connected by filaments, balancing efficiency and resilience. DNA may feel like the obvious candidate for the blueprint of life, and in one sense, it is. 4 chemical letters, A, T, C, G, arranged in endless combinations, producing the diversity of life on Earth. But step back, and DNA is just one implementation of a deeper set of rules. Encode, replicate, adapt. The real marvel is how those 4 letters can describe a body that obeys fractal branching, spiraling growth, and self-organizing networks. DNA doesn't defy physics, it takes advantage of it. Now consider the human microbiome. For every human cell in your body, there is roughly 1 microbial cell. Trillions of bacteria, viruses and fungi living on your skin, in your gut, even in your lungs. You're not a single organism, you're an ecosystem, a walking galaxy of microbes whose interactions are as vital as your own heartbeat. Without them, you couldn't digest food, train your immune system, or even produce certain vitamins. The blueprint here is ecological, a web of relationships, resilience through diversity. It mirrors the same principles that make rainforests thrive and galaxies cluster. Scaling laws also run through us. Across species, from shrews to whales, metabolism follows a predictable curve, body mass to the 3/4 power. This is why a hummingbird's heart races at over 1,000 beats per minute, while a whale's thuds only a handful of times each minute. Both extremes make sense when viewed through fractal transport networks. Smaller bodies must pump faster to keep up, larger bodies slower to conserve. You're part of this curve too, your own heartbeat ticking in rhythm with the same math that governs elephants and sparrows. Even your heartbeat contains hidden fractals. Healthy heartbeats show variability, tiny fluctuations that look similar across different timescales. Too regular, and it signals illness. Too chaotic, and the system collapses. The sweet spot, structured unpredictability, is exactly what complexity theory predicts for resilient systems. Your pulse is physics finding balance at the edge of order and chaos. Everywhere we look inside the human body, the same patterns surface. Branches to distribute, spirals to grow, networks to connect, fluctuations to adapt. It's the same language we've been tracing from snowflakes to galaxies. The blueprint doesn't just describe the cosmos, it describes us. And that realization reframes what it means to be human. We're not outside the system, not exceptions to the rules. We are the rules, written in flesh and thought. The same blueprint that shapes the Milky Way shapes the neurons firing as you listen to these words. The same geometry that sculpts a snowflake sculpts the branching airways inside your lungs. The blueprint is not just out there, it's in here, and it means that our search for understanding the universe is also a search for understanding ourselves. So far, the blueprint has been a story of wonder, of rivers that echo our lungs and galaxies that echo our brains. But this isn't just abstract beauty. The patterns are practical. They can be read, measured, and used. Humanity has already begun to borrow from the blueprint. Take medicine. Doctors have learned that healthy systems are fractal. The branching of airways in the lungs, the variability of heartbeats, the rough edges of tissues, all carry signatures of health. When those signatures change, it's an early warning. A lung scan that shows less fractal complexity can signal disease before symptoms become obvious. A heartbeat that loses its variability can predict trouble long before a patient collapses. Even tumours betray themselves through pattern. Aggressive growth often leaves a jagged fractal edge, while slower growth is smoother. By measuring these hidden geometries, medicine is learning to diagnose earlier, faster, better. Engineers too have taken notes from nature's sketchbook. Buildings in hot climates sometimes borrow their cooling strategies from termite mounds, whose branching tunnels regulate temperature. Computer chips use capillary-like channels to wick heat away, inspired by blood vessels. Architects design trusses that branch like trees, achieving strength with minimal material. This field has a name, biomimicry, but the principle is simpler. Find what physics already solved, then copy it. Climate science may be where the blueprint matters most. The ocean's invisible forests, phytoplankton, bloom in fractal whirls visible from space. These patterns reveal the hidden currents that drive Earth's climate engine. Phytoplankton produce roughly half of the oxygen in the air you're breathing right now. They also lock carbon away, steering the fate of the planet's atmosphere. To track them, satellites scan the ocean for subtle colour shifts, turning blooms into data. By reading the blueprint at this scale, scientists can model how Earth itself breathes. And then there is technology. Artificial neural networks, the engines of modern AI, were inspired, at least loosely, by the human brain. Billions of nodes passing signals, learning by adjusting connections. Today, these systems are already helping us read patterns we can't see ourselves, detecting early signs of disease in scans, mapping ecosystems, predicting weather. In a way, AI is a tool the blueprint built to help us recognize itself. The lesson in all of this is that the patterns are not just metaphors, they're instructions. They tell us how to build, how to heal, how to adapt. The same spirals and branches that shape seashells and snowflakes are now guiding skyscrapers and supercomputers. But there's also a humbler lesson. The blueprint existed long before us. We are not inventing these solutions, we are rediscovering them. And in doing so, we are reminded that we are not outside of nature, not separate from the cosmos. We are participants in a repeating story, one that physics has been telling since the beginning. From a drop of seawater to the sweep of galaxies, we've traced the same motifs: branches, spirals, networks, waves. They repeat not because nature is lazy, but because physics is relentless. Energy must flow, matter must move, and when it does, these shapes appear, again and again, across across every scale. Your lungs echo trees, your blood vessels echo rivers, your brain echoes the structure of the universe itself. The glass houses of diatoms mirror the symmetry of snowflakes, while the spiral of a shell mirrors the arms of a galaxy. This is the blueprint, not a single code, but a family of solutions that keep winning the same contests. Efficiency, connection, resilience. What does this mean? Perhaps life is not rare because it is miraculous, but because the rules are universal. Given energy, imbalance, and time, matter doesn't just sit. It organizes, branches, spirals, and weaves itself into webs. And sometimes, those webs open their eyes and wonder at themselves. So the next time you look at a tree or a river or the stars, remember, you are not just looking at nature. You're looking at the handwriting of the cosmos, written everywhere, including in you. In the darkness of an ancient ocean, two cells drifted toward each other. They had no brain to recognize the moment, no memory to record what would happen next. But in the space between them, smaller than a grain of sand, wider than the gulf between stars, the entire future of life hung suspended. For 1 billion years, every cell that had ever lived had followed one rule: survive alone, die alone. Competition was the only language life knew how to speak. Until this moment. Until these two cells did something that should have been impossible. They chose each other. What happened in that microscopic space would echo through time. It would build forests and fabricate mines. It would create love and construct civilizations. Because when life discovered teamwork, it discovered how to become more than the sum of its parts. It discovered how to become us. But here's what nobody tells you about cooperation. It began as a mistake. A failure so catastrophic that it should have meant death for both cells involved. Instead, it became the most successful accident in the history of existence. According to microbiologist Lynn Margulis, who spent her career studying these ancient alliances, the first act of cooperation was actually an attempted murder, one cell trying to consume another, both failing so spectacularly that they became something neither could have imagined. They became one. Tonight we're diving into the precise moment when life learned its most powerful secret, when the universe discovered that the only way to build complexity was to break the oldest rule of survival, when competition gave way to collaboration, and in doing so, wrote the first line of every love story, every friendship, every family that would ever exist. If you've ever wondered why humans form tribes, why your body works as one system instead of 37 trillion competing cells, why cooperation feels so right even in a world that rewards competition, the answer lives in this moment. The moment when two ancient cells looked at infinity alone and chose the terrifying alternative together. "3 billion years ago, Earth was an alien world. No flowers painted the landscape, no birds carved paths through the sky. No creatures crawled across the seafloor. There was only water, rock, and a thin soup of single cells drifting through endless darkness. And for 1 billion years that was enough. Picture the vastness of it. An entire planet wrapped in ocean, and in every drop of that ocean a war. Not a war of armies or ideologies, but something simpler and more brutal. Every cell for itself. Every organism a solitary empire of one, competing for the same dissolving minerals, the same fleeting photons of sunlight that managed to penetrate the murky depths. These weren't primitive beings, not in the way we might imagine. According to research from the NASA Astrobiology Institute, these early cells had already mastered the fundamental tricks of existence. They could harvest energy from chemicals, they could replicate their DNA with stunning accuracy, they could sense their environment and respond to it. Some could even swim, using whip-like flagella to navigate their liquid universe. They were in every sense that mattered perfect at being alone. The chemistry of these ancient cells was remarkably sophisticated. They possessed enzymatic machinery that would put modern factories to shame. Ribosomes, those protein-building machines, were already achieving accuracy rates of 99.99% in translating genetic code. DNA polymerases could copy billions of base pairs with fewer errors than a medieval scribe copying a single page. These cells had learned to build complex lipid membranes that could selectively allow certain molecules in while keeping others out. They had evolved protein pumps that could move materials against concentration gradients, defying entropy itself. Dr. William Martin from Heinrich Heine University describes these early cells as thermodynamic miracles. Each one was a tiny engine that could capture energy from its environment and use it to maintain order in a universe trending toward chaos. Some harvested light, splitting water molecules with molecular machinery so efficient that human technology still can't match it. Others oxidized sulfur compounds, extracting energy from rocks themselves. Still others had learned chemosynthesis, building organic molecules from inorganic carbon using nothing but chemical energy. But perfection, it turns out, has limits. The ocean was changing. Oxygen, that element we now depend on with every breath, was beginning to poison the water. Cyanobacteria, those blue-green architects of atmosphere, had learned to crack water molecules apart and harvest the hydrogen, discarding oxygen as waste. What they didn't know was that their waste would become the planet's first mass extinction event. The Great Oxidation Event, scientists call it now. Back then, it was simply the end of the world. The scale of this catastrophe is difficult to comprehend. Dr. Timothy Lyons from UC Riverside studies ancient ocean chemistry through rock formations that preserve chemical signatures from this period. His research reveals that oxygen levels rose from essentially 0 to about 2% of current levels over just a few million years. For organisms that had evolved in an oxygen-free world, this was like suddenly filling the atmosphere with chlorine gas. For the anaerobic cells that had ruled Earth for 2 billion years, oxygen was death itself. It ripped apart their proteins, shredded their DNA, turned their own chemistry against them. The oceans became killing fields. 99% of all life on Earth would die in this catastrophe. But in the depths of this disaster, something unprecedented was about to happen. The chemistry of oxygen toxicity is particularly brutal. Oxygen readily accepts electrons, forming reactive oxygen species like superoxide and hydrogen peroxide. These molecules are cellular wrecking balls, breaking carbon-carbon bonds, oxidizing amino acids, and causing DNA strand breaks. Modern cells have elaborate antioxidant systems to neutralize these threats. Catalase, superoxide dismutase, glutathione peroxidase. But ancient anaerobic cells had none of these defenses. They were naked before the oxygen storm. See, when you're drowning in poison, when the very air has turned against you, the ancient law of competition starts to look less like wisdom and more like suicide. Some cells found ways to hide from the oxygen, burrowing into mud, fleeing to the deepest trenches where the poison hadn't yet reached. But a few cells tried something different, something that had never been attempted in 2 billion years of evolution. They got close to each other, dangerously close. The scientist who first proposed this radical idea was ridiculed for years. Lynn Margulis, working at Boston University in the 1960s, suggested that the complex cells that make up all plants, animals and fungi arose from an ancient merger. Her paper was rejected 15 times before finally being published in 1967. The scientific community called it fantasy, impossible, a violation of everything they knew about evolution. The resistance to Margulis's idea wasn't just scientific conservatism, it challenged the fundamental narrative of evolution as a story of competition. Darwin himself had written about the struggle for existence. The phrase survival of the fittest had become synonymous with evolution in the public mind. Here was Margulis suggesting that the most important event in evolutionary history wasn't competition, but cooperation. It was heresy. But Margulis had seen something others had missed. She noticed that mitochondria, those power plants inside our cells, had their own DNA. They divided on their own schedule. They had double membranes, as if one cell had been wrapped inside another. They even had their own ribosomes, slightly different from the cell's own protein factories. These weren't just organs that had evolved inside cells, these were immigrants, ancient visitors who had never left. The evidence was overwhelming once people started looking. According to research from the University of California, the genetic code of mitochondria is more similar to certain bacteria than it is to the cells they live in. It's as if you opened up your laptop and found a completely different computer running inside it, speaking a different programming language, yet somehow making the whole system work better. The genetic evidence is particularly compelling. Mitochondrial DNA uses a slightly different genetic code than nuclear DNA. In mitochondria, the codon UGA codes for tryptophan instead of being a stop codon. The codon AGA is a stop signal instead of coding for arginine. These differences make no sense if mitochondria evolve from within eukaryotic cells, but they make perfect sense if mitochondria are descended from bacteria that were captured and domesticated. But how did that first encounter actually unfold? Dr. Nick Lane from University College London has spent his career reconstructing this moment. His models, published in Cell in 2023, suggest it began with hunger. A large, slow archaeon cell struggling in the oxygen-rich waters encountered a small, fast bacterium that had learned to process oxygen. The archaeon did what cells had done for billions of years. It attacked, it engulfed, it tried to consume. The mechanics of cellular predation are surprisingly complex. The archaeon would have extended pseudopods, finger-like projections of its membrane, to surround the bacterium. This process, called phagocytosis, requires sophisticated molecular machinery. Actin filaments must polymerize to push the membrane forward. Myosin motors must contract to pull the prey inside. Membrane fusion proteins must seal the compartment. It's a molecular ballet that modern cells still perform billions of times each day. Here's where physics becomes poetry. The bacterium should have been digested, should have been broken down into component parts, its proteins recycled, its DNA discarded. Instead, something extraordinary happened. The bacterium's ability to process oxygen created a gradient, a flow of energy that the archaeon had never experienced before. It was like a starving creature suddenly tasting sugar for the first time. The archaeon didn't digest its prey because the prey was more valuable alive. The energetics of this partnership were revolutionary. Before the merger, the archaeon might have generated 2 ATP molecules per glucose molecule through fermentation. The bacterium, with its oxygen processing abilities, could generate 36 ATP molecules from the same glucose. Together, they had an 18-fold increase in energy availability. It was like going from a candle to a power plant overnight. But the bacterium faced its own crisis. Trapped inside another cell, it should have suffocated, should have been cut off from nutrients. Instead, it found itself in a protected environment, surrounded by resources, shielded from the toxic oxygen levels outside. The prison became a paradise. The predator became a protector. Inside your cells right now, 37 trillion times over, an ancient negotiation continues. Every mitochondrion maintains its own genetic material, about 37 genes in humans, while the cell's nucleus holds the remaining 1,500 genes that once belonged to that original bacterial partner. It's a distributed system, like a business where some decisions are made in branch offices, while others require approval from headquarters. This division of genetic labour didn't happen randomly. The genes that remained in mitochondria are primarily those encoding proteins involved in the electron transport chain, the core of energy production. These proteins must be produced quickly in response to changing energy demands. Having their genes locally allows rapid response. It's like keeping emergency supplies in every room rather than in a central warehouse. Dr. Ian Johnston from the University of Bergen studies this cellular communication. His 2024 research reveals that mitochondria and their host cells engage in constant molecular dialogue. When the cell needs energy, it sends chemical signals to the mitochondria. The mitochondria respond not just by producing ATP, the universal energy currency, but by adjusting hundreds of cellular processes. They influence when cells divide, when they die, even when they decide to become cancer. The signaling between mitochondria and nucleus is bewilderingly complex. Calcium ions flow back and forth, creating waves of information. Reactive oxygen species, once thought to be purely harmful, act as signaling molecules at low concentrations. Metabolites like acetyl-CoA and NAD+ carry information about energy status. Small peptides encoded by mitochondrial DNA can even travel to the nucleus to influence gene expression. It's a conversation that never stops, a dialogue that has been refined over 2 billion years. This isn't just cooperation, it's integration so complete that neither partner can survive alone. Scientists have tried to remove mitochondria from cells to see if modern cells can live without their ancient partners. The cells die within hours. Conversely, mitochondria extracted from cells and given all the nutrients they could want still perish quickly. The partnership that began as optional became mandatory. The merger that started as temporary became permanent. The intimacy of this relationship extends to the molecular level. Mitochondrial proteins made in the cytoplasm must be tagged with specific targeting sequences, molecular zip codes that ensure they reach their destination. These sequences are recognized by receptors on the mitochondrial surface. TOM20, TOM22, TOM70. Each receptor recognizes different targeting sequences. It's a postal system more complex than anything humans have designed. But how did this molecular marriage actually work? The mechanics are more intricate than any human technology. According to research from the Max Planck Institute for Biology, the process involves 5 distinct systems working in harmony. First, the protein import machinery. Mitochondria can't make all their own proteins anymore. They depend on proteins made in the cell's cytoplasm, which must then be threaded through specialized pores in the mitochondrial membranes. The complexity of protein import defies belief. Proteins must be unfolded to pass through the import channels, then refolded on the other side. This requires molecular chaperones, proteins whose sole job is helping other proteins fold correctly. HSP70 in the cytoplasm keeps proteins unfolded. MT, HSP70 in the mitochondrial matrix pulls them through. HSP60 and HSP10 form barrel-shaped chambers where proteins can fold in isolation, protected from the cellular environment. The partnership between that first archaeon and bacterium was just the beginning. Once life discovered that cooperation could overcome competition, the strategy spread like wildfire through evolution's playbook. But not in the way you might expect. It wasn't a gradual transition, it was an explosion. Between the first eukaryotic cells and the emergence of true multicellularity, there was a period scientists call the boring billion, a billion years when not much seemed to happen. But beneath the surface, cells were experimenting with different ways of being together. Colonies formed and dissolved. Cells stuck together temporarily, then separated. It was like evolution was learning the grammar of cooperation before writing its masterpiece. Dr. Rosie Allegado from the University of Hawaii studies these transitional forms. Her work on choanoflagellates reveals that the switch between single-cell and colonial living can be triggered by molecules from bacteria. A single bacterial protein can cause choanoflagellates to form rosette-shaped colonies. It's cooperation induced by communication between different domains of life. Bacteria talking to proto-animals in chemical languages that predate words by billions of years. For nearly 2 billion years after that first merger, life remained mostly microscopic. Complex cells with mitochondria existed, but they seemed content to remain single. Then, around 600 million years ago, something shifted. The fossil record from this period, particularly the Ediacaran biota discovered in Australia, shows something unprecedented. Cells weren't just tolerating each other, they were organising, they were specialising, they were becoming something that had never existed before. Multicellular organisms. The Ediacaran fossils are unlike anything alive today. Dickinsonia, looking like a quilted bath mat, could grow over 1 metre long. Tribrachidium had threefold symmetry, unlike the bilateral or radial symmetry of modern animals. Charnia looked like a fern but was actually an animal, feeding by absorbing nutrients through its surface. These were evolution's rough drafts, experiments in multicellular living that would mostly end in extinction. Dr. Nicole King from UC Berkeley has spent her career studying this transition. Her research on choanoflagellates, single-celled organisms that can form temporary colonies, provides a window into how multicellularity might have evolved. These organisms can live perfectly well alone, but when conditions change, when food becomes scarce or predators appear, they do something remarkable. They come together. They form spheres and sheets and chains, each cell suddenly part of something larger than itself. The molecular basis of this transformation is surprisingly simple. A protein called ROS-R controls colony formation. When activated, it triggers expression of genes for adhesion proteins, allowing cells to stick together. But it also suppresses genes for cell division, ensuring the colony doesn't grow too large. It's a molecular switch between individuality and collectivity, and it can flip in minutes. Your body is a civilization. 37 trillion cells, more than all the stars in our galaxy, working in perfect coordination. Every second, millions of cells die and are replaced. Every heartbeat requires the synchronized contraction of billions of muscle cells. Every thought involves cascading electrical signals across billions of neurons. This isn't just cooperation. It's a symphony of impossible complexity, and it works so well that you never notice it happening. The scale of cellular cooperation in your body defies comprehension. Your heart beats 100,000 times per day, each beat requiring the coordinated contraction of millions of cardiac muscle cells. These cells are electrically coupled through gap junctions, protein channels that allow ions to flow directly between cells. When one cell depolarizes, the electrical signal spreads like a wave, ensuring perfect synchronization. It's been doing this since before you were born, and it will continue until your last breath. Your digestive system is another marvel of cooperation. It's not just human cells working together, but a vast ecosystem. Your gut contains 100 trillion bacteria, 10 times more than human cells in your body. These bacteria don't just hitchhike, they're essential partners. They break down complex carbohydrates your own enzymes can't touch. They synthesize vitamins K and B12. They train your immune system to distinguish friend from foe. Without them, you would starve no matter how much you ate. Dr. Michael Levin from Tufts University studies how bodies maintain this coordination. His groundbreaking research on bioelectricity reveals that cells don't just follow genetic instructions. They communicate through electrical fields, creating body-wide patterns that guide development and regeneration. When Levin's team manipulates these electrical patterns, they can cause frogs to grow eyes on their stomachs or regenerate lost limbs. The implications are staggering. Cooperation isn't just chemical, it's electrical, creating an information network that spans the entire organism. Levin's work reveals that cells can store pattern memories in their electrical networks. Planarian flatworms, when decapitated, can regenerate their heads. But if you briefly alter the electrical patterns during regeneration, they grow heads with different shapes, and amazingly, these new shapes persist through future regenerations even without further intervention. The electrical pattern has rewritten the morphological memory. Consider your immune system. It's not just a defense force, it's a molecular intelligence agency. T cells and B cells patrol your body, checking molecular passwords on every cell they encounter. Dendritic cells act as scouts, capturing invaders and presenting their molecular signatures to other immune cells. Memory cells maintain libraries of past threats, some keeping watch for decades. It's a distributed intelligence network with no central command, yet it can distinguish between trillions of different molecular patterns. The sophistication of immune recognition borders on miraculous. Your immune system can distinguish between billions of different antigens, including ones that have never existed before. It achieves this through a process called VDJ recombination, randomly shuffling gene segments to create unique antibodies. It's evolution happening in real time inside your body, with B cells competing to produce the best antibodies against each threat. The principles of cellular cooperation echo throughout the living world. From the smallest bacterial colonies to the largest animal societies, the patterns repeat: communication, specialization, sacrifice, and mutual benefit. But nowhere are these patterns more elaborate than in the social insects. Dr. Deborah Gordon from Stanford studies harvester ants in the Arizona desert. Her research reveals that ant colonies operate without central control. No ant understands the colony's overall state. Instead, they use simple rules based on chemical interactions. When an ant encounters another ant, they touch antennae, exchanging chemical information about what task they've been performing. If an ant meets many foragers, it might switch to foraging. If it meets many nest maintainers, it might switch to nest maintenance. From these simple interactions emerges complex colony behavior that can adapt to changing conditions. The parallels to cellular cooperation are striking. Just as cells use chemical signals to coordinate behavior, ants use pheromones. Just as cells specialize into different types, ants differentiate into castes. Just as cells undergo apoptosis for the good of the organism, worker ants sacrifice reproduction for the colony. The patterns of cooperation, evolved independently, converge on similar solutions. Consider the leafcutter ants studied by Dr. Cameron Curry at the University of Wisconsin. These insects have created one of nature's most complex cooperative systems. The ants don't eat the leaves they cut. They use them to farm fungus, which feeds the colony. But it goes deeper. The ants carry bacteria on their bodies that produce antibiotics to protect the fungus from parasites. It's a four-way partnership: ant, fungus, bacteria, and even viruses that infect the parasites. Each partner is essential. Remove one, and the whole system collapses. The fungus gardens of leafcutter ants are agricultural marvels. Worker ants of different sizes perform specialized tasks. The largest workers cut leaves and carry them back to the nest. Medium workers process the leaves into mulch. Tiny workers tend the fungus gardens, removing contamination and spreading fungal growth. The very smallest workers ride on the leaves being carried, protecting carriers from parasitic flies. It's division of labour refined over 50 million years of evolution. This isn't unusual, it's the norm. Dr. Margaret McFarlane Guy from the University of Hawaii studies the bobtail squid, which harbours bioluminescent bacteria in a special organ. The bacteria provide camouflage, eliminating the squid's shadow in moonlight. The squid provides nutrients and shelter. Every morning the squid expels 95% of the bacteria, keeping only the brightest. Its cooperation with quality control, partnership with performance reviews. The biochemistry of this partnership is exquisite. The squid's light organ has evolved tissues that focus and direct the bacterial light. The bacteria, in turn, have evolved to produce light only when they reach high density, ensuring bright illumination. The squid even has biochemical sensors that can detect the difference between its symbiotic bacteria and potential pathogens, welcoming one while rejecting the other. Humans have taken cooperation to unprecedented levels. Dr. Michael Tomasello from Duke University argues that human cooperation is qualitatively different from other species. We don't just work together, we share intentions. We create joint goals that exist nowhere in nature except human minds. When two people carry a log together, they're not just coordinating movements, they're sharing a mental representation of what they're trying to achieve. This shared intentionality appears early in human development. By 18 months, children engage in cooperative games with shared goals. They help adults complete tasks without being asked. They show concern when cooperative partners stop cooperating. These behaviours appear before language fully develops, suggesting that cooperation is foundational to human psychology. Here we are, at the edge of another transformation. Artificial intelligence is learning to collaborate with humans and each other. Synthetic biologists engineering new forms of cellular cooperation. Brain-computer interfaces creating hybrid cooperations between minds and machines. We stand where those ancient cells stood 2 billion years ago, at the threshold of a new kind of togetherness. The parallels are uncanny. Just as those first cells faced an oxygen crisis that forced them to cooperate or die, we face climate change, pandemic disease, and technological disruption that demands unprecedented global cooperation. Just as those cells had to overcome the ancient law of competition, we must transcend tribal instincts that evolved for a world that no longer exists. CRISPR gene editing allows us to rewrite the contracts between our cells. Dr. Jennifer Doudna, who won the Nobel Prize for developing CRISPR, describes it as molecular scissors that can cut and paste DNA with precision. But it's more than that. It's the ability to engineer cooperation at the molecular level. Scientists are using CRISPR to create cells that cooperate in new ways. Immune cells that team up to fight cancer, bacteria that work together to produce biofuels, plants that share nutrients more efficiently. Organoid research grows miniature brains where human neurons cooperate in dishes. Dr. Sergiu Pasca at Stanford creates assembloids, brain organoids from different brain regions that wire together and create circuits. These cellular cooperatives can model disease, test drugs, and might one day repair damaged brains. It's cooperation in a petri dish, but with profound implications for understanding consciousness itself. Xenobots, living robots made from frog cells, demonstrate programmable, biological cooperation. Created by Dr. Josh Bongard at the University of Vermont, these organisms are neither traditional robots nor typical animals. They're something entirely new. Cellular cooperatives designed by artificial intelligence and built from living tissue. They can move, heal themselves, even replicate. They represent a new form of life, one where cooperation is designed rather than evolved. We're not just observing cooperation anymore, we're designing it. Dr. Sam Bowles from the Santa Fe Institute studies how human cooperation might evolve from here. His models suggest we're at a critical juncture. Technology enables cooperation at unprecedented scales, but also creates new ways to defect. Social media connects billions, but also spreads division. Artificial intelligence could be humanity's greatest cooperative tool, or could fragment us further. The mathematics of future cooperation are complex. Network effects mean that cooperative systems can grow exponentially once they reach critical mass, but they can also collapse just as quickly if trust erodes. Dr. Robert Axelrod's research on the evolution of cooperation shows that successful cooperative strategies share key features. They're nice, never, defect first; retaliatory, punish defection; forgiving, return to cooperation after punishment; and clear, easy for others to understand. The blockchain represents one attempt to engineer trustless cooperation. Systems where cooperation doesn't require trusting other participants. Smart contracts automatically enforce agreements. Distributed ledgers make cheating impossible. It's cooperation through code rather than culture. But critics argue that removing trust from cooperation misses the point, that trust itself is valuable, that the vulnerability required for cooperation is what makes it transformative. The lessons from that first cellular merger have never been more relevant. Competition alone leads nowhere. The archaeon and bacterium that tried to destroy each other would have died in obscurity, but when they stopped trying to win against each other and started winning together, they inherited the Earth. Consider what's happening in medicine. CAR T therapy engineers immune cells to cooperate in fighting cancer, but it goes beyond individual treatments. The Human Cell Atlas project involves thousands of scientists worldwide cooperating to map every cell type in the human body. The COVID-19 vaccine development saw unprecedented cooperation between companies that usually compete. Moderna and Pfizer shared information. Oxford and AstraZeneca shared technology. It was cellular cooperation scaled up to corporate cooperation, saving millions of lives. In ecology, we're discovering that cooperation extends far beyond what we imagined. The Wood Wide Web, fungal networks that connect forest trees, allows trees to share nutrients, warn each other of insect attacks, and even support sick neighbours. Dr. Susan Simard from the University of British Columbia has shown that mother trees nurture their offspring through these networks, reducing competition to help them grow. Forests aren't collections of individual trees competing for resources. They're superorganisms cooperating at scales we're only beginning to understand. Climate change represents perhaps the ultimate cooperation challenge. It's a tragedy of the commons on a planetary scale. The atmosphere belongs to everyone and no one. Every nation benefits from others reducing emissions while wanting to emit freely themselves. Game theory suggests this should be unsolvable, yet we see glimpses of hope. The Montreal Protocol successfully eliminated ozone-depleting chemicals through global cooperation. It worked because it aligned incentives, provided clear verification mechanisms, and helped developing nations transition. It proved that planetary cooperation is possible. The Paris Agreement attempts something similar for carbon emissions, though with mixed results. The challenge isn't scientific or technological. We know how to solve climate change. The challenge is cooperation. Look at your hand. In just the skin you can see, there are more cells than there are people who have ever lived. Each cell carries mitochondria, descendants of that ancient partnership. Each mitochondrion contains DNA from a bacterium that lived 2 billion years ago. You are not an individual. You are a colony, a civilization, a walking testament to the power of cooperation. But you're more than that. You're also part of larger cooperatives. Your family sharing genes and resources, your community sharing labor and culture, your species sharing knowledge and technology. Life is cooperation all the way up and all the way down, from molecules to civilizations, from bacteria to biospheres. The neurons firing in your brain as you process these words, they're cooperating. The muscles adjusting your posture, cooperating. The immune cells patrolling your bloodstream, cooperating. Even the bacteria in your gut, breaking down your last meal, cooperating. You are cooperation incarnate, a hierarchical symphony of togetherness that stretches from molecules to consciousness. Dr. Lynn Margulis, who first proposed endosymbiotic theory, spent her final years studying Gaia theory with James Lovelock. They proposed that Earth itself might be understood as a cooperative system, with life and geology interacting to maintain habitable conditions. The oxygen in our atmosphere, the salt in our oceans, the temperature of our planet, all are regulated by feedbacks between living and non-living systems. Cooperation not just between cells, but between kingdoms of life, between life and rock, between earth and sky. Recent research supports aspects of Gaia theory. The Amazon rainforest creates its own rainfall through transpiration. Plankton in the oceans produce dimethyl sulfide, which seeds clouds that reflect sunlight and cool the planet. Weathering of rocks by bacteria and plants removes CO2 from the atmosphere, regulating temperature over geological time. Earth's habitability isn't an accident. It's maintained by countless cooperative feedbacks we're only beginning to map. We began this journey in the darkness of an ancient ocean, with two cells drifting toward each other. We've travelled through billions of years, from molecular mergers to human civilizations, from cellular contracts to global cooperation. And in all that time, across all those scales, the lesson remains the same. Alone, we are limited by what we are. Together, we become what we never imagined possible. Every friendship echoes that first partnership. Every act of kindness reverberates with 2-billion-year-old wisdom. Every moment you choose connection over isolation, collaboration over competition, you're honouring an ancient contract written in the very structure of your cells. Think about the profundity of this inheritance. The love you feel for family and friends, the satisfaction of working with a team, the joy of belonging to a community. These aren't cultural constructs. They're biological imperatives encoded in every cell of your being. The pull toward togetherness that humans feel isn't weakness or sentimentality. It's the deepest wisdom of life itself, learned through billions of years of trial and error. The universe spent 10 billion years perfecting the art of being alone. Stars burned in solitude. Planets spun in isolation. Even when life emerged, it spent another billion years in single-celled solitude. But then, in one impossible moment, two cells chose each other. And in that choice, in that first successful cooperation, the universe discovered something extraordinary. It discovered that the whole really can be greater than the sum of its parts, that 1 + 1 can equal 3, or 1,000, or 37 trillion. That cooperation isn't just a strategy for survival. It's the strategy that builds complexity, creates consciousness, enables love. We started by asking when life discovered teamwork, but maybe that's the wrong question. Maybe the question isn't when life discovered teamwork, but when teamwork discovered life. Because what is life, really, but cooperation made flesh? What is consciousness but cooperation made aware? What is love but cooperation made sacred? In your cells, that ancient partnership continues. Every breath you take, every thought you think, every beat of your heart is powered by the descendants of that first merger. You don't just carry the evidence of that moment. You are the evidence. You are the proof that cooperation works, that togetherness triumphs, that the universe's greatest discovery was the simple, revolutionary idea that we're better together. The moment life discovered teamwork, it discovered the future. It discovered us. And now, as we stand at the threshold of new forms of cooperation, with artificial minds, with engineered biology, with planetary systems, we carry forward that 2-billion-year legacy. The question isn't whether we'll cooperate. Life has already answered that question. The question is how we'll cooperate, at what scale, with what purpose. The answer lies not in our technologies or institutions, but in remembering what we are. Cooperative miracles, each one of us a society of trillions working as one, each society part of something larger still. When we remember this, when we truly understand that cooperation is our deepest nature, the path forward becomes clear. We don't need to learn cooperation. We need to remember it. We need to honour it. We need to embody it at every scale, from the molecular to the global. Because in the end, the moment life discovered teamwork wasn't just a moment in the past. It's a moment that continues in every heartbeat, every breath, every gesture of connection. It's happening right now, in you, as 37 trillion cells work as one to create the miracle of consciousness contemplating its own cooperative nature. The moment life discovered teamwork, it discovered that the universe could know itself. And through you, through all of us, it continues that discovery, one cooperation at a time. If you enjoyed this journey, consider subscribing to Omni, where we explore the hidden stories of science, nature, and the universe. Every like, every comment, and every share helps these ideas reach more curious minds. Thanks for watching, and as always, stay curious. In a studio on the south side of Chicago, an artist stands over a flat panel, pouring resin onto wet paint. Bruce Riley works in layers, dripping paint, manipulating it while still wet, then flooding the surface with clear resin before the colours can dry. Then he does it again, and again, layer after layer, building depth that catches light from multiple angles, creating shadows deep within the artwork. His paintings grow without preconception, in what he calls the biomimetic state, allowing the work to determine itself through process, through flow, through the interaction of materials that he can guide but never fully control. The results look like something pulled from a microscope. Organic forms blooming across the canvas, psychedelic mandalas that resemble cellular structures, patterns that seem simultaneously alien and familiar, like peering into a petri dish where life has just discovered colour. I'm addicted to the psychological risk-taking that is part of unknowing, Riley says. When I work too much from a place of intent, the life leaves a piece. He's describing his art, but he could just as easily be describing evolution. Because if you zoom in far enough, if you look closely at the actual petri dishes in laboratories around the world, you'll see Riley's paintings coming to life. Bacteria spreading across agar in blooming fractals, cells dividing, aggregating, forming structures that no single organism planned. Patterns emerging from simple rules applied by billions of individuals. The resemblance isn't coincidental. Riley's process, layering, flow, emergence, letting complexity arise from simplicity, mirrors the fundamental process that invented you. That invented every multicellular organism on Earth. Right now, beneath your feet, inside your body, in every drop of water and handful of soil, invisible communities are negotiating the same bargain that made complex life possible. Individual cells are deciding stay alone or come together, compete or cooperate, remain independent or sacrifice autonomy for something greater. This is the most fundamental question in biology, and the answer is written in every cell you're made of. 600 million years ago, cells faced a choice: stay alone or come together. And those that chose cooperation invented something the universe had never seen: multicellular life. You're watching the descendants of that choice right now. Every breath you take, every thought crossing your mind, every heartbeat echoing in your chest exists because microscopic cells learned to trust each other, to communicate, to sacrifice individual freedom for collective survival. But here's what haunts biologists about this transition. In every act of cooperation, there's temptation. Cheetah cells that take the benefits without paying the cost. Freeloaders that could destroy everything. So how did cells overcome their selfish nature? What invisible force convinced them to build bodies instead of staying free? This is Omni. If you're fascinated by the hidden mechanisms that built complex life from simple beginnings, subscribe and join us as we explore the invisible worlds that made you possible. Tonight, we're tracing the origin story written in microscopic ink. We'll follow the moment when single cells stopped dividing apart and started holding together. When they evolved the molecular glue of cooperation, the language of chemical signals, and the ruthless mechanisms that punish cheaters. We'll watch bacteria form biofilms that behave like primitive organs. We'll meet organisms that flicker between solitary and social states. And we'll discover that the boundary between me and we is more fragile than you ever imagined. Because understanding how cells learn to cooperate isn't just biology. It's the story of every complex thing that's ever existed. From a slime mold's tower to a human brain, from a beehive to a civilization, from Bruce Riley's paintings to the body watching them. The rules are older than animals, older than plants, older than anything with a face or a name. They were written by cells, in the invisible dark, learning to become something more. The story begins in darkness. Not the darkness of night, but the ancient, absolute darkness of an ocean 4 billion years old. No eyes exist yet to see, no minds to wonder. Just chemistry, endlessly cycling through the water. And yet, even here, something remarkable was happening. A single bacterial cell, invisible to any imagined observer, was releasing molecules into the water around it. These weren't waste products, or accidents of metabolism. They were messages, chemical sentences written in the universal language of molecular shapes, drifting outward from the cell like ripples on a pond. But here's the strange part. The cell was talking to itself, or more precisely, it was counting, measuring how many of its own kind surrounded it in the darkness. When those signal molecules accumulated to a certain threshold, when enough cells were present, something changed, genes switched on, behaviors transformed. Individual bacteria, acting alone, suddenly became something else entirely, a coordinated collective. This process, which scientists would eventually call quorum sensing, allowed bacteria to do something that seemed impossible for creatures without brains or nervous systems. They could assess their population density. They could wait for the right moment. They could act in unison. Think about what that means. A bacterium measuring a millionth of a meter across with no neurons, no awareness, no sense of self, had evolved the ability to know when it was alone and when it was among family, and it changed its entire life strategy based on that information. The ocean was full of these invisible conversations. Bacteria in the water column secreted autoinducers, signal molecules that accumulated as populations grew. When the concentration reached a threshold, when the quorum was reached, entire communities would synchronize their behavior. Some would begin glowing, creating bioluminescent displays that could be seen from the surface. Others would start producing toxins or digestive enzymes that no single cell could manufacture effectively alone. The chemical signals varied between species like different languages. Gram-negative bacteria produced acyl homoserine lactones that could passively drift through their thin cell walls. Gram-positive bacteria used peptide signals that had to be actively transported across their thicker peptidoglycan barriers. Each species had evolved its own dialect, its own frequency, its own way of saying, "we are many." But the most extraordinary thing wasn't the communication itself. It was what the communication enabled. When bacteria sensed a quorum, they would begin producing what scientists call public goods, molecules that benefited the entire community, exoenzymes that broke down complex nutrients, components of biofilms that created protective shelters, virulence factors that overwhelmed host defenses. These were expensive to make. They cost energy and resources. And here's the problem. Any cell that didn't contribute, any cheater that saved its resources while enjoying the benefits, would grow faster than the cooperators. This should have destroyed cooperation before it ever began. Natural selection should have favoured the freeloaders, the cells that took without giving. And in many bacterial populations, that's exactly what happened. Cheetah mutants that didn't produce the costly signals or public goods would invade populations of honest cooperators, threatening to collapse the entire social system. So why didn't cooperation disappear? The answer lies in something subtle, something that changes everything about how we think about individual and collective. Cooperation was more likely to evolve and persist when bacteria were highly related, when all the cells came from a single founding cell. When you're surrounded by your genetic siblings, helping the group means helping copies of your own genes. The distinction between selfish and altruistic behaviour begins to blur. Picture a bacterial mat forming on the ancient seafloor. A single cell divides. Its daughters divide. Within hours, a colony of thousands blooms into existence, all carrying identical genetic instructions. When these clones sense their own autoinducers, when they detect the quorum, they're not really communicating with strangers. They're coordinating with copies of themselves, like a distributed intelligence running the same program across multiple processors. The signals themselves were more sophisticated than anyone initially imagined. Concentrations of molecules like cyclic deguanylate would oscillate in well-defined patterns across generations, creating waves of information that spread through bacterial lineages like radio transmissions. Different frequencies, different amplitudes, encoding different messages about surfaces, about nutrients, about danger. This was mass communication without mass media, broadcasting without transmitters, a form of collective computation happening at the molecular level, where individual cells became nodes in a living network. And it enabled something extraordinary. When enough bacteria gathered in one place, when the quorum was reached, they would begin building biofilms, vast architectural structures made of cells embedded in a self-produced matrix of polysaccharides, proteins, lipids, and DNA. These weren't just random aggregations. They were organized three-dimensional communities with distinct zones and functions, channels for nutrient flow, layers with different metabolic activities, protective barriers against antibiotics and immune cells. Bacteria had invented cities, complete with infrastructure, resource distribution, and cooperative defense, all without a blueprint, without a foreman, without any cell understanding the larger structure it was helping to build. The biofilm spread across every surface in the ancient ocean. They coated rocks, formed mats on sediments, created living carpets that trapped nutrients and changed the chemistry of their surroundings. And within these structures, the real innovation emerged. Bacteria weren't just cooperating with their own kind, they were coordinating with different species entirely. Some bacteria evolved the ability to produce and detect AI2, a universal signaling molecule that could facilitate interspecies communication. This was profound. It meant bacteria could assess not just their own numbers, but the entire composition of the microbial community around them. They could detect when they were in heterogeneous environments and modulate their behavior accordingly. Competition and cooperation occurring simultaneously. Species that normally competed for resources would share information, coordinate biofilm formation, even enhance each other's survival. Some bacterial species like Burkholderia capacea and Pseudomonas aeruginosa would use quorum sensing to amplify each other's virulence, creating devastating co-infections. Others would form symbiotic partnerships, with one species creating conditions that allowed another to thrive. The ocean floor became a tapestry of these invisible alliances, bacterial mats meters thick containing billions of cells, all communicating through molecular signals, all responding to the changing concentrations of auto-inducers that informed them about their world. Some cells at the surface received more oxygen and light. Others, buried deep in the mat, lived in anoxic darkness. But they were connected by chemistry, by the diffusion of signals and nutrients, by the emergent properties of their collective behavior. This was the first step. The first time life discovered that many could be stronger than one. But bacteria remained fundamentally simple. Each cell was essentially identical to its neighbours. There was no specialisation, no division of labour, no sense that some cells should serve fundamentally different roles than others. The biofilms were impressive, but they were more like dense crowds than organised bodies. The next step would require something more radical. Approximately 2 billion years ago, in an ocean that was slowly accumulating oxygen from photosynthetic cyanobacteria, a different kind of cell was evolving. These were eukaryotes, cells with nuclei, with complex internal architecture, with the capacity for something bacteria could never achieve. And some of these cells were facing a crisis. Food was running out. In the soil and on decaying leaves, populations of tiny amoebae called Dictyostelium hunted bacteria. They moved independently, each one a solitary predator following chemical gradients to find prey. But when bacteria became scarce, when starvation loomed, something astonishing happened. One cell would begin secreting cyclic AMP, the same molecule that bacteria used for signalling, now repurposed for a new kind of communication. Other amoebae would detect this signal and respond in two ways. They would move toward the source, and they would release more cyclic AMP themselves. The effect cascaded through the population. Waves of chemical signals rippled outward from the initial broadcaster. Amoebae aligned themselves along these waves, forming distinct streams of cells flowing toward a central aggregation point. Within hours, between 10,000 and 1 million cells would converge, piling up into a mound. But they didn't stop there. The mound would extend itself, forming a slug-shaped mass that could migrate across surfaces. And here's the remarkable part. The cells were beginning to differentiate. Some cells in the slug would become specialized for movement. Others would prepare to become spores, and still others would commit to something that seems like suicide. When the slug found a suitable location, it would transform into a fruiting body, a stalk topped with a mass of spores. The cells in the stalk would die, sacrificing themselves to lift the spores away from the ground and into air currents that could carry them to new locations. This was a form of cooperation that bacteria had never achieved. Permanent differentiation, specialized cell types, some cells dedicating their entire existence to supporting others, with no possibility of reproduction, no direct genetic future. Why would natural selection favor this? Recent research revealed something unexpected. The trigger for this multicellular transformation wasn't just starvation in general, it was specifically the loss of sulfur-containing amino acids like cysteine. When cysteine levels dropped, when sulfur became limiting, it signaled to the cells that conditions were no longer suitable for growth and division. The best strategy was no longer to remain independent. It was to pool resources, to create a structure that could survive the crisis and disperse spores to potentially more favourable locations. And the slug could do things individual amoebae could never accomplish. Experiments showed that solitary amoebae couldn't cross soil barriers that slugs navigated easily. The multicellular form was better at local dispersal, better at finding new food patches. Cells shed from the migrating slug would consume bacteria in its path, clearing obstacles. But there was still a problem. When the multicellular slug formed by aggregation, when cells of different genotypes came together, the stage was set for conflict. Cheetah cells that contributed less to the stalk but positioned themselves in the spores would have a reproductive advantage. They would outcompete honest cooperators, potentially destroying the entire system. Yet in nature, something even stranger was discovered. About 1/3 of wild Dictyostelium colonies engaged in primitive farming, carrying bacteria within their fruiting bodies to seed new locations with food. This meant accepting a cost, smaller spores that couldn't disperse as far, in exchange for a long-term benefit, arriving at a new location with your food source already present. Cooperation was getting more complex, and it was beginning to resemble something we would recognize as multicellular life. But Dictyostelium only went part way. Its multicellular phase was temporary, triggered by crisis, and the cells that formed it often came from different genetic lineages. True multicellularity, the kind that built animals and plants and fungi, would require something more fundamental, a commitment from the very beginning, from a single founding cell. The real breakthrough came in a different lineage entirely. In shallow ponds and slow-moving streams, tiny green spheres were rolling through the water, each one a perfect geometric marvel. Meet Volvox, a multicellular green alga barely 2 millimeters across, containing just a few thousand cells arranged in a hollow sphere. But unlike Dictyostelium, every cell in Volvox descended from a single founding cell. No aggregation, no temporary coalitions, permanent commitment from the very beginning. This changes everything. The Volvocene algae, the family that includes Volvox, represents something extraordinary in evolutionary biology. They span the entire spectrum from unicellular ancestors to fully differentiated multicellular organisms, and they did it recently enough that we can trace the steps. The group includes Chlamydomonas, a single-celled alga that swims alone, then Gonium, a flat colony of cells, all identical, then Pandorina and Euderina, larger colonies still without specialized cell types, and finally Volvox, with its dramatic innovation, a complete division of labor between two cell types, germ cells and somatic cells, reproductive gonidia that would create the next generation, and smaller somatic cells with flagella that would never reproduce but would spend their entire existence swimming, propelling the sphere through water, supporting the reproducers. This was the first time in evolution that cells born from the same parent committed to fundamentally different fates, not because of environmental triggers or starvation stress, but because of their position and size during development. During embryonic division, some cells divided asymmetrically, producing large-small sister pairs. The large cells activated one set of genes, the small cells activated another. Size determined destiny. The large cells would never swim. They would grow, divide, and create the next generation. The small cells would never reproduce. A gene called Reg A would activate in them, Shutting down all aspects of reproductive development permanently, they became somatic cells, body cells, condemned to mortality, existing only to serve the immortal germline. Why would natural selection favour this sacrifice? The answer emerged when scientists compared Volvox to its relatives. The entire lineage showed a progressive pattern: increases in cell number, increases in body size, increases in the extracellular matrix surrounding cells, and increasing specialization. Larger size provided advantages. It made organisms harder for predators to consume. It allowed them to stay suspended in the water column more effectively. And critically, larger size provided an advantage in competing for limiting resources like phosphorus. But there's a physical constraint. A cell can't be both optimized for reproduction and optimized for movement. Flagella require tremendous energy and space. Chloroplasts for photosynthesis require different cellular real estate than the metabolic machinery for rapid growth and division. You can specialize for one or the other, but not both equally well. Analysis of gene expression in Volvox revealed the stark dichotomy. Gonadial cells expressed growth-related genes, ancient genes focused on reproduction and resource acquisition. Somatic cells expressed a more specialized program, younger, lineage-specific genes devoted to an altruistic metabolic program. They manufactured flagella. They converted storage carbon into sugars for producing the extracellular matrix glycoproteins that enabled the sphere to expand to its massive size. The somatic cells were metabolic servants, working for the collective good at the cost of their own reproduction. But here's what haunts biologists about this arrangement. The genetic difference between unicellular Chlamydomonas and multicellular Volvox is surprisingly small. Despite 200 million years of independent evolution, their gene content is nearly identical. The transformation from solitary cell to multicellular organism with differentiated cell types didn't require vast new genetic innovation. It required rewiring, taking genes that already existed and expressing them in new patterns, at new times and new combinations. Some of the cell type-specific genes in Volvox appeared to be co-opted from a unicellular ancestor's day-night cycle. Genes that were once expressed during dark phases became specialized to somatic cells. Genes once expressed during light phases became specialized to gonadia. Temporal programs, daily rhythms, were converted into spatial programs, permanent cell types. This is how evolution builds complexity. Not from scratch, but through tinkering, repurposing, taking systems that served one function and discovering they could serve another. And once the germ-soma division appeared, once some cells committed to dying so others could reproduce, natural selection could sculpt both types independently. Somatic cells could evolve to be better at motility, better at photosynthesis, better at building the extracellular matrix, without any concern for maintaining reproductive capacity. Germ cells could focus entirely on growth and division, protected and transported by their somatic siblings. The colony became an individual, the cells became tissues, but Volvox was still primitive, just two cell types. No organs, no complex development beyond the initial division between germ and soma. For the next transformation, for the leap to animal complexity, evolution would need something more. It would need cells that could not only differentiate but could also communicate position, coordinate movement, and build three-dimensional structures with sophisticated architecture. The path to animals began in a place both alien and strangely familiar. Ancient oceans, perhaps 800 million years ago, where the oxygen concentration was finally rising to levels that could support larger, more energetically expensive life. And in those waters swam creatures called chonoflagellates, single-celled organisms with a distinctive collar of finger-like microvilli surrounding a single flagellum. They looked remarkably similar to cells found in modern sponges. The chonocytes, collar cells, that line the interior of sponges and generate water currents for filter feeding are nearly identical in structure to free-living chonoflagellates. This wasn't convergent evolution. Chonoflagellates are the closest living relatives of animals. The last common ancestor of all animals was a creature very much like a modern chonoflagellate. A single cell with a collar and flagellum filter feeding on bacteria. But unlike their modern descendants, some ancient chonoflagellates discovered they could do something extraordinary. They could form colonies. certain species of chonoflagellates can transition between single-celled and multicellular states. They divide but fail to separate, remaining attached through their collars, forming beautiful rosette-shaped colonies of cells arranged in spheres. 4 cells, 8 cells, 16, sometimes 50 cells, all beating their flagella in coordination. And these colonies could do things single cells could never accomplish. Research showed that colony geometry affected feeding efficiency. In certain configurations, cells and colonies could capture more prey per cell than solitary chonoflagellates. The water currents generated by coordinated flagella beating created flow patterns that concentrated bacteria near the collars. But colony formation also came with costs. Larger colonies produced bigger hydrodynamic signals, making them more visible to predators. Raptorial protozoans would capture colonies while ignoring single cells. But larger colonies were also too big for some predators to handle, rejected by ciliate predators that could easily consume individual cells. The selective pressures were complex, contradictory, pushing and pulling in different directions. Sometimes it paid to be alone, sometimes it paid to be together. The ability to switch between unicellular and multicellular states, facultative multicellularity, might have been the key adaptation. In 2019, researchers discovered a new chonoflagellate species, Chuanuka flexa, that exhibited remarkable collective behavior. Individual organisms would join together by touching collars, forming a concave sheet with flagella pointing inward for feeding. But in darkness, the sheet would invert into a ball-like shape with flagella pointing outward, allowing the colony to swim rapidly, shape-shifting in response to light, a multicellular organism capable of coordinated behavior that no individual cell could achieve, swimming toward well-lit areas where food might be abundant, a navigation strategy impossible for solitary cells. When scientists examined the molecular toolkit of chonoflagellates, they found something astonishing. These unicellular organisms possessed many of the genes that animals use for cell adhesion, cell signaling, and even genes involved in regulating development. The genetic machinery for building multicellular bodies was already present in single-celled ancestors. Evolution hadn't invented these tools for multicellularity. The tools existed first, serving other functions in unicellular life. Cell adhesion proteins that would later hold tissues together originally helped cells stick to surfaces. Signaling molecules that would coordinate development originally helped cells sense their environment. The transition to multicellularity wasn't about gaining new capabilities. It was about keeping cells together after division and repurposing existing molecular machinery for new collective functions. Some chonoflagellates formed colonies through repeated cell division from a single cell. Clonal multicellularity, the same strategy that would define animal development. Every animal on Earth, from sponges to humans, develops from a single fertilized cell that divides repeatedly, with daughter cells remaining attached. This developmental mode was already established in our chonoflagellate-like ancestors. But the crucial question remained: how did temporary colonies become permanent bodies? How did facultative multicellularity become obligate? How did collections of cells that could separate become organisms that could never again exist as individuals? The answer required 3 fundamental evolutionary steps. First, cell-to-cell adhesion with an alignment of fitness among cells, keeping cells together and ensuring they benefited from cooperation. Second, cell-to-cell communication, cooperation and specialization, with an export of fitness to the multicellular organism level. And third, the evolution of mechanisms that prevented cells from reverting to unicellularity. These mechanisms were called ratchets, traits that provided benefits in a group context, but were detrimental to loners, ultimately preventing any return to single-celled life. Once cells became mutually dependent, once they divided labour so thoroughly that no single cell retained all necessary functions, there was no going back. Some cells might specialize in growing one vital molecule, while others grew a different essential compound, so these cells did better together than apart. The more specialized cells became, the more they needed each other, the stronger the ratchet clicked into place. And with each click, with each increment of mutual dependence, the individual cells became less like independent organisms, and more like parts of a larger whole. The colony became the individual, the cells became organs, the organism became irreversibly multicellular. Somewhere in the Precambrian oceans between 800 and 635 million years ago, this transition completed itself. A lineage of chonoflagellate-like organisms crossed the threshold, their cells no longer separated after division, their colonies no longer broke apart. They had committed fully, permanently, irrevocably to multicellular life, and from that commitment everything followed. $635 million years ago, as the Earth thawed from the last great cryogenian glaciation, when ice sheets finally retreated and oxygen levels began to rise in the ancient seas, something unprecedented appeared in the fossil record. Soft-bodied impressions in sandstone, frond-shaped organisms, disc-like creatures, strange quilted patterns unlike anything that came before. The Ediacaran biota, Earth's first large multicellular communities. These organisms lived for nearly 40 million years before the Cambrian explosion, representing early experimentation with complex life forms. They demonstrated mobility, heterotrophy by multicellular animals, the beginnings of skeletonization, and the assembly of complex ecosystems. They were building bodies at scales previously impossible, and somewhere among them, or perhaps shortly before them, the first true animals emerged. These earliest animals were likely sponges, simple creatures without muscles, without nerves, without guts, but with something revolutionary: multiple differentiated cell types working together in coordinated fashion. They weren't just colonies of identical cells. They were organisms with specialized tissues, each cell type performing distinct functions that the others couldn't. Chonocytes with collars and flagella that generated water currents and captured bacteria. Pinacocytes that formed the outer covering and provided structural support. Archaeocytes that migrated through the interior, capable of transforming into other cell types as needed. And critically, a mesohyl, a jelly-like matrix between cell layers that served as a scaffold, a medium for cell migration, a three-dimensional architecture that single-cell layers could never achieve. This was different from everything that came before. The innovation wasn't just sticking cells together. It was creating a polarized body plan with water flowing in through pores, circulating through internal chambers, and flowing out through an opening at the top. The sponge had invented a circulatory system without a heart, digestion without a gut, coordination without a nervous system. When scientists finally sequenced the sponge genome, they discovered something remarkable. Sponges possessed the same core genetic toolkit as all other animals: genes for cell division and growth, for programmed cell death, for cell adhesion, for signaling during development, for recognition of self and non-self, for the formation of different cell types. The molecular machinery for building animal bodies was already present in the simplest animals. What sponges lacked, muscles, neurons, guts, would come later. But the fundamental innovation, the transition from colonial organism to integrated individual, had already occurred. And here's the darker parallel that this transition revealed. The same genes that regulate cooperation in multicellular organisms are the genes whose disruption causes cancer. Multicellularity and uncontrolled cell growth are two sides of the same coin. When cells in a body decide to stop cooperating, when they revert to selfish behavior and grow without regard for the organism's needs, they become tumors. Cancer is cooperation breaking down, its cells remembering their unicellular past, rejecting the constraints of multicellular life, attempting to return to a more ancient strategy of individual reproduction at any cost. Every gene that helps cells cooperate, that makes them divide when they're supposed to, die when they're supposed to, stay in their designated roles, is a potential cancer gene when it malfunctions. The transition to multicellularity required building fail-safes, checkpoints, mechanisms to detect and eliminate cheaters. And when those mechanisms fail, the body pays the price. This is the cost of complexity, the price of becoming more than the sum of your parts. But the benefits were extraordinary. Once animals evolved spatial cell differentiation, the ability to create different cell types in controlled locations, they could build bodies with unprecedented sophistication. Not just random aggregations of cells, but architectures with purpose, layers that could fold, invaginate, create internal spaces, cells that could specialize to the point where no individual cell could survive alone. But together they could navigate, hunt, reproduce with complexity that dwarfed anything single cells could achieve. The cell types that evolved in early animals were built by parceling out functions that originally existed in multifunctional ancestral cells. A single choanoflagellate-like ancestor might have been sensory, contractile and digestive all at once. Evolution split these capabilities, gave them to different cells, and in doing so created the possibility of specialization. One cell type could become exquisitely good at sensing. Another could master contraction and movement. A third could focus entirely on digestion. Division of labour at the cellular level opened the door to everything that followed, and the ratchet clicked one final time. Animals committed to clonal development, every individual arising from a single fertilised cell that divides repeatedly, with all cells sharing the same genome. This wasn't aggregation like dictyostelium. This was a developmental program encoded in DNA, executed with precision every generation, building the same body plan from a single cell through an invariant series of divisions and differentiations. No going back, no reverting to single-cell life. The individual cell ceased to be the unit of selection. The multicellular organism became the individual. From that commitment emerged the Cambrian explosion. 540 million years ago, in a geological instant, animals diversified into all the major body plans we recognize today. Eyes evolved. Shells and exoskeletons appeared. Predation became sophisticated. The arms race of hunter and hunted drove innovation at a pace the Earth had never seen. But none of it would have been possible without those first uncertain steps. Without bacteria learning to count each other through chemical whispers. Without biofilms discovering that structure emerges from simple rules applied by many. Without slime molds sacrificing some cells so others could disperse. Without Volvox committing to permanent differentiation. Without choanoflagellates forming rosettes that could swim in coordinated patterns. Every innovation built on the previous one, every transition required cells to give up something. Autonomy, reproduction, immortality, in exchange for something greater. Stand at the edge of an ocean and look at the water. Billions upon billions of cells, invisible to your eyes, are negotiating this same ancient bargain. Bacteria are deciding whether to cooperate or cheat. Algae are coordinating cell division. Microscopic animals are building bodies that will last hours, days, weeks, before dissolving back into single cells that will try the experiment again. And inside your own body, trillions of cells are maintaining the cooperation that makes you possible. Your neurons aren't reproducing, sacrificing their genetic future so you can think. Your red blood cells don't even have nuclei anymore, committed so thoroughly to oxygen transport that they shed their DNA entirely. Your skin cells are programmed to die and slough away, making room for the next generation. You are a colony that forgot it was a colony, a temporary coalition of cells that believes it's a permanent individual. Your sense of self, your feeling of being a unified consciousness rather than a community of microsystems is the ultimate achievement of cellular cooperation. But the cooperation is always fragile, always one mutation away from breaking down, always requiring active maintenance, constant surveillance, perpetual enforcement of the rules that keep cells working together rather than pursuing their own reproduction. The question that haunted early multicellular life still haunts us. How do you keep cheaters from destroying the collective? Cancer is proof that we haven't completely solved this problem. Our immune system patrols constantly, destroying cells that have stopped cooperating. Our bodies execute elaborate checkpoints before allowing cells to divide, and still, sometimes, a cell escapes surveillance and begins to grow without constraint. The battle between individual and collective, between selfish gene and cooperative genome, never ended. It moved from oceans to tissues, from bacterial mats to tumor microenvironments, from simple biofilms to complex bodies. But here's what makes this story remarkable. Despite the fragility, despite the constant threat of defection, cooperation won. Multicellular life didn't just survive, it dominated. Today, the most massive organisms on Earth are multicellular. The most intelligent are multicellular. The most diverse forms of life are multicellular. The strategy that began with bacteria counting each other in darkness that continued with slime moulds forming towers, that accelerated with algae dividing labour, that culminated in animals building bodies from single cells, that strategy reshaped the planet. Forests emerged, transforming the atmosphere. Animals colonised every habitat from deep ocean vents to mountain peaks. Brains evolved, generating consciousness, asking questions about their own origins, and all of it traced back to that first moment when a cell decided not to separate from its sister after division. When two became one, when individual gave way to collective, the invisible worlds that invented multicellular life are still here, still experimenting, still negotiating the balance between self and group. In every drop of pond water, in every handful of soil, in every surface covered with biofilm, the ancient drama continues. Cells are learning to cooperate, and sometimes, against all odds, cooperation becomes something more permanent, something that can build bodies, minds, societies, something that can look back across billions of years and ask, how did we become possible? The answer was written in molecular language, in the chemical dialects that bacteria first spoke in ancient darkness, in the sacrifice of soma for germ, in the ratchet mechanisms that made cooperation irreversible, in the developmental programs that build bodies from single cells. You are the answer. Every multicellular organism is the answer. A temporary victory of cooperation over conflict. A momentary alignment of individual interest toward collective survival. A body built from billions of cells that mostly remember to work together. The invisible worlds invented you, and you carry their story in every cell, in every gene, in every molecular mechanism that keeps your cells cooperating instead of competing. Remember that the next time you take a breath, or move a muscle, or think a thought. You are not an individual, you are a community, a coalition, a democracy of cells that somehow convinced themselves they were one. And the story of how that became possible, the story of cells learning to trust each other enough to build something bigger than themselves, is the most fundamental story in biology. It's the story of how chaos learned to create order, how individuals learned to become communities, how invisible worlds learn to build visible ones. It's the story of cooperation. And cooperation, for all its fragility, for all its constant threat of collapse, remains the most powerful force that evolution ever discovered. Every cell in your body, all 37 trillion of them, is in constant conversation. Chemical signals, electrical pulses, molecular messages passing between membranes faster than thought. They're negotiating, coordinating, remembering. They've been doing this since before you had a brain to think with. But here's what we never asked. If cells have been talking for 4 billion years, long before mouths, before brains, before anything we'd call consciousness, then what were they saying? And who are they saying it to? Because the first conversation wasn't between two humans. It was between two single cells trying to survive the same ocean, and every word you've spoken since is written in the same language they invented. Tonight we'll go back to the beginning, to the first chemical signal sent in the dark, to the first time a cell heard and answered. Because the language of cells isn't a metaphor, it's the foundation everything else is built on, and it's still speaking, right now, in you. You're watching Omni, a space for those who wonder why life exists the way it does. Each film is a story about connection, chaos and discovery. Comment what you think. Hit like if it sparked something in you, and subscribe to keep exploring ideas that matter. let's start with a moment most of us never think about. You're reading these words right now, processing symbols, extracting meaning, forming thoughts in response. It feels effortless, natural, human. But zoom in. Zoom past your eyes, tracking the screen, past the neurons firing recognition patterns in your visual cortex, past the electrochemical signals racing through your brain at 300 miles per hour. Zoom all the way down to a single synapse. The gap between two nerve cells where one neuron is trying to tell another neuron something important. What you'd see is this: a tiny vesicle, a bubble of membrane, approaching the edge of the first neuron. It fuses with the cell wall, and in that moment, thousands of molecules spill out into the gap. Neurotransmitters, serotonin, dopamine, glutamate, each one a word in a sentence your cells are writing without your permission. They drift across the space, 20 nanometers, smaller than a wavelength of light, and lock into receptors to the other side. Specific shapes for specific locks. The second neuron receives the message. It changes, its voltage shifts. It decides, fire or stay silent. You didn't command this. You didn't choose the molecules or design the receptors. But somewhere in that exchange, in that negotiation between two cells you'll never meet, a thought happened. This sentence appeared in your mind. And here's the thing we forget. That negotiation, cell to cell, molecule to molecule, meaning passed through chemistry, is older than you. Older than your species. Older than anything with a brain. It's the same conversation that happened in the first bacterial colony 4 billion years ago, when life was nothing but single cells floating in a primordial ocean, trying not to die alone. Because here's the question nobody asks in biology class. Why did life ever become complex? Why didn't every organism stay single, simple, independent? Why cooperate? Why trust? Why build something bigger than yourself when the universe rewards selfish replication? The answer is language, not words, not grammar, not even sound. But the ability to send a signal and have it mean something to someone else. The moment one cell could tell another cell, I'm starving, or danger is coming, or let's work together, that's the moment life stopped being chemistry and became communication. And once communication existed, everything else followed. Cooperation, specialization, multicellular life, tissues, organs, nervous systems, brains, consciousness. All of it, every layer of complexity that makes you you, rests on a foundation of cells talking to each other in a language invented before there were ears to hear it. But we don't see it that way. We think of language as the thing that separates us from the rest of life, the tool that makes us special, human. We think animals have signals, humans have language, but that's backwards. What if language is the thing that connects us to everything else? What if every conversation you've ever had, every inside joke, every confession, every misunderstanding is just a higher resolution version of what cells have been doing since the beginning? What if I love you is the same message as a chemical gradient pulling two bacteria together in the dark? Tonight, we're going to follow that thread. We'll start at the origin, 4 billion years ago, in an ocean that had no voices, and trace the evolution of meaning itself, from the first molecule that said, "Here I am," to the moment 100 trillion cells learned to say I. We'll see how bacteria invented democracy, how dying cells write wills, how your gut and your brain negotiate you're not invited to. And we'll ask the question at the heart of it all. If cells have been speaking this whole time, what have they been trying to tell us? because the language of cells isn't silent. We just forgot how to hear it. Until now. Let's go back. Not to the beginning of human language, not even to the first animal calls or bird songs, but to a time when Earth had no voices at all. 4 billion years ago, the planet was an ocean punctuated by volcanic islands, thick with dissolved minerals and organic molecules. The atmosphere was toxic by our standards. Methane, ammonia, carbon dioxide. No oxygen to breathe, no ozone layer to filter radiation, just water, chemistry, and the relentless churn of geological time. And somewhere in that hostile sea, in the space between two molecules that had accidentally learned to copy themselves, the first signal was sent. We don't know exactly what it looked like. We can't rewind the tape and watch it happen. But we can infer it from what remains, from the molecular machinery that every living cell still carries, like an evolutionary heirloom too useful to discard. Because here's what we know for certain. Life, from the very beginning, required recognition. A cell membrane is a border, a declaration of this is me, that is not me. And once that border exists, once there's an inside and an outside, chemistry becomes information. A molecule on the outside of your membrane isn't just a molecule anymore, it's a message. And if you have receptors, proteins embedded in your membrane that can detect specific shapes, specific chemicals, then you can read it. This is the foundation of all biological communication. Receptor and ligand, lock and key, signal and response. It's elegant in its simplicity. A molecule floats through the environment. It encounters a cell. If that cell has the right receptor, the molecule binds, clicks into place like a puzzle piece, and something changes inside. A cascade of chemical reactions begins. Genes activate or shut down. The cell moves, divides, releases its own molecules in response. The message has been received. According to research published in Nature Reviews Microbiology in 2021, even the most ancient bacteria possess sophisticated receptor systems, suggesting that chemical communication is not an innovation, but a prerequisite, something that had to exist for life to exist at all. But here's the question that haunted early evolutionary biologists. Why? If you're a single-celled organism floating in the primordial ocean, what advantage is there in signaling? You can't talk to yourself. You already know your own state. And if you're alone, truly alone in a vast empty sea, there's no one to signal to. Sending out molecules costs energy. Building receptors costs energy. Why bother with communication infrastructure if there's no one on the other end of the line? For decades, the assumption was that cellular signaling must have evolved later, after populations grew dense enough to make communication useful. Single cells were thought of as isolated chemical machines, and communication was a luxury that came with crowding. But then scientists started looking more closely at bacteria, not the sterile, isolated cultures grown in labs, but bacteria in their natural environments. Oceans, soil, biofilms, host organisms, and what they found overturned the assumption. These cells weren't just bumping into each other randomly, oblivious to their neighbours. They were counting, they were measuring population density, they were coordinating their behaviour based on how many others were around them. This process, discovered in the 1960s but not fully understood until the 1990s, is called quorum sensing, and it revealed something profound. Single cells are not solitary. They are social organisms. They have always been social organisms. Because the ocean, it turns out, was never empty. Think about it from the cell's perspective. You're a bacterium drifting in the water column. Resources are scarce. Predators, if anything can be called a predator at this scale, are everywhere. Viruses that can hijack your machinery and turn you into a factory for their own replication. Larger cells that might engulf you. Chemical gradients that signal danger or opportunity. You can't see. You can't hear. You can't think, at least not in any way we'd recognize, but you can sense. You release molecules into the water as a byproduct of your metabolism, signaling molecules called autoinducers. They diffuse outward, spreading in every direction. If you're alone, the concentration stays low. The molecules drift away, diluted by the vastness of the ocean. But if other cells like you are nearby, also releasing the same autoinducer, the concentration rises. It builds. And when it crosses a threshold, When enough of your kind are present, your receptors detect it. The message is clear. You are not alone, and that changes everything. Behaviors that make no sense for a single cell suddenly become viable for a group. Bioluminescence, for instance. One glowing bacterium in the dark is invisible, a waste of energy. But a million glowing bacteria together create light bright enough to be useful, to attract a host, to communicate across distances, to coordinate movement. According to a landmark study by Bonnie Basler at Princeton University, published in Cell in 2002, this is exactly what Vibrio fishery does. These marine bacteria live in the light organs of Hawaiian bobtail squid. During the day, they stay dark, silent, conserving energy. But as their population grows, as more and more bacteria colonize the squid's specialized tissue, the autoinducer concentration rises. When it hits the threshold, the entire population lights up at once, not gradually, not one by one. All together, in a synchronized flash. The squid uses this light to camouflage itself against moonlight filtering down from above, erasing its shadow from predators below. And the bacteria get a safe home, rich in nutrients, its cooperation, its negotiation, its language. But here's what makes this more than a curiosity, more than just a neat trick some ocean bacteria figured out. Quorum sensing is everywhere. It's not unique to Vibrio. It's not even unique to one type of bacteria. According to recent genomic surveys published in Science in 2023, virtually every bacterial species studied possesses some form of quorum sensing machinery. They use different molecules, different dialects if you will, but the grammar is the same. Measure population density, coordinate behavior, act as one when numbers justify it. Pathogenic bacteria use it to time infections, waiting until their numbers are high enough to overwhelm a host's immune system before releasing toxins. Soil bacteria use it to form biofilms, structured communities where cells specialize, some producing protective slime, others gathering nutrients, still others defending against invaders. Cyanobacteria use it to coordinate nitrogen fixation, a metabolically expensive process that only makes sense when the collective can share the cost. This is not communication as metaphor. This is communication as survival strategy, encoded in chemistry, refined over billions of years. These cells are making decisions, not conscious decisions, but decisions nonetheless, based on information received from their environment and their neighbours. They're listening, they're responding. They are, in the most fundamental sense, speaking, and the language they invented, receptor and ligand, signal and threshold, message and meaning, is the same language your neurons use right now to form the thought you're currently thinking. The vocabulary changed, the speed increased, the complexity exploded, but the syntax, the deep structure, remains. So, when did this begin? We don't know the exact moment, but we know it was early. Before photosynthesis, before oxygen, before anything we'd recognize as complex life. Because communication isn't something life developed after it became sophisticated. Communication is what allowed life to become sophisticated in the first place. The first signal sent in the dark wasn't a luxury, it was a question. Is anyone there? And the first response wasn't words, it was chemistry. It was a molecule binding to a receptor, a threshold crossed, a behavior changed. It was two cells recognizing each other across the emptiness, and choosing for the first time to act together instead of alone. But talking to strangers is one thing. What about talking to yourself? What happens when cells stop being individuals and start being parts of something larger? That's where the conversation gets complicated, because the leap from single-celled to multicellular life required a different kind of language entirely, one that didn't just coordinate behavior but coordinated existence itself. The Hawaiian bobtail squid is about the size of your thumb. It spends its days buried in sand and its nights hunting in shallow water under moonlight. To anything looking up from below, predators mostly, the squid would be a perfect silhouette, a dark shape against the faint glow of the moon filtering through the surface, easy prey. But the squid has a trick. On its underside, tucked into specialized pockets of tissue, live millions of Vibrio fishery bacteria, and every night as the moon rises, these bacteria light up. Not randomly, not chaotically, but in perfect coordination. generating a soft blue-green glow that matches the intensity of moonlight above. The squid becomes invisible, its shadow disappears, and the bacteria, in exchange for this service, get a safe home and a steady supply of nutrients from the squid's body. This arrangement, this symbiosis, depends entirely on quorum sensing. But here's what makes it remarkable. The bacteria don't glow immediately when they colonize the squid. A single bacterium or even a few thousand stay dark. They're silent, waiting. Because bioluminescence is energetically expensive. Producing light requires a cascade of biochemical reactions, each one burning through ATP, the cell's energy currency. For one cell glowing alone in the dark, it's a waste. The light would be too dim to matter, the energy spent for nothing. But if you're one of a 10 million bacteria glowing together, then the investment makes sense. Then you create enough light to be useful. So the bacteria wait. They measure. They count their neighbours through chemistry. And only when their population crosses a threshold, only when the vote says we are enough, do they all switch on at once. The mechanism is startlingly simple. Each Vibrio fischeri bacterium constantly produces a small signalling molecule called an autoinducer. In this case, a compound called 3-oxo-C6HSL, though the exact name matters less than what it does. The bacterium releases this molecule into its surroundings as a byproduct of normal metabolism. If the bacterium is alone, the autoinducer diffuses away into the environment, its concentration staying low. But inside the squid's light organ, space is confined. Bacteria are packed close together. As the population grows, more and more cells release the autoinducer. The concentration rises, and here's the elegant part. The autoinducer can pass back through the bacterial membrane. It's not trapped outside, it equilibrates, so the concentration inside each cell starts to match the concentration outside, and when that internal concentration crosses a specific threshold, it binds to a receptor protein inside the cell, a transcription factor called LuxR. This binding changes the protein's shape, activating it. The activated LuxR then binds to DNA and switches on a whole suite of genes, including the ones responsible for producing light. This is democracy at the molecular level. No single bacterium decides when to glow. The decision is collective, determined by population density. Each cell casts a vote in the form of an autoinducer molecule. When enough votes accumulate, when the quorum is reached, the behaviour changes. According to research by Bonnie Barsler and colleagues, published in Annual Review of Genetics in 2005, this isn't unique to bioluminescence. Bacteria use quorum sensing to regulate dozens of behaviours: biofilm formation, toxin production, antibiotic resistance, spore formation, even DNA transfer between cells. The vocabulary changes depending on the species. Different bacteria use different autoinducer molecules, different receptors, different threshold concentrations, but the grammar remains the same. Measure the crowd, coordinate the response, act as one. And it's not just cooperation. Quorum sensing also allows bacteria to be ruthless. Pathogenic bacteria, the ones that cause disease, use it to time their attacks. Pseudomonas aeruginosa, a common opportunistic pathogen, colonizes human lungs, especially in people with cystic fibrosis. When the bacterial population is small, the immune system can manage it. White blood cells patrol, identifying and destroying invaders. But Pseudomonas is patient. The bacteria grow slowly, keeping their population just below the radar. They don't produce toxins yet. They don't form protective biofilms yet. They wait, measuring their own density through quorum sensing. And when their numbers are high enough, when the autoinducer concentration tells them the population is large enough to overwhelm the host's defences, they flip the switch. Toxins flood out. Biofilms form, creating slimy fortresses that antibiotics can't penetrate. The immune system is suddenly facing not a few isolated invaders, but a coordinated army. According to a 2020 study in Nature Microbiology, disrupting quorum sensing in Pseudomonas significantly reduces its virulence, suggesting that the communication system itself, not just the toxins, is a key part of the infection strategy. This raises an unsettling question. If bacteria can count, coordinate, and strategize through chemistry, are they thinking? Not in the way we think, of course. There's no consciousness, no self-awareness, no internal monologue. But there is information processing. There is decision-making based on environmental context. There is memory, in a sense. Quorum sensing systems often include feedback loops where the activated genes produce even more auto-inducer, amplifying the signal and locking the population into the new behavior state until conditions change. Some researchers argue this fits the minimal criteria for cognition, sensing the environment, integrating information, and producing an adaptive response. Others push back, insisting that without neurons, without brains, the word thinking is inappropriate. But perhaps that's the wrong question. Perhaps the better question is, what is thinking made of, if not this? If your thoughts are neurons signaling to other neurons through chemical messages, and bacterial quorum sensing is cells signaling to other cells through chemical messages, then the difference is one of scale and complexity, not kind. Consider what quorum sensing reveals about language itself. We think of language as requiring intent, someone, trying to convey meaning to someone else. But bacteria don't try to communicate. They release auto-inducers as metabolic byproducts, and other bacteria detect them. There's no conscious sender, no conscious receiver, yet meaning emerges anyway. The concentration of the molecule means something. We are few, or we are many, and behavior changes in response to that meaning. This is language stripped to its evolutionary minimum: signal, detection, response. No grammar, no syntax, no recursion, just information encoded in chemistry, transmitted through diffusion and decoded by receptors. Everything else, from pheromones to birdsong to human speech, is elaboration on this theme. And here's where it gets stranger. Bacteria don't just talk to their own species, they eavesdrop. Many bacterial species produce enzymes called lactanases or acylases that can degrade other species' auto-inducers, effectively jamming their communication. It's chemical warfare disguised as signal interference. Other bacteria produce auto-inducers that mimic their neighbours' signals, sending false messages to confuse competitors. According to a 2019 study in PLOS Biology, some bacteria even engage in what researchers call cross-species quorum sensing, where signals from one species influence the behaviour of another, creating multilingual conversations in microbial communities. A biofilm on your teeth, for instance, isn't a single species. It's a consortium, dozens of bacterial species living together, each speaking its own chemical dialect, but somehow coordinating their collective structure. Some species form the foundation layer, attaching to the tooth surface. Others build on top of them. Still others form protective outer layers or produce nutrients that benefit the community. They're negotiating, they're organising, they're building something no single species could build alone. This is the power of cellular communication laid bare. It transforms individuals into collectives. It makes cooperation possible even in the absence of kinship, even in the absence of anything we'd call empathy or altruism. Because the signal, the auto-inducer molecule, doesn't care about motivation. It only cares about threshold. When enough cells are present, behaviour changes. The group acts as one, not because any individual cell decided to cooperate, but because the chemistry of communication makes cooperation the default outcome when population density is high. According to evolutionary biologists, this is likely how multicellular life began, not through some dramatic mutation that made cells suddenly want to stick together, but through pre-existing communication systems like quorum sensing that allowed cells to coordinate behaviour when they found themselves in close proximity. But quorum sensing has a limit. It works across distances of millimetres, maybe centimetres in still water. The auto-inducers diffuse outward, their concentration dropping with distance. Beyond a certain range, the signal becomes noise, indistinguishable from background chemical fluctuations. For bacteria living as independent cells, this is fine. They only need to know about their immediate neighbours. But once cells committed to living together permanently, once they formed multicellular organisms, diffusion-based signalling wasn't enough anymore. A human body is trillions of cells spread across nearly 2 meters of height. Chemical signals released from your brain would take hours to diffuse down to your toes, and by then they'd be diluted to uselessness. Multicellular life needed a faster, more direct communication system. It needed to wire itself together, and that's exactly what it did. Somewhere around 600 million years ago, Give or take a few 100 million years, cells made a choice they couldn't take back. They stopped being individuals, not metaphorically, literally. Single-celled organisms that had spent billions of years perfecting independence, figuring out how to survive alone in hostile environments, began clustering together, and then they stayed. They didn't just form temporary colonies like bacteria in a biofilm, where each cell could theoretically leave and survive on its own. They committed, they specialized, they divided labor. Some cells became the outside, facing the environment, protecting the others. Some became the inside, handling digestion, reproduction, storage. And once that specialization happened, once cells began depending on each other for survival, there was no going back. A skin cell can't survive without a blood cell to bring it oxygen. A blood cell can't survive without a bone marrow cell to produce it. They are, in the most literal sense, incomplete without each other. This was the multicellular revolution, and it required a communication upgrade. Because here's the problem diffusion-based signaling can't solve: coordination at scale. When you're a colony of bacteria using quorum sensing, you're measuring average population density in your local environment. The signal, the autoinducer, spreads outward, dilutes, and eventually fades into noise. That works fine for loose associations, but once cells commit to a permanent structure, once they form tissues, organs, an organized body, they need precision. A heart cell in your left ventricle needs to contract at exactly the same moment as a heart cell in your right ventricle. Not roughly the same moment, not within a few seconds, but simultaneously, within milliseconds. A nerve cell in your spinal cord needs to send a signal to a muscle cell in your leg, and it needs to arrive now, not after the predator has already caught you. Diffusion is too slow. Chemical gradients are too imprecise. Multicellular life needed something faster, more direct, more reliable. It needed to hardwire itself. The solution came in the form of gap junctions. These are protein channels that physically connect adjacent cells, creating cytoplasmic bridges between their interiors. Imagine 2 rooms separated by a wall. Quorum sensing is like shouting through the wall. You can hear each other, but the message is muffled, delayed, approximate. Gap junctions are like cutting a doorway through the wall. Now you can step through. Now the 2 rooms are functionally one continuous space. Gap junctions do this at the molecular level. They're made of proteins called connexins in vertebrates, or annexins in invertebrates, that assemble into hexagonal rings. 2 cells each build half of the channel, and when they meet in the middle they dock together, forming a continuous pore. Small molecules, ions, second messengers, metabolites, can pass directly from one cell's interior to the others. No release into extracellular space, no diffusion, no delay. Direct transfer of information, cell to cell, faster than any chemical signal could travel through the environment. According to research published in Nature Reviews Molecular Cell Biology in 2018, gap junctions are ubiquitous in multicellular organisms. They're found in nearly every tissue type: cardiac muscle, smooth muscle, neurons, glia, epithelial cells, even immune cells, and their function is always the same: synchronization. In your heart, gap junctions connect cardiac muscle cells into a functional syncytium, a network where electrical signals propagate instantly across thousands of cells. When one cell depolarizes, ions flow through gap junctions into neighboring cells, triggering their depolarization. The wave spreads outward from the pacemaker cells in the sinoatrial node, sweeping through the atria, then down through the ventricles, coordinating contraction so that blood is pumped efficiently, rhythmically, 60 to 100 times per minute for your entire life. This isn't a suggestion. It's not a vote. It's a command that propagates through physical connection. If gap junctions fail, if mutations in connexin genes disrupt their formation, the heart loses synchrony. Cells contract out of rhythm. Arrhythmias develop. The organ stops functioning as a unified whole and reverts to a chaotic collection of individuals. The multicellular contract is broken. But gap junctions aren't just about speed. They're about commitment. When cells wire themselves together, they lose a degree of autonomy. A heart cell can't decide not to contract when the electrical signal arrives. It doesn't get a choice. The depolarization wave passes through the gap junction, and the cell responds. This is fundamentally different from quorum sensing. where each bacterium retains individual agency, it measures the auto-inducer concentration and responds according to its own internal threshold. In a multicellular organism, the decision has already been made at a higher level of organisation. The individual cell follows instructions encoded in electrical and chemical gradients that it didn't create and can't override. According to a 2021 study in Developmental Cell, this loss of autonomy is actually reinforced over evolutionary time. Cells in multicellular organisms have tumour suppressor genes, genetic machinery designed to detect when a cell tries to act independently, to grow without regard for the collective. If a cell starts dividing without permission, ignoring the signals from its neighbours, those genes trigger apoptosis. The cell is forced to kill itself. Autonomy in a multicellular body is treated as cancer. And yet, the coordination is staggering. Your body contains roughly 37 trillion cells, organised into hundreds of specialised types, each performing specific functions, all communicating constantly through a network of gap junctions, chemical signals, and electrical impulses. The scale alone is difficult to comprehend. If each cell were a person and each gap junction a phone line, your body would be the most densely connected communication network in the known universe. But here's the uncanny part. No single cell is in charge. There's no master control room, no CEO cell issuing orders. The coordination is distributed, emergent. Your heartbeat isn't commanded by your brain. It's generated by the heart's own pacemaker cells, which synchronize through gap junctions, which respond to chemical signals from the nervous system and endocrine system, which are themselves responding to signals from other tissues. It's a conversation with no clear beginning, no single author. Just trillions of cells negotiating moment to moment, using a language invented billions of years ago and refined through evolutionary trial and error. But gap junctions have a range limit. They only work between cells that are physically touching, or nearly so. They're perfect for tissues, for hearts and muscles and epithelial sheets where cells are packed tightly together. But what about communication across distances? What about signals that need to travel from your brain to your gut, or from your pancreas to your liver, or from your pituitary gland to your gonads? For that, multicellular organisms needed a different strategy. They needed to send messages through the environment again, but with far more precision than bacterial quorum sensing could provide. They needed targeted long-distance communication, and so they invented hormones. But before we get to hormones, there's one more implication of gap junctions we need to address, one that cuts to the heart of what it means to be an individual. If your cells are connected through cytoplasmic bridges, if ions and small molecules flow freely between them, then where exactly do you draw the boundary of self? A single cell is easy to define. It's the membrane, the barrier separating inside from outside. But in a multicellular organism, that boundary blurs. Your heart cells share cytoplasm through gap junctions. They are in a very real sense continuous with each other. So are the cells of your nervous system, and your smooth muscles, and your lens epithelium. Functionally, they are not separate individuals but modules of a larger entity. And that larger entity, you, is not a thing but a process. A conversation that has been ongoing since the moment your first cell divided and never stopped. You are not a noun, you are a verb, conjugated across trillions of participants. This is the cost of multicellularity. To gain coordination, to build complexity, to become something larger than a single cell, you must surrender the fiction of independence. You must accept that you are not one but many, and that the many must constantly communicate to remain one. Bacteria can survive alone. You cannot. Remove the communication network, silence the gap junctions, block the chemical signals, sever the electrical connections, and the organism collapses. Not because the cells die immediately, but because they lose coherence. They stop being a body and become a collection of parts. The language fails, and with it, the self. Cooperation solved survival. It allowed cells to build bodies capable of things no single cell could achieve: movement, perception, thought. But it created a new problem, one that seems almost paradoxical. How do you kill yourself for the greater good? Because sometimes, in a multicellular body, cells must die. Not by accident, not by injury, but deliberately. And when they do, they don't just vanish. They speak. They send messages to their neighbours, explaining what's happening, why it's necessary, and what should be done with the remains. Death, it turns out, has a language of its own. There's a moment in your development, back when you were an embryo, about 8 weeks after conception, when your hands were not hands. They were paddles, flat, webbed structures with no individual fingers, just the scaffolding for what fingers would become. And then, over the course of a few days, the webbing disappeared. Not because it was resorbed, not because it stretched and thinned, but because the cells between your future fingers received a message. That message told them to die, and they did. They activated a genetic program that dismantled themselves from the inside out, cleanly, quietly, without inflammation or damage to their neighbours. They committed suicide, and in doing so, they freed your fingers to become separate, articulated, capable of grasping. This process is called apoptosis, programmed cell death, and it's one of the most crucial forms of communication in multicellular life. Because sometimes the most important thing a cell can say is goodbye. Apoptosis is not murder. It's not necrosis, the chaotic death that happens when a cell is injured, poisoned, or starved. Necrotic cells swell, their membranes rupture, and their contents spill out into the surrounding tissue, triggering inflammation and often damaging nearby cells. It's messy, destructive, a failure of cellular integrity. Apoptosis is the opposite. It's controlled, orderly, almost ritualistic. The cell receives a signal, maybe from its neighbours, maybe from its own internal surveillance, systems detecting irreparable DNA damage, and it responds by activating a cascade of enzymes called caspases. These are molecular scissors that cut specific proteins inside the cell, dismantling the cytoskeleton, chopping up the DNA into tidy fragments, and breaking down the organelles. The cell shrinks, its membrane blisters, forming small blebs, and then it packages itself into neat little vesicles, apoptotic bodies, that neighbouring cells or immune cells can engulf and digest without fuss. No inflammation, no collateral damage. The cell is there one moment and then it's gone, as if it had never existed. Except it left a message. According to research published in Cell in 2020, dying cells don't just disappear passively. They actively communicate with their neighbours throughout the process. As apoptosis proceeds, the dying cell releases signalling molecules, find me signals and eat me signals, that attract phagocytes and mark the cell for removal. One of the most important eat me signals is a phospholipid called phosphatidylserine, normally kept on the inner leaflet of the cell membrane hidden from view. But during apoptosis, the cell flips it to the outer surface, displaying it like a flag. It's a universal code: I'm dead, please clear me away. Phagocytes, immune cells that patrol tissues looking for debris, have receptors that recognize phosphatidylserine. They bind to it, engulf the apoptotic body and digest it. The entire process is so efficient that in healthy tissue, you almost never see dead cells accumulating. They're gone before you'd notice them. But that's not the only message apoptosis sends. Dying cells also release signals that influence the behaviour of surviving neighbours. In some contexts, apoptotic signals tell nearby cells to stop dividing, maintaining tissue size and preventing overgrowth. In other contexts, they trigger compensatory proliferation, signals that tell surviving cells to divide and replace the ones that were lost. This happens during wound healing, during normal tissue turnover, and even during development. According to a 2019 study in Nature, dying cells in the developing Drosophila wing disc release a signalling molecule called hedgehog that instructs surrounding cells to proliferate, ensuring that the wing reaches the correct size despite ongoing cell death. The dead are literally shaping the living. Their absence is not silence, it's instruction. And here's where it gets philosophically strange. Why would evolution favour a system where cells willingly kill themselves? It seems counterintuitive. Natural selection is supposed to reward organisms, and by extension cells, that survive and reproduce, not ones that self-destruct. But the logic becomes clear when you remember that in a multicellular organism, the cell is not the unit of selection. The organism is. A cell that refuses to die when it's supposed to, a cell that ignores apoptotic signals and continues dividing, is not just selfish. It's dangerous. It's a potential cancer. Tumour cells are in many ways cells that have forgotten how to listen. They've lost the receptors that detect death signals, or they've mutated the genes that activate caspases, or they've overproduced anti-apoptotic proteins that block the suicide program. They've opted out of the multicellular conversation, and the result is catastrophic for the organism. The cell wins in the short term. It divides uncontrollably, hoards resources, ignores its neighbours. But the organism loses, and eventually when the organism dies, the rogue cell dies too. Evolution solved this problem by making apoptosis non-negotiable. Cells that can't or won't undergo apoptosis when instructed are eliminated over evolutionary time, because the organisms they inhabit don't survive to reproduce. But apoptosis isn't just about quality control, it's about development, sculpting. Your body wasn't built additively, like stacking bricks. It was carved, like a statue emerging from marble. You started as a single fertilized egg that divided into 2 cells, then 4, then 8, then thousands, millions, billions. But not all of those cells remained. Many of them died deliberately to shape your form. The webbing between your fingers, gone. The tissue connecting your eyelids in the womb, gone, apoptosed away so you could open your eyes after birth. The tail you had as an early embryo, yes, you had a tail, a vestigial remnant of our evolutionary past, gone, resorbed through apoptosis. Even your brain was sculpted this way. You were born with far more neurons than you have now. During childhood, your brain went through waves of synaptic pruning, where neurons that weren't being used, weren't forming functional connections, received signals to die. According to research in Neuron published in 2021, roughly half of the neurons generated during early brain development undergo apoptosis before adulthood. The brain you have now is not the brain you were born with. It's the brain that survived. The one carved by experience, by communication, by the conversation between neurons deciding which connections matter and which don't. There's a haunting beauty in this. Every structure in your body exists not just because cells grew there, but because other cells agreed to leave. Your neural circuitry, your skeleton, your immune system, all of it shaped by death as much as by life. And the cells that died did so with language. They sent signals, they marked themselves, they instructed their neighbours on what to do next. This is cooperation at a scale that defies intuition. These cells had no consciousness, no awareness of the larger organism they were building. They couldn't see the hand taking shape, or the brain forming, or the immune system learning to distinguish self from non-self. But they had a language that encoded those outcomes anyway, molecular instructions refined over hundreds of millions of years of evolution. And when their part in the script was finished, they exited quietly, leaving only the message. We were here, we did what was needed. Continue without us. But apoptosis is local. The signals it sends, phosphatidyl serine on the surface, cytokines released into nearby tissue, travel only short distances, microns to millimeters. They coordinate death and replacement within a tissue, but they can't carry information across the body. They can't tell your liver what your brain is doing or inform your muscles that your stomach is empty. For that, multicellular organisms needed a communication system that could operate at scale, across centimeters and meters, integrating information from every tissue and organ. They needed long-distance signaling. They needed hormones. And with hormones came a new kind of complexity. Messages that meant different things depending on who was listening. Your pancreas has no idea what your muscles are doing right now. It can't see them. It can't touch them. It's sitting quietly in your abdomen, behind your stomach, a soft organ about the size of your hand, with no direct physical connection to the skeletal muscles in your arms and legs. And yet somehow it knows when they need fuel. When you exercise, when your muscles are burning through glucose faster than usual, your pancreas responds. It reduces insulin secretion, allowing blood sugar to rise slightly so your muscles can access it. When you eat, when glucose floods into your bloodstream from your intestines, your pancreas detects it and releases insulin, signaling muscle and fat cells to absorb the excess. This coordination happens across distances that, at a cellular scale, are astronomical. From pancreas to muscle is a journey of tens of centimeters, millions of cell lengths. Gap junctions can't bridge that distance. Diffusion-based signaling like quorum sensing would take hours and be hopelessly diluted. So the body uses a different strategy. It sends messages through the bloodstream. It uses hormones. Hormones are molecules, usually proteins, peptides, or steroids, released by specialized cells into the circulatory system. They travel through blood vessels carried passively by fluid flow until they encounter cells with the right receptors. This is communication by broadcast. Unlike gap junctions, which create private channels between adjacent cells, or apoptotic signals, which speak only to immediate neighbors, hormones are public announcements. They're released into the blood and distributed everywhere, to every tissue, every organ. But not every cell listens. Only cells with the appropriate receptor can detect the hormone and respond. It's like a radio signal, transmitted broadly, but only certain receivers are tuned to the right frequency. According to research published in Physiological Reviews in 2018, the human body produces over 50 different hormones, each with specific receptor distributions, creating a communication network of staggering complexity. Insulin, for example, binds to insulin receptors on muscle, fat, and liver cells, but not on neurons. Testosterone binds to androgen receptors in muscle, bone, and reproductive tissues, but has little effect on the pancreas. The message is the same for everyone, but only some are equipped to hear it. This creates an elegant solution to the problem of coordinating a large, complex body. Your brain doesn't need to know the metabolic state of every individual cell. Instead, specialized sensor cells, like the beta cells in your pancreas that detect blood glucose levels, monitor specific variables and release hormones in response. Those hormones travel through the bloodstream, reaching millions of target cells simultaneously, and each target interprets the signal according to its own context. A muscle cell experiencing insulin interprets it as glucose is available, take it in, and store it as glycogen. A fat cell interprets the same insulin molecule as glucose is available, convert it to triglycerides for long-term storage. A liver cell interprets it as stop releasing stored glucose, we have enough circulating now. One message, multiple meanings, depending on who's listening. According to a 2022 study in Cell Metabolism, this context-dependent interpretation is built into the receptor signaling pathways. The same hormone can activate different downstream cascades in different cell types, producing tissue-specific responses. But hormones are slow compared to electrical signaling. A nerve impulse travels at speeds up to 120 meters per second. A hormone, carried by blood flow, moves at maybe half a meter per second, and that's only once it reaches the bloodstream. The actual release, diffusion into capillaries, circulation through the heart, and distribution to distant tissues can take seconds to minutes. This delay matters. Hormones are not for emergencies. When you see a predator, you don't have minutes to respond. Your nervous system handles that. Electrical signals racing from your eyes to your brain to your muscles in milliseconds. But when you need to coordinate slower processes, growth, metabolism, reproduction, stress response over hours or days, hormones are ideal. They provide sustained, widespread signaling that doesn't require continuous input. A single release of growth hormone from the pituitary gland can influence tissues throughout the body for hours. A surge of cortisol in response to chronic stress can alter gene expression in immune cells, neurons, and muscle cells, preparing the body for prolonged challenge. And here's where the communication becomes layered. Hormones don't act in isolation. They form cascades, hierarchies, feedback loops. Your hypothalamus, a small region at the base of your brain, releases hormones that travel a short distance to your pituitary gland. The pituitary responds by releasing its own hormones into the bloodstream, which travel to distant glands, thyroid, adrenals, gonads. Those glands release yet more, hormones which affect tissues throughout the body, and many of these hormones feed back to the hypothalamus and pituitary, modulating their own production. It's a conversation with multiple participants, each response shaping the next signal. According to research in Endocrine Reviews from 2020, these feedback systems allow the endocrine system to maintain homeostasis, stable internal conditions despite external fluctuations. When thyroid hormone levels rise too high, they inhibit the release of thyroid-stimulating hormone from the pituitary, which reduces thyroid hormone production. When they drop too low, the inhibition lifts and production increases. The system self-regulates through communication. But there's a darker side to this broadcast model. Because hormones travel through the bloodstream reaching every tissue, they're vulnerable to disruption. Endocrine disruptors, synthetic chemicals that mimic or block natural hormones, can hijack the system. Bisphenol A, commonly known as BPA, is structurally similar to estrogen. It can bind to estrogen receptors, triggering inappropriate responses or blocking natural estrogen from binding. Phthalates, found in plastics and cosmetics, interfere with androgen signaling. These chemicals don't need to be present in high concentrations. Because the endocrine system is designed to respond to tiny amounts of hormone, concentrations measured in nanograms per milliliter, even trace amounts of disruptors can cause effects. According to a 2021 study in the Lancet Diabetes and Endocrinology, exposure to endocrine disruptors during critical developmental windows can alter growth, metabolism, and reproductive function, sometimes with effects that persist across generations. The language of hormones is so sensitive, so finely tuned, that even whispers from foreign molecules can distort the message. Yet for all their power, hormones still have limits. They're excellent at broadcasting general instructions: store energy, grow, prepare for stress, but they're not precise. They can't target a specific cell or a specific moment with the accuracy of a nerve impulse, and they can't carry complex information. A hormone is essentially a single word: insulin, cortisol, oxytocin. You can't encode a paragraph, a narrative, a detailed instruction set in a hormone. For that, you need a communication system that combines the speed of electrical signaling with the chemical specificity of molecular messages. You need something that can send targeted, rapid, information-rich signals across long distances. You need neurons. But neurons didn't replace hormones. They supplemented them. And in doing so, they created an even stranger layer of communication. One that blurs the line between body and mind, between chemistry and thought. But before we get to neurons, there's one more unsettling discovery about hormonal communication we need to address. Because it turns out you're not the only one producing hormones in your body. Your gut bacteria are too, and they're talking to your brain. There's a nerve in your body called the vagus nerve, and it's unlike almost any other. Most nerves are short, connecting nearby structures, a patch of skin to the spinal cord, a muscle to a motor neuron. But the vagus is a wanderer. Its name literally means wandering in Latin. It originates in your brainstem and travels down through your neck, branching into your heart, your lungs, your stomach, your intestines, even reaching as far as your colon. It's one of the longest nerves in your body, and it carries information in both directions. Signals travel from your brain downward, regulating heart rate, digestion, and inflammation. But just as many signals travel upward, from your gut to your brain, carrying information about the state of your digestive system, the presence of nutrients, the composition of your microbiome. This is the anatomical basis of the gut-brain axis, a communication highway that connects two organs we don't usually think of as related. And the conversation happening along that highway is far stranger than anyone suspected until recently. Because here's what we now know. Your gut is not just digesting food, it's hosting trillions of bacteria, fungi, viruses, and archaea, collectively called the microbiome, that outnumber your own cells. These microbes live in a complex ecosystem along the lining of your intestines, especially in the colon, where they ferment indigestible fibers, produce vitamins, train your immune system, and compete with pathogens. But they're also doing something else. They're producing neurotransmitters, the same signaling molecules your neurons use to communicate with each other. Serotonin, dopamine, GABA, norepinephrine, chemicals we associate with mood, cognition, and emotion, are being synthesized by bacteria in your gut. According to research published in Nature Reviews Neuroscience in 2022, approximately 90% of the body's serotonin is produced in the gut, primarily by enterochromaffin cells in the intestinal lining, but also by certain bacterial species. And while most of this serotonin doesn't cross the blood-brain barrier directly, it doesn't need to. It acts locally on the enteric nervous system, the network of neurons lining your gut, sometimes called the second brain, and sends signals up the vagus nerve to the brain. This means bacteria are influencing your nervous system, not metaphorically, not indirectly, literally. They're producing molecules that neurons respond to, and those responses propagate up to your brain. Studies in germ-free mice, animals raised in sterile conditions with no gut bacteria, have shown dramatic differences in behavior stress response and neurochemistry compared to normal mice. According to a 2020 study in Cell, germ-free mice exhibit increased anxiety-like behaviour, altered social interactions, and changes in the expression of genes related to neurodevelopment. When researchers colonize these mice with gut bacteria, many of these effects reverse. The bacteria are communicating with the brain, and the brain is listening. But what exactly are they saying? The signals are complex, layered, and context-dependent. Some gut bacteria produce short-chain fatty acids, butyrate, propionate, acetate, as byproducts of fermenting dietary fibre. These molecules can cross into the bloodstream, reach the brain, and influence the production of brain-derived neurotrophic factor, a protein critical for neuron growth and plasticity. Other bacteria produce metabolites that modulate inflammation, which affects mood and cognition. Chronic low-grade inflammation, driven in part by an imbalanced microbiome, has been linked to depression, anxiety, and even neurodegenerative diseases like Alzheimer's. According to research in Gut Microbes published in 2021, the composition of the microbiome, the relative abundance of different bacterial species, correlates with mental health outcomes. People with depression, for instance, tend to have lower microbial diversity and reduced populations of certain beneficial bacteria like Faecalibacterium and Coprococcus. But the relationship isn't one-directional. Your brain talks back to your gut. Stress, anxiety, and emotional states alter gut motility, secretion, and permeability through signals sent down the vagus nerve and through the release of stress hormones like cortisol. These changes shift the gut environment, favouring some bacterial species over others. It's a feedback loop. Your mental state shapes your microbiome, and your microbiome shapes your mental state. You're not just hosting bacteria, you're negotiating with them. They provide metabolites, neurotransmitters, and immune signals that influence your behaviour. You provide shelter, food, and a stable environment. It's symbiosis, but it's also communication, chemical messages exchanged between kingdoms of life, between human cells and bacterial cells, mediated by molecules that predate both. And here's where it gets philosophically uncomfortable. If bacteria in your gut are producing serotonin, dopamine, and GABA, the neurotransmitters that regulate your mood, motivation, and cognition, then who's in control? When you feel anxious, is that you, or is that your microbiome signaling distress? When you crave certain foods, is that your brain's decision, or are gut bacteria manipulating your preferences to favour their own survival? Research suggests bacteria can indeed influence host eating behaviour. According to a 2019 study in Bioassays, certain gut bacteria produce metabolites that affect vagal signalling and reward pathways in the brain, potentially driving cravings for the nutrients those bacteria need. It's not mind control in the science fiction sense. There's no intentionality, no consciousness behind it. But it is influence, it's communication shaping behaviour, and it raises an unsettling question. Where do you end, and the ecosystem inside you begin? The gut-brain axis reveals something fundamental about multicellular life that we often ignore. You're not a single organism, you're a collection of organisms, human cells, bacterial cells, fungal cells, viral particles, living in constant communication, negotiating resources, coordinating responses, influencing each other's behaviour. The language they use is chemistry, neurotransmitters, metabolites, immune signals, hormones. The same molecular vocabulary that cells have been using for billions of years, now operating at a scale and complexity that creates something new. Not just cooperation, but integration. A human being is not a human alone. It's a human plus microbiome plus mitochondria, descendants of ancient bacteria that merged with our ancestors a billion years ago and still retain their own DNA, their own communication systems. You're a conversation between multiple genomes, multiple species, multiple evolutionary lineages, all negotiating to produce the entity you experience as yourself. But the gut-brain axis is just one example of this distributed communication. Your immune system is having similar conversations constantly. Immune cells patrol your body, sampling molecules, detecting pathogens, communicating threats to each other through cytokines and chemokines. They're making decisions, what to attack, what to tolerate, when to trigger inflammation, based on signals from bacteria, from damaged cells, from your own tissues. You don't consciously control these decisions. You're not aware of them happening, but they shape your health, your energy levels, your susceptibility to disease. They are, in a very real sense, part of your cognition. Distributed cognition. Intelligence that emerges not from a central processor, but from millions of communicating agents, each responding to local information, together producing coherent behavior. So if bacteria can talk to your brain, and your immune cells can make decisions, and your heart cells can coordinate without input from your brain, then what is the brain actually doing? What makes neural communication special? The answer is speed, complexity, and integration. Neurons are the most sophisticated communication specialist evolution has ever produced. They combine electrical and chemical signaling in ways that allow information to travel faster, encode more complexity, and integrate across greater scales than any other cell type. They didn't replace the older forms of communication: hormones, gap junctions, paracrine signaling, immune cytokines. They layered on top of them, and in doing so, they created something unprecedented: the ability to model the world, to simulate scenarios, to remember the past and anticipate the future. Neurons didn't just communicate, they built minds. A neuron is, at its core, just a cell. It has a nucleus, mitochondria, ribosomes, all the same machinery as any other cell in your body, but it's been modified, specialized, pushed to an extreme. Its membrane is studded with ion channels, proteins that open and close in response to voltage changes, allowing charged particles like sodium, potassium, and calcium to flow in and out. Its shape is wildly elongated, with a cell body that branches into dendrites, short tree-like extensions that receive signals, and a single axon that can stretch extraordinary distances. The longest neuron in your body runs from the base of your spine to your big toe, nearly a meter long. That's a single cell, one continuous strand of cytoplasm enclosed in membrane, spanning a distance equivalent to millions of bacterial cell lengths, and along that length, it can transmit information at speeds up to 120 meters per second. This is communication at a scale and speed that no other cell type can match. Neurons didn't just refine the language cells had been speaking for billions of years. They accelerated it. They amplified it. They turned whispers into lightning. The key innovation is the action potential, a rapid, self-propagating wave of electrical charge that travels along the axon. Here's how it works. At rest, a neuron maintains an electrical imbalance across its membrane. The inside is negatively charged relative to the outside, about -70 millivolts. This is maintained by ion pumps that actively push sodium out and pull potassium in, fighting against the natural tendency of these ions to diffuse. But when the neuron receives enough excitatory input, neurotransmitters binding to receptors on its dendrites, opening ion channels, the voltage starts to shift. Sodium rushes in. If the voltage crosses a threshold around -55 millivolts, something dramatic happens. Voltage-gated sodium channels snap open all along the membrane. Sodium floods in, and the inside of the cell briefly becomes positive, around +40 millivolts. This depolarization triggers adjacent sodium channels to open, and the wave propagates down the axon like a line of falling dominoes. Within milliseconds, potassium channels open, potassium rushes out, and the voltage resets. The whole cycle, depolarization and repolarization, takes about 1/1000th of a second. This is exponentially faster than any chemical diffusion. A hormone traveling through your bloodstream might take 30 seconds to reach a target organ. A neurotransmitter diffusing across a synaptic gap takes less than a millisecond, but it only travels 20 nanometers. The action potential travels meters in milliseconds, carrying information from your brain to your fingertips before you're consciously aware you've decided to move. According to research published in Nature Neuroscience in 2019, the speed of action potentials is further enhanced in many neurons by myelination, insulating sheaths of fatty tissue wrapped around axons by glial cells. Myelin forces the action potential to jump between gaps, called nodes of Ranvier, in a process called saltatory conduction. This increases transmission speed up to 50 times compared to unmyelinated axons. It's an evolutionary arms race. Faster communication means faster reactions, better survival, more successful reproduction. Organisms with faster neurons outcompeted those without. but speed is only part of the neural revolution. The other part is complexity. Neurons don't just send binary signals, fire or don't fire. They integrate information. A single neuron can receive input from thousands of other neurons, each synapse contributing either an excitatory signal, pushing the membrane voltage toward threshold, or an inhibitory signal, pushing it away. The neuron sums these inputs, weighing them against each other in real time. If the total excitatory input exceeds the total inhibitory input by enough to cross threshold, the neuron fires. If not, it stays silent. This is computation. The neuron is performing a calculation, a weighted sum of its inputs, and producing a binary output. And because neurons are connected in vast networks, with each neuron's output becoming input for many others, the collective behavior produces something far more sophisticated than any individual neuron could achieve. According to Research in Science from 2020, the human brain contains approximately 86 billion neurons, each forming an average of 7,000 synaptic connections. That's roughly 600 trillion synapses, 600 trillion points of communication, each one dynamically adjustable, each one capable of strengthening or weakening based on experience. This adjustability is crucial. It's called synaptic plasticity, and it's the mechanism underlying learning and memory. When two neurons fire together repeatedly, when the presynaptic neuron signal consistently contributes to the postsynaptic neuron's firing, the synapse between them strengthens. More receptors are inserted into the postsynaptic membrane. The presynaptic terminal releases more neurotransmitter. The connection becomes more efficient. This is Hebbian plasticity, often summarized as cells that fire together, wire together. It's how your brain encodes experience. Every time you practice a skill, every time you form a memory, you're strengthening specific synapses, making certain communication pathways more likely to activate in the future. Conversely, synapses that aren't used weaken and eventually disappear. Your brain is constantly rewriting its own communication network based on experience, sculpting itself through the language of electrical and chemical signals. But here's the eerie part. No neuron knows what the network knows. An individual neuron can't represent a thought, a memory, a sensation. It can only fire or not fire in response to its inputs. The meaning, the information content, emerges from patterns of firing across populations of neurons. When you see a face, it's not a single neuron lighting up with face detected. It's thousands of neurons in your visual cortex firing in a specific spatiotemporal pattern, and that pattern is what your brain interprets as a face. When you remember your childhood home, it's not stored in one place. It's distributed across multiple brain regions, visual details in occipital cortex, emotional associations in amygdala, spatial layout in hippocampus, all reactivating in synchrony. The memory is the pattern, the thought is the conversation, and no single participant in that conversation has access to the whole. Each neuron only knows its own state and the signals it's receiving from its neighbours. The larger meaning is emergent, inaccessible to the parts that create it. This is fundamentally different from a computer, where information is stored in discrete locations and a central processor retrieves and manipulates it. In the brain, there is no central processor, no command center, no CEO neuron issuing orders. The organization is distributed, parallel, redundant. Damage to one region can often be compensated for by others, because the information is spread across the network. According to a 2021 study in Neuron, this distributed architecture makes the brain incredibly robust to local failures, but also makes it difficult to pinpoint where specific functions live. Consciousness, memory, decision-making, these aren't located in a particular place. They're properties of the network as a whole, of the communication structure, not of any individual cell. And the network is dynamic. It's not a fixed circuit. Neurons are constantly adjusting their connections, forming new synapses, pruning old ones, modulating the strength of existing ones. Every experience, every thought, every sensory input subtly reshapes the network. You are, quite literally, a different person at the level of neural connectivity than you were yesterday. The continuity you experience, the sense of being the same self over time, is an illusion created by the relatively slow pace of synaptic change and the persistence of certain core patterns. but the network itself is in flux. The conversation is ongoing, and the you that emerges from that conversation is not a thing but a process, a pattern of activity that persists through constant turnover of its components. But if meaning emerges from patterns, if no neuron has access to the whole, if your sense of self is a distributed property of 86 billion communicating cells, then where does consciousness come from? When does the conversation between neurons become the experience of being someone? That's the hard problem, the mystery at the centre of neuroscience, philosophy, and perhaps all of science. And the answer, if there is one, lies in understanding not individual cells, but the language they speak, the patterns, the rhythms, the emergent coherence that arises when trillions of signals align into something that can look back at itself and ask, "What am I?" There's an experiment you can do right now that reveals something strange about consciousness. Close your eyes and try to locate where. You are not your body, you know where that is, but you, the experiencer, the one who's reading these words, making decisions, having thoughts. Where is that located? Most people, if pressed, will point to somewhere behind their eyes, in the centre of their head. That's where it feels like you are, the command centre, the place where experience happens. But here's the problem, there is no command centre. If a neurosurgeon were to open your skull and search for the place where consciousness lives, they'd find only neurons, billions of them, firing in patterns, sending signals to each other through synapses. No single cell is conscious. No particular region, when isolated, contains your sense of self. You can remove large portions of the brain through surgery, stroke, or injury, and consciousness persists, often with deficits but still present. So where is it? How does a collection of unconscious cells, each one just responding to chemical signals from its neighbours, produce the unified experience of being someone? This is the hard problem of consciousness, and nobody has solved it. We can describe the neural correlates of consciousness, the patterns of brain activity that correlate with specific experiences. We can map which regions activate when you see red, or hear music, or feel pain. According to research published in Nature Reviews Neuroscience in 2020, certain signatures appear consistently across conscious states. Widespread synchronization of neural firing, particularly in gamma frequency oscillations around 40 Hz, information integration across distributed networks, and recurrent feedback loops where sensory areas send signals to higher processing regions, which send predictions back down. When these patterns are present, people report being conscious. When they're absent, during deep sleep, general anesthesia, or certain brain injuries, consciousness fades. But correlation is not explanation. Knowing which neurons fire during consciousness doesn't tell us why those firing patterns feel like something. Why there is an experience at all, rather than just information processing happening in the dark. One influential theory, integrated information theory, proposes that consciousness is a property of systems that integrate information in specific ways. According to this framework, developed by neuroscientist Giulio Tononi and colleagues, a system is conscious to the degree that it forms a unified whole that's more than the sum of its parts, where the state of the whole cannot be reduced to independent states of the components. A brain, with its densely interconnected neurons forming feedback loops and distributed representations, has high integrated information. A collection of isolated neurons or a simple computer performing serial calculations has low integrated information. The theory predicts that consciousness isn't binary. It's a spectrum, and it's not unique to humans or even to animals with brains. Any system with sufficient integration could, in principle, have some degree of experience. According to a 2023 paper in Science Advances testing predictions of integrated information theory, brain regions that are highly interconnected and show strong feedback dynamics correlate with reports of conscious experience, while regions that process information in a more feed-forward, modular way do not. But even if integrated information theory is correct, and it remains controversial, it doesn't fully resolve the mystery. It tells us what kind of system can be conscious, but not why integration produces experience. Why does it feel like something to be an integrated information system? This is where the cellular perspective becomes illuminating, even if it doesn't provide a complete answer. Because if we trace consciousness back through evolutionary history, we see that it didn't emerge suddenly, fully formed. It's the endpoint of a long trajectory of cellular communication becoming more complex, more integrated, more capable of modelling and responding to the environment. Single-celled organisms already exhibit primitive forms of information integration. They sense chemical gradients, respond to touch, move toward food and away from toxins. They have memory in a limited sense. Bacteria can remember past infections and mount faster immune responses the next time. But they lack the architecture to integrate information across multiple timescales and modalities simultaneously. Multicellular organisms, once they evolved nervous systems, could integrate more. A hydra, a simple animal with a diffuse nerve net and no brain, can coordinate its tentacles, pull in food, contract when threatened. There's more integration than in a bacterium, but still no centralized processing. As nervous systems centralized, as brains evolved, the degree of integration increased. More regions talking to each other, more feedback loops, more capacity to hold representations of the world stable over time. And somewhere along that continuum, integration became rich enough, recursive enough, that it produced something new. Not just responsiveness to the environment, but an internal model of the environment. Not just processing information, but experiencing it. The exact threshold is unclear. We don't know if insects are conscious. We're not certain about fish. But by the time you reach birds and mammals, the evidence is strong. Complex behaviours, self-recognition in mirrors, flexible problem-solving, signs of suffering and pleasure. The conversation between neurons has become sophisticated enough to reflect on itself. And here's the part that connects back to the beginning. The language neurons use to create consciousness is the same language cells have always used. Neurotransmitters are just signalling molecules, the same category as bacterial autoinducers or hormones or apoptotic signals. Synapses are just specialized communication channels, refined versions of gap junctions and receptor-ligand interactions. The action potential is an elaboration of the voltage changes that even bacteria can produce. There's no fundamentally new vocabulary. What changed is the grammar, the structure, the density, the speed, the feedback, the integration. Neurons took the ancient language of cellular communication and wrote literature with it. They wrote symphonies. They wrote questions like, "What am I?" and had the capacity to search for answers. But here's the unsettling implication. If consciousness emerges from cellular communication at a certain threshold of complexity and integration, then you, the unified self that feels like a single entity, are not. You are a process, a pattern, a conversation between 86 billion neurons that produces the illusion of singularity. There is no homunculus, no little person inside your head watching the show. There are only cells talking to each other, and somehow, from that conversation, the experience of being you arises. According to philosopher Thomas Metzinger's work on the self-model theory of subjectivity, what we call the self is a construct, a model your brain builds to represent the organism as a whole, a useful fiction that simplifies the overwhelming complexity of coordinating trillions of cells. The model is so convincing that you mistake it for a fundamental truth. You feel like a unified agent making decisions, but the decisions emerge from distributed negotiations between brain regions, many of which you're not aware of. The conscious self is the last to know, receiving a simplified summary of conclusions reached through processes you don't have access to. This isn't to say the self is meaningless, or that consciousness is an illusion in the sense of being false. The experience is real. The subjective feeling of being someone, of having continuity over time, of making choices, these are real phenomena produced by real neural activity. But the interpretation, the story your brain tells about what's happening, is a construction. And recognizing it as such has philosophical weight. It means the boundaries of self are less clear than they seem. Where do you end? At your skin. But your microbiome influences your thoughts, your hormones shape your emotions, your environment modulates your gene expression. You are not a closed system. You're a node in a web of communication that extends outward, to other people, to ecosystems, to the physical and chemical environment. The conversation that makes you doesn't stop at your membrane. It includes the signals you receive from the world and the signals you send back. And this brings us to the strangest realization of all. If human consciousness is cellular communication scaled up, then human language, the words you're reading right now, is cellular communication scaled up again. When I write a sentence, neurons in my brain fire in patterns that encode meaning. Those patterns trigger motor neurons that move my fingers. My fingers type symbols onto a screen. Light from the screen enters your eyes, where photoreceptors convert it into electrical signals. Those signals travel to your visual cortex, triggering patterns of neural firing that reconstruct the meaning I encoded. Your neurons are now firing in patterns influenced by my neurons, across space and time, mediated by symbols. This is cellular communication between bodies, the language of cells, externalized. And every conversation you've ever had, every book you've read, every song that moved you, it's all an extension of the same process that began 4 billion years ago, when one cell sent a chemical signal and another cell responded. When you speak, you think you're doing something uniquely human, something that separates you from every other organism on the planet. You're using words, arbitrary symbols strung together according to grammatical rules, to transmit thoughts from your mind into someone else's. No other species does this, at least not at this level of complexity. Birds have songs, whales have calls, bees have dances, but none of them have syntax recursion, the ability to generate infinite novel meanings from finite elements. Language, we tell ourselves, is the line that divides us from the rest of the living world. It's what made civilization possible, what allowed us to accumulate knowledge across generations, to coordinate at scales beyond small tribes, to build cities and write symphonies and argue about philosophy. Language is the tool that made us human. But here's the reframe. Language didn't appear out of nowhere. It's not a discontinuity, not a miracle that descended on one species 100,000 years ago. It's an extension, an elaboration, the latest iteration of a communication system that's been developing for 4 billion years. When you speak a sentence, you're using the same basic mechanism that bacteria use for quorum sensing, that neurons use for synaptic transmission, that dying cells use to signal their neighbours. You're detecting a signal, processing it, and producing a response. The signal just happens to be sound waves encoded with symbolic meaning instead of molecules encoded with chemical meaning. But the underlying logic, sender, message, receiver, response, is ancient. Language is cellular communication that learned to leave the body. Think about what happens when you hear a word. Sound waves, pressure fluctuations in air, enter your ear and vibrate the eardrum. Those vibrations are transmitted through tiny bones to the cochlea, where they bend hair cells, which convert mechanical motion into electrical signals. Those signals travel along the auditory nerve to your brainstem, then up to your auditory cortex, where neurons fire in patterns that correspond to the frequency and amplitude of the sound. Other regions, Wernicke's area, parts of the temporal lobe, activate matching the sound pattern to stored representations, extracting meaning. If the sound is a word you know, the meaning is retrieved almost instantly. If it's a sentence, your brain parses the syntax, identifies subjects and verbs and objects, constructs a mental model of what's being described. All of this happens in milliseconds, unconsciously. And the mechanism, at its core, is neurons signaling to other neurons through chemical and electrical messages. According to research published in Nature Communications in 2021, language processing involves synchronized activity across multiple brain regions, with information flowing from auditory cortex to frontal regions and back in recurrent loops, integrating sound with meaning with context in real time. Now think about what happens when you speak. You have an idea, a pattern of neural activity representing a thought. Your brain translates that pattern into a sequence of phonemes, the basic sound units of language. Motor regions, Broca's area, motor cortex, activate, sending signals down to your vocal cords, tongue, lips, diaphragm. Muscles contract in precise coordination, modulating airflow, shaping sound waves. The sound propagates through the air, and if someone is listening, it enters their ear, and the whole process reverses. Your neural pattern has been converted into sound, transmitted through a medium, and reconstructed as a neural pattern in someone else's brain. This is information transfer. It's encoding, transmission, and decoding. And it's functionally identical to what happens when a neuron releases neurotransmitters that cross a synapse and bind to receptors on another neuron. The scale is different, the medium is different, but the structure is the same, and just like cellular communication, language is context dependent. The same sound can mean different things depending on tone, emphasis, surrounding words, shared knowledge between speaker and listener. The word bank means one thing in I went to the bank and another in the river bank. Your brain disambiguates based on context, just like a cell interprets a hormone differently, depending on which receptors it has and what other signals it's receiving simultaneously. According to a 2022 study in psychological science, meaning in language is never fully contained in the words themselves. It's constructed by the listener based on prior knowledge, expectations, and situational cues. This is why miscommunication happens so easily. The speaker encodes meaning based on their mental model of the world. The listener decodes based on theirs. If the models don't align, the message distorts. It's the same problem cells face when signaling across noisy environments, or when receptors are damaged, or when the concentration of a signaling molecule falls below threshold. The message is there, but it's not received as intended. But language does something cells alone can't do. It externalizes memory. When neurons communicate, the information exists only as long as the pattern of firing persists, or as long as synaptic changes encode it in the network. When you forget something, it's because the synaptic pattern weakened or was overwritten. But language allows information to escape the brain. It can be written down, stored in books, inscribed on stone, encoded in digital media. The thought pattern that existed in one brain can be reconstructed in another brain decades, centuries, millennia later. According to anthropological research, this externalization of memory is what enabled cumulative culture, the ability to build on previous generations' knowledge without having to rediscover everything from scratch. A child born today inherits not just genes, but language, and with language comes access to the accumulated knowledge of every human who's contributed to the linguistic record. It's inheritance of acquired characteristics, something evolution through genes alone doesn't allow. Language is a second evolutionary system, running in parallel to biological evolution, and it operates on much faster timescales. And here's the deeper connection. Language isn't just about transmitting facts. It's about coordinating behavior, building shared reality, creating social bonds. When you tell a story, you're not just conveying information. You're synchronizing your mental model with the listeners. You're creating a shared experience, a common framework for interpreting the world. This is what quorum sensing does for bacteria. It synchronizes their behavior based on shared chemical information. This is what gap junctions do for heart cells. They synchronize contractions based on shared electrical signals. Language synchronizes minds. It allows humans to coordinate at scales far beyond what any individual could manage alone, to build institutions and economies and cultures that persist across generations. It's the ultimate expression of the principle that drove the very first cells to communicate. Cooperation amplifies survival. But language also reveals something about the limits of cellular communication when it tries to describe itself. Every word I've written, every explanation I've given, is using patterns of neural activity to point at patterns of neural activity. I'm using the language of cells to describe the language of cells, and there's a recursive loop here, a strange self-reference. You can't step outside the system to observe it objectively, because the observer is made of the same communication networks being observed. This is why consciousness is hard to explain, why the self feels irreducible even though it's clearly constructed from parts. The tools we're using to investigate language, thought, scientific reasoning, are themselves products of cellular communication. We're asking neurons to explain neurons, using signals to explain signals. It's not impossible. But it means every answer will be, at some level, a translation, a metaphor, a model rather than the thing itself. And maybe that's okay. Maybe the point isn't to find a final, complete explanation, but to recognize the continuity, to see that the conversation you're having with yourself right now, the internal monologue, the stream of thoughts, is not fundamentally different from the conversation your neurons are having with each other, which is not fundamentally different from the conversation your gut bacteria are having with your brain, which is not fundamentally different from the conversation ancient bacteria had in the primordial ocean. It's all the same language, spoken at different scales with different vocabularies, producing different kinds of meaning. But the grammar remains, signal and response, message and interpretation, the dance of molecules and membranes and electrical potentials, cycling endlessly, creating the illusion of separation, of sender and receiver, of self and other, when really it's always been one continuous exchange. Language didn't make us human. It revealed what we'd been all along, participants in the oldest conversation on Earth. Right now you are speaking without words. We started with that line, and now, tens of thousands of words later, we return to it. But it means something different now, because you've seen what was invisible before. You've traced the thread backward, from the sentence you just read, through the neural signals that decoded it, through the synaptic conversations that constructed meaning, through the hormones and gap junctions and apoptotic signals that built your body, through the bacterial negotiations happening in your gut, through the quorum sensing that taught cells to count, all the way back to the first molecule that meant something to someone else. The language of cells isn't a metaphor. It's not a poetic way of describing chemistry. It's literal. Every interaction between molecules that carries information, every signal that produces a coordinated response, every threshold crossed that changes behavior, that's language, and it's been speaking since the beginning. But here's what we didn't say explicitly until now. Language isn't what separates you from the rest of life, it's what connects you to it. Every word you speak is made possible by neurons, which are made possible by multicellular coordination, which is made possible by gap junctions and hormones, which are made possible by membrane receptors that evolved from bacterial sensing systems. You're not using language, you are language, embodied, scaled up, reflected back on itself until it became aware of its own existence. The conversation that started 4 billion years ago didn't end. It complexified, it accelerated, it built bodies and brains and cultures, but it never stopped. You're a chapter in an unfinished sentence, a clause in a dialogue that has no final punctuation. And the most staggering part is this. None of the participants in that conversation know the whole story. The bacteria in your gut don't know they're influencing your mood. Your immune cells don't know they're making decisions. Your neurons don't know. They're constructing consciousness. Each cell, each molecule, each ion channel is responding only to local information, the signals it receives from its immediate neighbours. But from those local interactions, global coherence emerges. Your heartbeat, your thoughts, your sense of being a unified self. These are not commanded from above. They are negotiated from below, through trillions of simultaneous exchanges, each one blind to the larger pattern it's creating. According to research in complexity science, this is the hallmark of emergent systems. Order arising from decentralized communication, without a blueprint, without a conductor, without any single agent understanding the whole. This is both humbling and profound. Humbling because it means you're not as in control as you feel. The decisions you make, the emotions you experience, the preferences you have, they emerge from processes you're not conscious of, negotiations between cells and systems that were underway long before you became aware of them. But it's also profound because it means you're never alone. You are quite literally a community, a walking ecosystem, an alliance of genomes, human, bacterial, mitochondrial, viral, all communicating, all contributing to the entity that calls itself by your name. According to a 2023 study in Cell Systems, the average human body contains not just 37 trillion human cells, but an estimated equal number of bacterial cells, plus viruses, fungi, and archaea. You are, by cell count, roughly half non-human, and all of those cells are in constant dialogue, using the same molecular language refined over billions of years. So when you talk to another person, what's really happening? Your neurons fire, encoding a thought into motor commands. Your muscles contract, producing sound waves. Those waves travel through air, a medium made of molecules bouncing against each other, transmitting vibration. The waves reach another person's ear, where they're converted back into neural signals. And in their brain, patterns activate that approximate the patterns in yours. Information has moved from one network of communicating cells to another, across the gap between bodies. Your cells talking to cells, mediated by air and symbols and grammar. Every conversation is, at root, the same kind of communication that happens inside you. Signals sent, received, interpreted, responded to. The boundary between internal and external communication is thinner than it seems. Speaking is just another synapse, stretched across space. And this explains something we often forget. Communication is never perfect. Misunderstandings aren't failures, they're inevitable, because meaning is always constructed by the receiver based on their own context, their own history, their own network state. When you speak, you're not transferring a thought directly from your mind to someone else's. You're sending a signal, encoded in words, gestures, tone, and hoping their brain reconstructs something close to what you meant. Sometimes it works, often it doesn't. This is the same problem cells face. Hormones sent through the bloodstream might not reach their target if the receptors are damaged. Neurotransmitters might not bind if the synaptic gap is too wide. Bacterial signals might be jammed by competing species. Every level of biological communication deals with noise, with distortion, with the fundamental uncertainty of whether the message was received as intended, and yet, despite this, coherence emerges. Bodies function, ecosystems stabilize, societies form. The conversation continues, imperfect but persistent. There's a question people ask sometimes, usually late at night, usually after contemplating the vastness of space or the strangeness of existence. Are we alone in the universe? And the answer, viewed through the lens of cellular communication, is no. Not in the way the question usually means. We don't know if there's intelligent life on other planets. But you're not alone in a deeper sense. You've never been alone. From the moment the first cell budded off from your fertilized egg, you've been in conversation. With your own cells, with your microbiome, with the people around you, with the accumulated voices of every human who contributed to the language you speak. You're not an isolated consciousness floating in a biological machine. You're a node in a network that extends across time and space, connected through the language of molecules and symbols and meaning. And here's the final turn. The first conversation, the one between two ancient cells in a primordial ocean, never had a clear beginning. We can trace it back to quorum sensing, to receptor-ligand binding, to the first membranes that separated cell from environment. But those membranes were made of molecules that interacted according to chemical rules, and those rules are themselves a form of communication. Atoms signaling to each other through electromagnetic forces, electrons sharing information about energy states. Go back far enough, and communication dissolves into physics. The universe has been exchanging information since the Big Bang, matter and energy in constant interaction, following laws that encode relationships, constraints, possibilities. Life didn't invent communication, it inherited it, it refined it. It turned the background hum of chemistry into something that could produce meaning, memory, consciousness. So maybe the right way to think about language, cellular, neural, human, is not as a tool we use, but as a process we participate in. A process older than life itself, woven into the structure of reality. You speak because cells speak. Cells speak because molecules speak. Molecules speak because the universe is, at its foundation, relational. Nothing exists in isolation. Everything is connected through exchange of forces, of particles, of information. The language of cells is just one dialect in a conversation that includes gravity pulling galaxies together, photons carrying information across space, quantum entanglement linking particles instantaneously across distances. It's all communication, all the way down. And this brings us back to you, reading these words, neurons firing, meaning constructed, the ancient language flowing through circuits built over billions of years of evolutionary refinement. You started this journey thinking language was something humans invented. Now you know it's something you inherited. The first language wasn't words, it was chemistry, electricity, the meeting of the molecules across a membrane. But every word since has been written in it. Every thought you think, every connection you make, every moment of understanding or confusion or wonder, it's all an expression of the same conversation that began in the dark, when life was nothing but chemistry learning to listen. The conversation continues, in your heart, your gut, your brain, in the spaces between people, in the accumulated knowledge of civilizations. It will continue after you, through the cells that outlive your body, through the words you leave behind, through the patterns you set in motion that ripple outward in ways you'll never see. You're not the author of this conversation, you're a sentence within it. But what a sentence! Complex, recursive, self-aware, capable of looking back at its own structure and marvelling at the improbability and beauty of it all. The first language wasn't words, but every word since has been written in it, and the last word, if there ever is one, will be too. Beneath the surface of every pond, every ocean, every drop of water on Earth, something is spinning. Right now, as you sit still, trillions of microscopic motors are running inside you, in your lungs, in your brain, in your throat. They're beating in perfect rhythm, coordinating without commands, moving things you'll never see. They've been running since the moment you were born, and they haven't stopped once. These aren't machines in the way you think of machines. They're molecules. Proteins folded into shapes that can spin, bend, and beat. They're 50,000 times smaller than the width of a human hair, and they move faster than anything you'll ever touch. But here's what we didn't understand until recently. These motors are ancient, older than animals, older than plants, older than anything with tissues or organs or a body plan. They were life's first solution to an impossible problem. How do you move when you don't have muscles? How do you swim through water that feels like concrete? How do you coordinate thousands of moving parts when you don't have a brain to control them? Life solved these problems 3 billion years ago, and the solution, molecular motors spinning inside cells, became the blueprint for every movement in every living thing that would ever exist. Tonight, we're going inside the machinery of motion itself, from the first spinning engines in ancient bacteria to the coordinated arrays powering cells in your body right now. We'll explore how life learned to move in a world where the physics are alien, where swimming shouldn't work, and where motion requires solutions stranger than anything humans have engineered. Because understanding these motors changes everything. It reveals that movement didn't begin with muscles, it began with chemistry that learned to spin. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a single bacterium in a drop of pond water. It's smaller than a dust mote, smaller than a pollen grain. So small that if you lined up 10,000 of them end to end, they'd barely cross the period at the end of this sentence. From your perspective, it's invisible. But from its perspective, it's navigating a universe with completely different rules. The bacterium has a tail, a flagellum, and it's spinning. Not waving back and forth like a fishtail or undulating like an eel. Spinning. Like a propeller, and at the base of that tail, anchored in the cell membrane, is a motor, a molecular motor. It's built from about 30 different proteins, each one locking into place with atomic precision. There are rings that rotate, a rotor that spins, a stator that holds everything in place. It looks engineered, but no one designed it. It assembled itself, protein by protein, guided by nothing more than chemical attraction, and the shape of molecules folding in just the right way. And this motor is fast. The flagellum spins at about 100,000 rotations per minute. If you scaled that up to the size of a car wheel, it would be spinning faster than a jet turbine, and it runs constantly. For hours, for days, for the entire life of the cell. It doesn't overheat, it doesn't wear out, it just keeps spinning. But here's what makes it extraordinary. It's spinning through water that doesn't behave like the water you know. At the bacterium scale, water isn't a fluid, it's thick, dense, almost solid. If you were shrunk down to bacterial size and tried to swim through water, it would feel like swimming through honey, or tar, or wet concrete. Every stroke would require enormous effort, and the moment you stopped pushing, you'd stop moving. Instantly. There's no coasting at this scale, no gliding, no momentum carrying you forward. This is the world of low Reynolds number. It's a regime of physics most of us never experience. In our world, at human scales, inertia dominates. When you throw a ball, It sails through the air. When you dive into a pool, you glide forward even after you stop kicking. Momentum carries you, but shrinks small enough, and inertia disappears. Viscosity takes over completely, and motion becomes something else entirely. Physicist Edward Purcell described it in a famous lecture in 1977. He said that at low Reynolds number, moving through water is like trying to swim in molasses while someone pours more molasses on you from above, and he proved mathematically that certain types of swimming simply can't work. If you open and close your body symmetrically, like a scallop opening and closing its shell, you go nowhere. The water you push forward on the opening stroke comes right back on the closing stroke. Zero net displacement. It's called the scallop theorem. At low Reynolds number, reciprocal motion is useless. So bacteria can't swim like fish. They can't undulate their tails back and forth symmetrically. That would leave them motionless, trapped in the viscous world. They had to invent something else. They invented rotation. The bacterial flagellum doesn't wave. It spins in one direction, like a corkscrew drilling through the viscosity. The motion is asymmetric. Every rotation is different from every other rotation in time, and that asymmetry generates thrust, slowly, inefficiently by our standards, but it works. The bacterium moves forward at about 20 micrometers per second. That's slow in absolute terms. A snail would laugh. But relative to its body size, it's fast. 20 micrometers per second is about 10 body lengths per second. If you scaled that up to human size, you'd be running at 60 miles per hour, and the bacterium can change direction. When the motor spins counterclockwise, the flagellum pushes the cell forward in a straight line. But when the motor reverses and spins clockwise, the flagellum comes apart. Multiple flagella that were bundled together separate, the cell tumbles randomly, and when the motor switches back to counterclockwise, the bacterium shoots off in a new direction. This is chemotaxis. The cell isn't just swimming randomly, it's navigating. It can sense chemical gradients, concentrations of nutrients or toxins in the water, and it moves toward what it needs and away from what will kill it. Without a brain, without eyes, without any centralized control system, the bacterium is processing information, making decisions, and directing its motion using nothing but molecular machinery. This motor, the bacterial flagellum, is one of the oldest continuously running machines on Earth. It evolved more than 3 billion years ago, back when Earth's atmosphere had almost no oxygen, and the only life on the planet was microbial. And it's still here, still spinning, in bacteria in your gut, in the soil, in the ocean, in the clouds, billions of trillions of these motors running simultaneously, all descended from that first molecular engine that learned to spin. But bacteria aren't the only ones who had to solve this problem. There's another kind of cell, larger, more complex, that face the same challenge. These are protists, single-celled organisms that are neither bacteria nor animals nor plants. They're eukaryotes, which means they have a nucleus and compartments inside their cells. And many of them move, but they don't use rotating flagella. They use something else, something that looks like hair. Cilia. If you look at a paramecium under a microscope, its entire surface is covered in thousands of tiny projections, all beating in coordinated waves. Each cilium is about 10 micrometers long, much shorter than a bacterial flagellum, and instead of spinning, it bends. It sweeps forward in a fast power stroke, then recovers backward in a slower return stroke. And because the motion is asymmetric, the forward stroke is fast and stiff, the return stroke is slow and flexible. It generates thrust. But here's what's remarkable. A single cilium doesn't do much. The power stroke pushes water one way, the return stroke pulls it back. At low Reynolds number, that might seem like it would cancel out, but the timing matters. The asymmetry in speed and shape means that slightly more water is pushed forward than pulled back. The net effect is small, but it's not zero. And a paramecium doesn't have just one cilium, it has about 3,000 of them, and they all beat together in perfect coordination, creating waves that ripple across the cell surface from front to back. It's called metachronal rhythm, like wind moving through a field of wheat, or fans doing the wave in a stadium. Each cilium is slightly out of phase with its neighbours, so the waves flow smoothly across the surface, generating continuous thrust. The paramecium doesn't have a brain coordinating this. It doesn't have nerves sending signals to each cilium, telling it when to beat. The coordination is mechanical. Each cilium is connected to its neighbours through the cell's internal structure, through a network of proteins and microtubules beneath the surface. When one cilium bends, it creates a slight mechanical tension that pulls on the next cilium, triggering its power stroke a fraction of a second later. The wave propagates through mechanical coupling, not neural commands. It's self-organized, emergent. The cell doesn't control the rhythm. The rhythm arises from the structure itself. And this isn't unique to paramecium. Cilia are everywhere. In your lungs, cilia beat constantly, sweeping mucus and trapped particles up and out of your airways. In your brain, cilia line the ventricles, moving cerebrospinal fluid. In the fallopian tubes, cilia move the egg toward the uterus. In your nose, in your throat, in your reproductive system, cilia are beating, moving fluids, clearing debris, transporting cells. Every cilium in your body is a descendant of the first cilia that evolved in single-celled organisms over 1 billion years ago. The structure is almost identical. At the core of every cilium is a scaffold called the axoneme, 9 pairs of microtubules arranged in a circle around a central pair. This 9+2 structure is universal. It's in paramecium, in algae, in the cells lining your lungs. And attached to those microtubules are motor proteins called dynein. Dynein is the engine. It's a molecular motor that walks along the microtubules, pulling them past each other. When dynein on one side of the cilium pulls, the microtubules slide, the cilium bends in that direction. When dynein on the other side pulls, the cilium bends back. The coordinated action of thousands of dynein motors, all pulling in sequence, creates the cilium's beat. And dynein, like the bacterial flagella motor, is powered by chemistry, specifically by a molecule called ATP, adenosine triphosphate. ATP is the energy currency of all cells. When a molecule of ATP is broken down into ADP and a phosphate group, energy is released. Dynein captures that energy and uses it to change shape, to take a step to pull on the microtubules. Each step is tiny, about 8 nanometers. But dynein takes millions of steps, and thousands of dynein motors work together, and the result is a cilium that beats 10 to 40 times per second for hours, for days, for your entire life. This is the machinery of motion. Not muscles contracting, not neurons firing, just proteins, folded into shapes that can harvest energy from broken chemical bonds and convert it into mechanical work. These motors are so small you need an electron microscope to see them. But they're powerful enough to move cells through water, to clear mucus from your lungs, to circulate fluid through your brain. And they're ancient. Bacterial flagella evolved over 3 billion years ago. Eukaryotic cilia and flagella evolved at least 1.5 billion years ago, maybe earlier. For most of Earth's history, these molecular motors were the only way anything moved. Long before fins or legs or wings, long before muscles or bones or nervous systems, life was moving, navigating, hunting, escaping, using nothing but spinning and beating at the molecular scale. So tonight we're going deeper. We'll explore how these motors work, how they evolved, how they coordinate without commands. We'll see what happens when you try to move through a universe where viscosity dominates and inertia disappears. We'll trace the lineage from the first bacterial motor to the cilia in your lungs, revealing a continuity of motion that spans billions of years. Because motion didn't start with muscles, it started with molecules that learned to spin and beat. And every moving thing alive today carries that legacy inside. Let's begin where motion begins, with forces so small they're measured in piconewtons, and structures so precise they're built one atom at a time. Maybe motion was always going to be a problem. If you're alive, you need to find things. Food, light, safety, mates. And if you're rooted in place, you're at the mercy of whatever drifts past. You can't hunt, you can't escape. You're waiting for the world to come to you. And sometimes it doesn't. So life needed to move. But at the smallest scales, movement isn't intuitive. It's not like walking or swimming in our world, where you push off the ground or stroke through water and momentum carries you forward. At microscopic scales, the physics are fundamentally different. The rules change, and life had to figure them out from scratch. Let's start with the problem, the Reynolds number. It's a dimensionless number that compares inertial forces to viscous forces. When Reynolds number is high, like when you swim in a pool, or a bird flies through air, or a fish moves through water, Inertia dominates. You can glide, you can coast. Your momentum carries you even after you stop exerting force. But when Reynolds' number is low, viscosity dominates, and below a Reynolds number of about 1, the physics invert completely. A bacterium swimming through water operates at a Reynolds number around 0.1. That's 5 orders of magnitude below the threshold where inertia matters. For the bacterium, water isn't a fluid you can push against and glide through. It's a viscous medium that resists every movement and stops you the instant you stop pushing. According to research by Howard Berg at Harvard University in the 1970s and 80s, if a bacterium stops spinning its flagellum, it comes to a complete halt within a microsecond. Not a gradual slowdown, an instant stop. There's no drifting, no momentum. Motion and force are directly coupled. You move only as long as you push. Purcell explained this with a thought experiment in his 1977 lecture, Life at Low Reynolds Number. Imagine you're a bacterium, you have a little paddle, and you decide to row. You push the paddle forward through the water, then pull it back. At high Reynolds number, that works. The forward stroke moves you forward more than the backward stroke moves you back because of inertia and the way you can orient the paddle. But at low Reynolds number, there's no inertia to help you. The water is so viscous that every movement is reversible. If you push forward and pull back in the same path, you end up exactly where you started. Zero net displacement. Purcell called this the scallop theorem. A scallop opens its shell slowly, then snaps it shut quickly, and the asymmetry in speed creates thrust at high Reynolds number. But at low Reynolds number, speed doesn't matter. Only geometry matters. If your motion is reciprocal, if it looks the same played forward or backward in time, you go nowhere. So how do you move when symmetry doesn't work and momentum doesn't exist? You need asymmetry, not in speed, but in shape, in the path your motion traces through space. The bacterial flagellum solves this with rotation. The flagellum is a helical filament shaped like a corkscrew, and it spins. As it rotates, it traces a 3-dimensional spiral path through the water. That spiral is asymmetric. Every point on the helix follows a different trajectory, and that asymmetry generates thrust. The bacterium is pushed forward, slowly, steadily, as long as the motor keeps spinning. But here's the elegant part. The motor itself is reversible. It can spin clockwise or counterclockwise. When it spins counterclockwise, the flagellum bundles together with other flagella on the cell, and they form a coordinated propeller that pushes the cell forward in a smooth run. But when the motor switches to clockwise, the bundle flies apart. Each flagellum goes its own way. The cell tumbles, rotating randomly in space. Then the motor switches back to counterclockwise. The flagella rebundle, and the cell shoots off in a new direction. This run and tumble behavior is how bacteria navigate. They can't sense gradients directly. They're too small to detect a difference in concentration between their front end and their back end. Instead, they measure concentration over time. As they swim, they're constantly sampling the chemical environment. If the concentration of a nutrient is increasing, if things are getting better, they run for longer before tumbling. If the concentration is decreasing, if they're swimming the wrong way, they tumble more frequently, reorienting randomly until they happen to swim in a better direction. It's a biased random walk. And it works. Studies by Berg and others in the 1980s show that E. coli bacteria can climb chemical gradients with remarkable precision, finding food sources in complex environments using nothing but this motor and a handful of sensory proteins embedded in their membrane. The motor itself is a marvel of molecular engineering. At its core is a rotor, a set of proteins called FLIG that form a ring in the cell membrane. Surrounding the rotor is a stator, proteins called MOTA and MOTB that anchor to the cell wall and don't rotate. The stator proteins are proton channels. Protons, hydrogen ions, flow through them from outside the cell to inside, driven by a concentration gradient, and as each proton passes through, it pushes on the rotor, causing it to turn slightly. It's like a water wheel powered by a stream of ions. Thousands of protons flow through every second, and each one contributes a tiny push. The cumulative effect is continuous rotation at about 100,000 RPM. The efficiency is staggering. According to research by Yoshiyuki Soa and colleagues published in Nature in 2005, The bacterial flagella motor converts nearly 100% of the proton motive force into mechanical work. There's almost no energy lost as heat. For comparison, human-engineered electric motors typically operate at 70 to 90% efficiency. The bacterial motor is more efficient than anything we've built, and it self-assembles. No one puts it together. The proteins are synthesized in the cell, they diffuse to the membrane, and they find each other. They lock into place based on their shapes, complementary surfaces that fit together like puzzle pieces. The rotor forms first, then the stator proteins arrive and anchor around it. The hook and filament grow afterward, protein by protein, threaded through a hollow channel in the center of the structure. It takes about 20 minutes for a complete flagellum to assemble, and once it's built, it starts spinning. There's no instruction manual, no blueprint external to the system. The information for how to build the motor is encoded in the shapes of the proteins themselves, molecular self-assembly, and it works every time in billions of bacteria across thousands of species. But bacteria aren't the only ones who mastered low Reynolds number motion. Eukaryotic cells, larger, more complex cells with nuclei and internal compartments, face the same problem, and they came up with a different solution. Cilia. Where bacterial flagella spin, cilia beat. They bend back and forth in an asymmetric stroke, a fast, straight power stroke in one direction and a slow, curved recovery stroke in the other. The asymmetry means they push more water forward than they pull back, generating net thrust. But a single cilium doesn't generate much force. The real power comes from having thousands of them, all beating together in coordinated waves. A paramecium has about 3,000 cilia covering its surface. Each one is about 10 micrometers long, and they're arranged in neat rows like the bristles on a brush. And they don't all beat at the same time. They beat in sequence, one after another, creating a wave that travels across the cell surface from front to back. It looks like wind rippling through a field of grain. Scientists call this metachronal rhythm, meta meaning after, and chronal meaning time. Each cilium beats just slightly after the one in front of it, and the phase delay creates the wave. But how do they coordinate? The paramecium doesn't have a brain. It doesn't have neurons sending signals to each cilium orchestrating the beat. The coordination is mechanical, built into the structure of the cell itself. At the base of every cilium is a basal body, a cylindrical structure made of microtubules that anchors the cilium to the cell surface. And beneath the surface, the basal bodies are connected to each other by a network of protein fibers called striated rootlets. These rootlets create mechanical tension. When one cilium beats, it pulls on its basal body, which pulls on the rootlets, which tug on the neighbouring basal body, which triggers the next cilium to beat. The wave propagates through direct mechanical coupling. No commands, no signals, just structure and physics. The cell doesn't tell the cilia when to beat. The cilia tell each other. And the beating is powered by dynein. Inside every cilium is a scaffold called the axoneme. 9 pairs of microtubules arranged in a circle with 2 single microtubules in the centre. This 9+2 structure is ancient and universal. It's the same in algae, in paramecia, in the cells lining your lungs. Attached to the outer microtubule pairs are motor proteins, dynein arms, that reach out and grab the neighbouring microtubule pair. When dynein binds to its target and breaks down ATP, it pulls, sliding the microtubules past each other. But the microtubules are anchored at the base, so instead of sliding freely, they bend. The cilium bows to one side, then dynein on the opposite side pulls, and the cilium bows back. The result is a whip-like beat. Research by Jonathan Howard at Yale University and others in the 1990s showed that the dynein motors are mechanically regulated. When the cilium bends too far in one direction, the microtubules on that side are stretched, which deactivates the dynein motors. Meanwhile, the microtubules on the opposite side are compressed, which activates their dynein. The system has built-in feedback. The motors know when to stop pulling and when to start pulling again, based purely on the mechanical strain in the structure. There's no external control. The cilium regulates itself, and this isn't a new invention. Cilia evolved at least 1.5 billion years ago, possibly earlier. The last common ancestor of all eukaryotes, the cell that gave rise to animals, plants, fungi and protists, almost certainly had cilia. We know this because cilia are everywhere. Unicellular algae use them to swim. Coral larvae use them to drift through the ocean. Your cells use them to move fluids and sense the environment. The structure is so conserved, so fundamental, that losing it is usually lethal. According to research published by David Mitchell at SUNY Upstate Medical University in the 2000s, mutations that disrupt the 9+2 axoneme structure cause severe diseases in humans: primary ciliary dyskinesia, polycystic kidney disease, retinal degeneration. Cilia aren't optional, they're essential, and they've been essential for over 1 billion years. So here's what we're seeing. 2 different lineages, bacteria and eukaryotes, both facing the same problem. How to move at low Reynolds number, where the rules of motion are alien and intuition from our world doesn't apply. And both lineages solved it with molecular motors. One lineage built rotary engines, the other built beating arrays. But both solutions rely on the same fundamental principles: asymmetry, continuous energy input, and mechanical coordination without central control. These motors are the foundation. Everything that moves, everything that swims, crawls, flows, or flies, descends from cells that first learned to move with engines built from folded proteins. And those engines are still running, still spinning and beating inside you right now. Let's go deeper into the machinery itself, the motors. Because when we talk about molecular motors, we're not speaking metaphorically. These aren't just proteins that happen to move. They're engines in the most literal sense, structures that convert chemical energy into mechanical work, with rotors, levers, power strokes and recovery phases. They have moving parts, they generate force, they do work against resistance, and they operate with a precision that would be impossible to achieve with anything built by human hands at that scale. The motor protein that powers cilia, dynein, is enormous by molecular standards. It's made of multiple subunits, with a combined molecular weight of about 1.5 million daltons. For context, a typical protein might be 50,000 daltons. Dynein is as giant, and its structure reflects its function. At one end of the dynein molecule is a ring of 6 protein domains arranged in a hexagon. This ring binds and breaks down ATP, the fuel molecule. At the other end is a stalk that reaches out and grabs onto a microtubule, the track it walks along. And connecting them is a linker domain that changes shape when ATP is broken down, converting chemical energy into a mechanical swing. Here's how it works. Dynein starts attached to one microtubule. Its stalk reaches out and binds to a second microtubule nearby. At this point, dynein is locked in place, holding the 2 microtubules together. Then an ATP molecule arrives. It binds to the ring. Dynein breaks the ATP into ADP and a phosphate group, and the energy released causes the linker domain to swing. The motion is like a lever pulling. The linker bends, dragging one microtubule relative to the other. The step size is about 8 nanometers. That's roughly the distance between tubulin subunits, the building blocks of microtubules. One step, one building block. Then dynein releases, resets, and takes another step, and another, and another. At full speed, a single dynein motor can take about 8 steps per second. That doesn't sound fast. But remember, there are hundreds of dynein motors attached along the length of a single cilium. They all step together, and the cumulative effect is that the microtubules slide past each other at speeds of several micrometers per second. For a structure only 10 micrometers long, that's fast. The cilium bends, sweeps forward, recovers and beats again, 10 to 40 times per second, depending on the cell type. Research by Takahide Kon and colleagues at Osaka University in 2012 used cryo-electron microscopy to visualize dynein in different states, bound, unbound, with and without ATP, and revealed the structural changes in atomic detail. The linker swings like a hinge. The stalk binds and releases in a coordinated cycle. Every part of the motor is optimized for its task. There's no wasted motion, no unnecessary complexity, just a machine that does one thing: convert ATP into sliding motion, and does it with near-perfect reliability. But dynein isn't the only motor protein. There's another one closely related called kinesin. Kinesin walks along microtubules too, but it walks in the opposite direction. Dynein generally moves toward the minus end of microtubules, toward the base of the cilium. Kinesin moves toward the plus end, toward the tip, and kinesin is faster. It can take about 100 steps per second, covering a micrometer in about 12 milliseconds. That's roughly the speed of a tortoise, if you scale it up to human size. Not fast by our standards, but at the molecular scale, kinesin is sprinting. And kinesin doesn't just walk, it carries cargo. Inside your cells, kinesin motors are constantly transporting vesicles, organelles, and protein complexes from one part of the cell to another. They're like delivery trucks on a highway system made of microtubules. The cell manufactures something in the nucleus or the endoplasmic reticulum, packages it in a membrane-bound vesicle, attaches a kinesin motor to it, and the motor walks it down a microtubule to wherever it needs to go. Without kinesin, cells couldn't move materials over long distances. Neurons, for example, are enormous. Some human neurons extend over a meter in length. Getting proteins from the cell body to the tip of the axon would take weeks by diffusion alone. Kinesin makes the journey in hours. Kinesin structure is simpler than dynin's. It has two motor domains, two heads, that take turns stepping forward along the microtubule, like feet walking. One head binds to the microtubule, breaks down ATP, and swings forward. The motion pulls the second head forward. That head then binds, breaks down its own ATP, and swings, pulling the first head forward again. The two heads alternate, and the motor walks hand over hand, or head over head, down the microtubule track. The walking motion was directly visualized in the late 1990s by Ron Vale and colleagues at UC San Francisco, using fluorescence microscopy and single molecule tracking. They attached fluorescent tags to individual kinesin molecules and watched them walk along microtubules in real time. The steps were consistent, 8 nanometers per step, perfectly matching the spacing of tubulin subunits. Kinesin doesn't slip. It doesn't stumble. It places each foot exactly where the next binding site is waiting. The precision is absolute. And here's what's remarkable about both dynin and kinesin. They operate stochastically. At the molecular scale, thermal motion is huge. Molecules are constantly being jostled by random collisions with water molecules, Brownian motion. The forces involved are on the order of piconewtons, and the energies are comparable to the thermal energy at room temperature. That means molecular motors are constantly being knocked around, buffeted by noise. Yet they still walk in one direction. They still generate force. They still transport cargo reliably. How? The answer is that the motors are designed to bias the random motion. They don't fight against Brownian motion, they harness it. When dynin or kinesin breaks down ATP, the energy release doesn't just push the motor forward, it changes the motor's shape in a way that makes it more likely to bind to the next site on the microtubule rather than the previous one. The motor is essentially lowering the energy barrier for moving forward and raising it for moving backward. It's using chemical energy to create a directional bias in an otherwise random walk. This concept, Brownian ratchets, as they're sometimes called, was explored theoretically by physicists in the 1990s and 2000s. The motor doesn't need to overpower thermal noise, it just needs to tip the statistical odds in favour of forward motion. And because ATP hydrolysis is irreversible, you can't spontaneously reassemble ATP from ADP and phosphate under cellular conditions. The motor's steps are irreversible too. The directionality is built into the thermodynamics, so every motor protein is, in a sense, extracting order from chaos, taking the random thermal jostling of molecules and converting it into directed, purposeful motion. Now scale this up. Imagine not one motor, but thousands, all working together. That's what you have in a single cilium. Hundreds of dynin motors, distributed along 9 pairs of microtubules, all pulling in coordinated bursts. The coordination doesn't come from external signals, it comes from mechanical feedback. When one set of dynin motors pulls and bends the cilium to one side, the microtubules on that side are stretched. The stretching changes the binding affinity of the dynin motors. They become less likely to stay attached. Meanwhile, the microtubules on the opposite side are compressed, which makes their dynin motors more likely to bind and pull. The result is a self-regulating oscillation. The cilium bends one way, then the other, over and over, driven entirely by the mechanical properties of the structure. This is self-organization. No central controller. No clock ticking in the background telling the motors when to fire. just a collection of molecular machines, each following simple rules based on local mechanical strain, and the global behaviour, a coordinated beat, emerges from their interactions. And it's not just cilia. The same principles apply to other motor-driven systems. Consider a sperm cell. A human sperm has a flagellum, not a bacterial-style rotating flagellum, but a eukaryotic one, powered by dynin, with the same 9+2 axoneme structure as cilia. But instead of beating like a cilium, the sperm flagellum undulates. It sends waves down its length, from base to tip, and those waves push against the fluid, propelling the sperm forward. The flagellum is about 50 micrometers long, 5 times longer than a typical cilium, and the wave propagates at a speed of about 20 micrometers per second, faster than the sperm itself swims. The wave travels down the flagellum, and as it goes, it generates thrust. The sperm moves forward at about 200 micrometers per second, about 4 body lengths per second. Not as fast as a bacterium relative to size, but fast enough. Sperm have to cover a distance of about 15 centimeters to travel from the vagina to the fallopian tube. At 200 micrometers per second, that takes about 12 minutes, assuming they swim in a straight line. In reality, it takes longer. Sperm don't swim straight. They spiral, they get stuck in mucus, they take wrong turns. But the motors keep running. The flagellum keeps beating, and eventually, if everything goes right, one sperm reaches the egg. The energy cost is enormous. Sperm are packed with mitochondria, cellular power plants that produce ATP. The mitochondria are wrapped around the base of the flagellum, forming a tight spiral called the midpiece. They produce ATP continuously, feeding it to the dynin motors, which burn through it at an astonishing rate. A single sperm uses about 10 to the 9 molecules of ATP per second. That's a billion ATP molecules every second just to keep the flagellum beating, and the sperm only has enough energy stored to swim for a few hours at most. It's a sprinter, not a marathoner. Use it or lose it, but it works. Sperm have been using this design for hundreds of millions of years. Every vertebrate, every insect, every organism with motile gametes uses some version of this system. Dynin-powered flagella, undulating or beating, all descended from the same ancient motor proteins that evolved over 1 billion years ago. And it's not just animals. Plants have motor proteins too, though they don't use them for swimming. Most plant cells are immobile, but inside plant cells, organelles are constantly being moved around by myosin motors walking along actin filaments. Chloroplasts rearrange themselves to maximize light capture. The cytoplasm streams in circular currents, distributing nutrients, and during cell division, motor proteins pull chromosomes apart, ensuring each daughter cell gets a complete set of genes. Myosin is another ancient motor protein, related to dynein and kinesin, but specialized for a different track, actin filaments instead of microtubules. Myosin is also the motor that powers muscle contraction in animals, but long before muscles existed, myosin was moving things inside single cells. The motor came first, the muscle came later. In fact, all 3 major families of motor proteins, dynein, kinesin, and myosin, appear to share a common evolutionary ancestor. They have similar structural motifs, particularly in the ATP binding domain. The details differ, but the core mechanism is the same. Bind ATP, break it down, use the energy to change shape, generate force, reset, repeat. It's a solution that evolution discovered early and refined over billions of years. And here's the stunning part. These motors are reversible in principle, but unidirectional in practice. If you could somehow push a dynein motor backward and force it to synthesize ATP from ADP and phosphate, the motor would run in reverse. But under normal cellular conditions, that doesn't happen. The concentration of ATP is high, the concentration of ADP and phosphate is lower, and the thermodynamic gradient drives the reaction in one direction. The motor walks forward, the cilium beats, the cell moves. This is life at the molecular scale. Chemistry becomes motion, energy gradients become direction, and billions of years of evolution have shaped these motors into machines so efficient, so precise, and so reliable that we're still trying to figure out how to replicate them with human engineering. But we're not done yet, because understanding how a single motor works doesn't explain how thousands of motors coordinate across an entire cell. or how cells with different motor architectures navigate toward food, away from danger, toward light, or toward each other. That's where the story gets even stranger. Because motion without a brain isn't just about motors, it's about sensing, responding, and making decisions with nothing but chemistry and structure. Moving is one thing, moving towards something is another. If you're a single cell in a vast ocean spinning or beating your way through water, you're not just trying to move, you're trying to go somewhere useful, toward food, toward light, away from toxins, toward other cells of your kind. But you don't have eyes, you don't have a nose, you don't have a brain to process sensory information and plan a route. You have molecules embedded in your membrane, and you have motors that spin or beat. Somehow that has to be enough. And it is. Bacteria navigate chemical gradients with a precision that rivals anything more complex organisms can do. The process is called chemotaxis, and it's one of the most elegant examples of molecular computation in biology. Here's the setup. A bacterium swimming through water is surrounded by chemicals, nutrients like sugars and amino acids, toxins like acids or heavy metals. These chemicals diffuse through the water, creating gradients, regions where the concentration is higher or lower. If the bacterium could measure the concentration at two different points in space and compare them, it could figure out which direction the gradient is pointing and swim that way. But the bacterium is too small. It's only about 2 micrometers long. The difference in concentration between its front end and its back end is negligible, lost in the noise of random molecular fluctuations. It can't sense a spatial gradient directly, so instead, it measures concentration over time. As the bacterium swims, receptor proteins on its surface are constantly binding to molecules in the surrounding water. These receptors, called chemoreceptors, change shape when they bind to their target molecule. That shape change triggers a cascade of chemical reactions inside the cell, altering the behavior of the flagellar motor. If the concentration is increasing, if the bacterium is swimming up the gradient toward higher concentrations of food, the motor keeps spinning counterclockwise, and the cell continues running straight. But if the concentration is decreasing, if the bacterium is swimming down the gradient away from food, the motor switches to clockwise, the flagella fly apart, and the cell tumbles, reorienting randomly in space. Then the motor switches back and the bacterium swims off in a new direction. The result is a biased random walk. When swimming in the right direction, runs are long. When swimming in the wrong direction, tumbles are frequent. Over time, the bacterium drifts up the gradient, not because it knows where it's going, but because the statistics favour it. But here's the clever part. The chemoreceptors adapt. If the bacterium swims into a region of higher concentration and stays there, the receptors gradually become less sensitive. They reset their baseline. That way, the bacterium doesn't just respond to absolute concentration, it responds to change. It's measuring the time derivative of concentration. It's asking, am I getting closer to food or farther away? Not, how much food is here right now? This adaptation happens through a process called methylation. There are enzymes inside the cell that add or remove methyl groups from the chemoreceptors, changing their sensitivity. When the receptor binds to a nutrient, methylation increases, making the receptor less responsive. When the nutrient unbinds, demethylation decreases, making it more responsive again. The system finds a balance, a steady state where the receptor is tuned to detect changes rather than absolute levels. Research by Howard Berg and Dennis Bray in the 1980s and 90s showed that E. coli bacteria can detect changes in chemical concentration as small as 0.1% over distances of a few micrometers. That's astounding sensitivity. They're measuring tiny differences in the arrival rate of molecules, filtering out noise, and using that information to control a motor. And it all happens with proteins. No neurons, no synapses, just receptors, kinases, and flagellar motors interacting through chemical signals. And bacteria don't just sense chemicals. Some sense magnetic fields. Magnetotactic bacteria, discovered by Richard Blakemore in the 1970s, contain chains of magnetite crystals inside their cells. These crystals are magnetic. They align with Earth's magnetic field, and the entire chain acts like a compass needle, orienting the bacterium north or south. In the northern hemisphere, magnetotactic bacteria swim north and downward, following the magnetic field lines toward the sediment at the bottom of ponds and lakes where oxygen is scarce, which is where they thrive. In the southern hemisphere, they swim south and downward. Same strategy, different hemisphere. The magnetite crystals are called magnetosomes, and they're synthesized by the bacteria, grown inside membrane-bound compartments with exquisite control over crystal size and shape. Each magnetosome is about 50 nanometers across, small enough to be a single magnetic domain, meaning the entire crystal acts as one tiny magnet. String a dozen of them together in a line, and you have a biological compass sensitive enough to detect Earth's weak magnetic field, which is only about 50 microteslas. But magnetotaxis isn't just about following a compass, it's about efficiency. Magnetotactic bacteria still use chemotaxis. They still sense oxygen gradients. But instead of swimming in 3 dimensions, randomly exploring all directions, they're constrained to swim along one axis, north-south. That reduces the search space. They're essentially navigating in 1 dimension instead of 3, which makes finding the optimal oxygen concentration much faster. The magnetic field isn't the destination. It's a guide rail keeping them on course. Other organisms sense light. Phototaxis, movement toward or away from light, is widespread among single-celled organisms. Algae do it, protists do it, even some bacteria do it. And the reason is simple. If you photosynthesize, light is food. You need to position yourself where the light is strong enough to power your metabolism, but not so strong that it damages your cells. You need to find the sweet spot. But sensing light at the single-cell level is tricky. You don't have a lens, you don't have a retina, you don't have image-forming optics. You need something simpler. Many algae solve this with an eye spot, a patch of pigmented proteins near one side of the cell. The pigment absorbs light, creating a shadow on the opposite side. And near the base of the flagellum there's a light-sensitive protein, a photoreceptor, that detects the intensity of light hitting the cell. As the cell rotates while swimming, the eye spot periodically blocks the light from reaching the photoreceptor. The photoreceptor experiences a flicker, bright, dim, and the frequency of the flicker depends on the cell's orientation relative to the light source. If the cell is swimming directly toward the light, the eye spot never blocks it. The photoreceptor sees constant illumination, and the flagellum beats normally. But if the cell is swimming at an angle, the eye spot passes in front of the photoreceptor once per rotation, and the flicker tells the cell it's off course. The photoreceptor triggers a response, usually a change in the beating pattern of the flagellum, that causes the cell to turn, reorienting toward the light. This system was worked out in detail in the green alga Chlamydomonas by researchers in the 1990s and 2000s. Chlamydomonas has 2 flagella, and they beat slightly differently. One is primarily responsible for forward propulsion, the other for steering. When the eye spot detects that the cell is veering away from the light source, the steering flagellum changes its beat, causing the cell to turn back. It's a feedback loop, continuously active, constantly correcting. The cell spirals toward the light, tracing a helical path through the water, homing in on the brightest region. And the photoreceptor itself, channel rhodopsin, is a light-gated ion channel. When light hits it, it opens, allowing ions to flow into the cell. That ion current changes the membrane voltage, which affects the calcium concentration near the flagella, which modulates the dynein motors. Light becomes electrical signal, becomes chemical signal, becomes mechanical motion, all in milliseconds. Channel rhodopsin, by the way, is the same protein that neuroscientists now use in optogenetics, using light to control neurons in the brains of mice and other animals. The technique was borrowed directly from algae. Evolution invented it first, for navigation. We adapted it for research. But not all sensing is about following gradients. Sometimes it's about responding to touch. Mechanotransduction, the conversion of mechanical force into biochemical signals, is fundamental to how cells interact with their environment. When a cilium bends because it hits an obstacle, or when a cell is compressed by neighbouring cells, or when fluid flow creates shear stress on a membrane, the cell needs to know. And it does, through mechanosensitive proteins embedded in its membrane. These proteins are essentially molecular springs. When the membrane is stretched or compressed, the protein changes shape, opening a channel that allows ions to flow through. The ion flux changes the cell's internal chemistry, triggering responses. Maybe the cell stops moving, maybe it changes direction, maybe it reinforces its membrane, maybe it divides. One of the best studied mechanosensors is the MSCL channel in bacteria, mechanosensitive channel of large conductance. It's a pore in the membrane that opens when the membrane is stretched. If a bacterium suddenly finds itself in a low-salt environment, say, fresh water, after living in seawater, water rushes into the cell by osmosis, and the membrane swells. Without a release valve, the cell would burst. But MSCL opens, releasing ions and small molecules, reducing the internal pressure and preventing lysis. It's an emergency relief valve, activated purely by mechanical tension. In eukaryotic cells, mechanotransduction is even more sophisticated. The cilia on your cells aren't just for moving fluids, they're also sensors. When fluid flows over a cilium, the cilium bends, and that bending is detected by proteins at the base. The cell receives information about flow rate, flow direction, and shear stress. This is how kidney cells sense urine flow, how cells in blood vessels sense blood flow, how cells in your inner ear detect sound vibrations and head movements, giving you the sense of balance. And sometimes mechanotransduction coordinates collective behavior. Take Volvox, a multicellular green alga that forms a hollow sphere about 0.5 millimeter across. Each cell in the sphere has 2 flagella, and they all beat in coordination, spinning the sphere as it swims through the water. But the cells on one side of the sphere beat slightly faster than the cells on the other side, creating a front and a back, an axis of polarity. The faster beating cells are pulled more taut by the surrounding extracellular matrix, and that mechanical strain feeds back into their beating pattern, reinforcing the asymmetry. The colony knows which way is forward, not because there's a command center, but because the geometry and mechanics of the structure create a stable pattern. Mechanical feedback, chemical signals, electrical gradients, all of this is happening simultaneously in every swimming cell, coordinating movement without a brain. And then there's predation, because not all movement is about finding nutrients in the water. Sometimes it's about finding other cells and eating them. Didinium is a ciliate, a relative of paramecium that hunts other protists. It's barrel-shaped, covered in cilia, and it swims through the water scanning for prey. Its preferred target is paramecium. When a didinium encounters a paramecium, it doesn't just bump into it passively. It fires trichocysts, harpoon-like structures that shoot out from the cell surface, penetrating the paramecium's membrane and anchoring the 2 cells together. Then didinium opens its oral groove, a specialized region that can expand enormously, and begins to engulf the paramecium. The entire prey cell, often as large as the predator itself, is pulled inside, where it will be digested over the next several hours. The hunting behavior is complex. Didinium doesn't swim randomly. According to research by Akihiro Ishijima and others in the early 2000s, didinium responds to chemical cues released by paramecium, detection at a distance, possibly through chemoreceptors similar to those in bacteria. When it detects prey, its swimming pattern changes. It spirals more tightly, staying in the region where the chemical signal is strongest. When contact is made, the trichocysts fire automatically, a mechanical trigger, like stepping on a landmine. This is predation at the cellular level, hunting strategies, weapons, digestion, all orchestrated by molecular motors, sensory proteins, and self-organizing membranes. No nervous system, no muscles. Just a single cell with thousands of cilia beating, trichocysts loaded, and a hunger that drives it through the microscopic world in search of prey. And paramecium, for its part, has defenses. It can swim away, fast. It can reverse the beating direction of its cilia, swimming backward to escape. Some species even have trichocysts of their own, which they discharge when attacked, creating a cloud of tangled proteins that confuses or repels predators. It's an arms race, playing out at the microscopic scale, driven by the same evolutionary pressures that shape predator-prey dynamics in the visible world. And all of it, every escape, every chase, every navigation toward light or food or safety comes down to motors, molecular machines, spinning or beating, coordinated by nothing more than chemistry and structure. No brain required, just motion, refined over billions of years into something that looks for all the world like intentionality. So where did all of this come from? Because molecular motors didn't just appear, fully formed, spinning at 100,000 RPM, or walking along microtubules with 8 nanometer precision. They evolved. They had ancestors. And somewhere, billions of years ago, there was a first motor, a protein that learned to convert chemical energy into directed motion, setting the foundation for every movement that would follow. Tracing that origin is difficult. Proteins don't fossilize. DNA degrades. The molecular record of early life is fragmentary at best, but we can make inferences. We can compare modern motor proteins across different organisms, look for shared structural features, and reconstruct what the ancestral motor might have looked like. And what we find is this. All the major motor proteins, dynein, kinesin, myosin, share a common core. They have similar ATP binding domains. They undergo similar conformational changes when ATP is hydrolyzed. They generate force through similar mechanical principles. The details differ. Dynein is huge and complex. Kinesin is smaller and simpler. Myosin is specialized for actin instead of microtubules, but the underlying mechanism is the same. This suggests a common evolutionary origin. Not that one motor evolved from another, but that all of them evolved from some ancestral ATPase, an enzyme that could bind and break down ATP long before it was harnessed for mechanical work. ATPases are everywhere in biology. They're enzymes that hydrolyze ATP, releasing energy that drives chemical reactions. Pumps that move ions across membranes are ATPases. Chaperones that fold proteins are ATPases. Helicases that unwind DNA are ATPases. The ability to bind ATP and extract energy from it is ancient, fundamental, and universal. At some point, one of these ATPases acquired the ability to change shape in a way that generated force against a substrate. Maybe it was bound to a filament, an early version of actin or tubulin, and when it broke down ATP, the conformational change pushed or pulled on the filament. That's not yet a motor in the full sense. It's just a protein that twitches when it consumes energy. But that twitch, repeated over and over, could move things, slightly, incrementally. And if that movement gave the cell even a tiny advantage, better access to nutrients, faster escape from toxins, natural selection would amplify it. Over millions of years, the protein would be refined. Mutations that made the conformational change more efficient, more directional, more forceful would be favoured. Proteins that could bind to the filament more tightly, release more predictably, reset more quickly, would outcompete those that couldn't. And eventually you'd have a motor, a protein specialised for converting ATP into motion, with a stepping cycle, a power stroke, and the ability to walk or slide along a track. This probably happened multiple times, independently, in different lineages. Myosin evolved to work with actin filaments. Dynein and kinesin evolved to work with microtubules. The bacterial flagellar motor evolved to spin in the membrane. The solutions are different because the problems were different. But they all share the same basic logic. Harness the energy from ATP hydrolysis, convert it into a shape change, use that shape change to generate force, and repeat. Now, here's where it gets interesting. Because there's one motor protein that isn't just moving things, it's making ATP. ATP synthase. It's the enzyme that produces most of the ATP in your cells, and it's also a rotary motor, arguably the most important motor in all of biology. ATP synthase sits in the membrane of mitochondria, and in bacteria it sits in the cell membrane. It has two main parts, a rotor embedded in the membrane, and a catalytic head that sticks out into the cytoplasm. The rotor spins, driven by a flow of protons across the membrane. As it spins, the catalytic head synthesizes ATP from ADP and phosphate. The rotation is coupled directly to chemical synthesis. Spin the motor one way, and you make ATP. Spin it the other way, and you break down ATP. And that's exactly what happens. In mitochondria, the proton gradient drives the motor forward, synthesizing ATP. But in some bacteria, when ATP is abundant and the proton gradient is weak, the motor runs in reverse. It consumes ATP and pumps protons, maintaining the gradient. ATP synthase is reversible. It's both a motor and a generator. The structure was worked out by John Walker and colleagues in the 1990s. earning Walker a Nobel Prize in 1997. The catalytic head has 3 active sites arranged in a triangle. As the rotor spins, it pushes on a central stalk that runs through the middle of the head, and that stalk forces the active sites to cycle through 3 different shapes: open, loose, and tight. In the open state, ADP and phosphate bind. In the loose state, they're held in position. In the tight state, they're squeezed together so hard that they spontaneously form ATP. Then the site opens again, releasing the ATP, and the cycle repeats. 3 active sites cycling out of phase, driven by one continuous rotation. It's mechanochemistry, mechanical force directly driving a chemical reaction, and the rotor spins fast. At full speed, ATP synthase rotates at about 150 rotations per second. That's 9,000 RPM, and every full rotation produces 3 molecules of ATP, 1 from each active site. So a single ATP synthase molecule can produce about 450 ATP per second. Your body has trillions of them, running continuously, generating the roughly 60 kilograms of ATP you synthesize and consume every day just to stay alive. But here's the kicker: the same structure exists in bacteria. ATP synthase is ancient. It predates mitochondria. It predates eukaryotes. It was probably present in the last universal common ancestor, the organism from which all life on Earth descends. That means ATP synthase is at least 3.5 billion years old, possibly older. And when mitochondria evolved, when an ancient bacterium was engulfed by an early eukaryotic cell and became a permanent resident, an endosymbiont, it brought its ATP synthase with it. That bacterial motor is still in your mitochondria, still spinning, still making the ATP that powers every motor protein in your body. So the motor that makes the fuel also works like the motors that consume the fuel. ATP synthase spins, dynein walks, kinesin walks, myosin pulls. The bacterial flagellum spins. They're all variations on the same theme: proteins that convert energy gradients into motion. And this raises a profound evolutionary point. Once life figured out how to build molecular motors, it could do almost anything. Motors enabled motility. Cells could swim, hunt, escape. Motors enabled transport. Cells could grow larger because they could move materials internally over long distances. Motors enabled cell division. Motor proteins pull chromosomes apart during mitosis, ensuring each daughter cell gets a complete genome. Motors enabled multicellularity. When cells started forming colonies, they needed to coordinate, moving in concert, sharing resources, differentiating into specialized types. Motors made that possible. Consider the transition from single cells to multicellular organisms. It's one of the major evolutionary transitions in the history of life, and it happened multiple times, in animals, plants, fungi, algae. But every time it happened, motor proteins were involved. Take Volvox again. It's a colony of several thousand cells, each with 2 flagella, all beating together to spin the sphere through the water. But the cells aren't just identical copies. There's differentiation. Some cells are somatic cells. They swim and photosynthesize but don't reproduce. Others are germ cells. They divide and form the next generation. The colony has polarity, a front and a back. It has coordination. Cells on one side beat faster than cells on the other. And it has development. Germ cells undergo a complex series of divisions and rearrangements to form daughter colonies inside the parent, which are then released to live independently. All of this is orchestrated by motors, the flagella beating, the cytoplasmic bridges connecting cells, the contractile mechanisms that invert the developing daughter colonies during embryogenesis. None of it would work without molecular motors, coordinating action across thousands of cells. And the same is true in animals. The cilia in your lungs sweeping mucus upward, the flagellum on a sperm cell swimming toward an egg, the contractions of your heart driven by myosin pulling on actin filaments in cardiac muscle, the neurons in your brain transporting neurotransmitter vesicles down axons using kinesin. All of it traces back to the same molecular machinery that evolved billions of years ago in single cells. But scaling up motor coordination wasn't trivial. A single cell with a few thousand, Cilia can coordinate through mechanical coupling, each cilium pulling on its neighbours, waves propagating through direct physical connection. But a multicellular organism with trillions of cells, spread across centimetres or metres of space, can't rely on mechanical coupling alone. You need other systems: chemical signalling, electrical signalling, nervous systems, hormones. These are the mechanisms that let multicellular organisms coordinate motor activity on a larger scale. But even then, the motors themselves haven't changed much. The dynin in your lung cilia is structurally almost identical to the dynin in a single-celled paramecium. The myosin in your muscles is closely related to the myosin in amoeba, which use it to crawl across surfaces by extending pseudopods. Evolution didn't reinvent motors when organisms got bigger, it reused them. It placed them in new contexts, under new control systems, in new combinations. But the motors themselves, the molecular machines doing the work, are ancient, conserved, and universal. And this conservation tells us something profound. Once evolution solves a problem well, it doesn't discard the solution. It builds on it, it adapts it, it scales it. But the core machinery remains. You can see this in the structure of cilia and flagella. The 9+2 axoneme, 9 pairs of microtubules in a circle, 2 in the centre, dynin arms reaching between them, is the same in algae, in protists, in the cells of your lungs, in sperm, in the sensory cells of your inner ear. It's been conserved for over 1 billion years. Mutations that disrupt it are almost always lethal or severely debilitating. You can't mess with a 9+2 structure without breaking motility entirely, but you can modify it. Some organisms have 9+SD cilia, no central pair. These are usually sensory cilia, not motile ones. They don't beat, they just sit there, detecting chemical or mechanical signals. The motor proteins are gone, but the scaffold remains, repurposed for a different function. Other organisms have more elaborate structures. Some ciliates have compound ciliary organelles, cirri, where multiple cilia fuse together into a leg-like structure that the cell uses to walk along surfaces. The individual cilia are still there, still beating, but they're coordinated into a higher order unit. It's like fingers forming a fist. The basic components are the same, the arrangement is new, and in animals cilia diversified even further: motile cilia for moving fluids, primary cilia for sensing, nodal cilia for establishing left-right symmetry during embryonic development. These are cilia in a structure called the node, and they rotate in a specific direction, creating a leftward flow of fluid that tells the embryo which side is which. If the nodal cilia don't beat, or if they beat in the wrong direction, you get situs inversus, organs on the wrong side of the body, your heart on the right, your liver on the left. All because molecular motors didn't spin the way they were supposed to during development. That's how fundamental these motors are. They don't just move cells, they pattern bodies, they establish axes, they determine left from right, and they do it with the same basic machinery. Proteins, ATP, microtubules, that bacteria were using 3 billion years ago to swim toward food. So when we ask, where did molecular motors come from? The answer is, they came from chemistry that learned to move, from ATPases that twitched, from twitches that became steps, from steps that became coordinated walks, spins, beats, and from those first motors, everything else followed: motility, transport, division, coordination, multicellularity, sensation, development. Every major innovation in the history of life has been, at some level, an innovation in how to use motors, and those motors are still running, in you, in every organism, in every moving cell, an unbroken chain of motion stretching back billions of years, powered by the same molecules that were spinning and beating in the earliest oceans. Now let's bring this home, literally, to your body. Because everything we've been talking about, molecular motors, ciliary beating, coordinated waves, mechanotransduction, isn't just happening in pond water or ancient oceans, it's happening inside you, right now, in your lungs, your brain, your ears, your nose, your reproductive system. The same motors that evolved billions of years ago are still running, still beating, still keeping you alive. Start with your airways. Every breath you take pulls in air and with it dust, pollen, bacteria, fungal spores, smoke particles if you're near a fire, pollution if you're in a city, viral particles if someone nearby is sick. Your lungs are constantly being bombarded with foreign material, And if that material isn't cleared, it accumulates, bacteria multiply, infections set in, your lungs fill with mucus and debris, you can't breathe, but that doesn't happen, not usually, because lining your airways, from your trachea down through your bronchi and bronchioles, are millions of ciliated cells. Each cell has about 200 cilia on its surface, and those cilia are beating continuously, 10 to 15 times per second, 24 hours a day, for your entire life. They never stop. The cilia are submerged in a thin layer of mucus, a sticky gel produced by specialized goblet cells scattered among the ciliated cells. Particles that land in your airways get trapped in the mucus, and the cilia, beating in coordinated metachronal waves, push the mucus upward, against gravity, up from the depths of your lungs, through the bronchi, up the trachea into your throat. The mucus, carrying all the trapped debris, reaches the back of your throat and you swallow it, or you cough it out. Either way, it's gone, cleared from your lungs. This process is called mucociliary clearance, and it's one of the most important defense mechanisms your body has. Without it, every respiratory infection would be life-threatening, and it depends entirely on cilia, molecular motors, beating in rhythm, coordinated through mechanical coupling, powered by dynein and ATP. The coordination is remarkable. The cilia don't all beat in phase. If they did, they'd just churn the mucus in place. Instead, they beat in waves, each cilium slightly out of phase with its neighbours, creating a travelling wave that moves from the bottom of the lungs upward. It's the same metachronal rhythm we saw in paramecium, scaled up across millions of cells, spanning the entire respiratory tree. And the beat itself is asymmetric. During the power stroke, the cilium extends fully, reaching up into the mucus layer and sweeps forward, pushing the mucus along. During the recovery stroke, the cilium bends close to the cell surface, moving back through the watery layer below the mucus, where there's less resistance. The asymmetry ensures that more mucus is pushed forward than pulled back. Net flow, upward, continuous, always clearing. But when the cilia fail, everything falls apart. Primary ciliary dyskinesia, PCD, is a genetic disorder caused by mutations in the genes that encode ciliary proteins. Most commonly, the dynein motors are defective. The cilia are still there, structurally intact, but they can't beat properly. Some beat weakly, some beat erratically, out of sync, some don't beat at all. The result is devastating. Mucus accumulates in the lungs. Bacteria thrive in the stagnant mucus, causing chronic respiratory infections. Patients develop bronchiectasis, permanent damage to the airways. Lung function declines over time. Many patients need daily chest physiotherapy to manually clear the mucus, pounding on their chest to dislodge it, because their cilia can't do the job. And it's not just the lungs. People with PCD often have chronic sinus infections, because the cilia in the nose and sinuses are also paralysed. They have chronic ear infections, because the eustachian tubes, which drain fluid from the middle ear, are lined with cilia that can't clear the fluid. Male infertility is common, because sperm can't swim without functional flagella, and about half of PCD patients have situs inversus, organs on the wrong side of the body, because the nodal cilia that establish left-right asymmetry during embryonic development didn't beat properly. One defect in one type of motor protein, and the consequences cascade across multiple organ systems. That's how fundamental these motors are. They're not optional, they're not redundant, they're essential. And PCD isn't the only ciliopathy. There are dozens of genetic diseases linked to ciliary dysfunction. Polycystic kidney disease, cysts form in the kidneys because sensory cilia on kidney cells can't detect fluid flow properly, and the cells overproliferate in response. Retinitis pigmentosa. Photoreceptor cells in the retina degenerate because the connection between the inner and outer segments of the cell, which is a modified cilium, breaks down. Bardet-Biedl syndrome, obesity, cognitive impairment, polydactyly, kidney disease, all caused by defects in proteins that traffic through cilia. Cilia are everywhere, and when they break, everything breaks. But cilia aren't just for moving things, they're also sensors. Most of your cells have a single primary cilium, a non-motile cilium that sticks out from the surface like an antenna. It doesn't beat, it just sits there, but it's packed with receptors and signaling proteins, and it detects changes in the environment, chemical signals, mechanical forces, even changes in fluid flow. In your kidneys, the epithelial cells lining the tubules have primary cilia that extend into the flowing urine. When urine flows past, it bends the cilium. That bending opens mechanosensitive channels. Calcium flows into the cell, and the cell adjusts its behaviour, regulating ion transport, controlling cell division, maintaining tissue homeostasis. If the cilia are missing or defective, the cells lose their ability to sense flow, and they start proliferating uncontrollably, forming cysts. That's polycystic kidney disease. In your blood vessels, endothelial cells, the cells lining the inside of arteries and veins, have primary cilia that detect blood flow. When blood flows smoothly, in one direction, the cilia are bent consistently, and the cells remain quiescent. But in regions where flow is turbulent, near arterial branches, or in areas of narrowing, the cilia are jostled irregularly, and the cells respond by activating inflammatory pathways. This contributes to atherosclerosis, plaque formation in arteries, because turbulent flow, sensed by cilia, triggers the cascade of events that leads to vessel damage. Even in your brain, cilia are sensing. The ventricles, fluid-filled spaces deep in the brain, are lined with ependymal cells, and those cells have motile cilia that beat constantly, circulating cerebrospinal fluid. But they also have sensory roles. The cilia detect pressure, flow rate, and chemical composition of the CSF. If the cilia stop beating, or if the flow is obstructed, fluid accumulates, pressure increases, and you get hydrocephalus, water on the brain. It's dangerous, often fatal if untreated, and it's fundamentally a problem of ciliary function. Your sense of smell depends on cilia too. The olfactory epithelium, the patch of tissue at the top of your nasal cavity, is where you detect odours. Each olfactory sensory neuron has a single dendrite that extends to the surface of the epithelium, and at the tip of that dendrite are 10 to 30 cilia. These cilia are non-motile, but they're covered in olfactory receptors, proteins that bind to odour molecules. When you breathe in, air-carrying volatile chemicals swirls through your nasal cavity. The chemicals dissolve in the mucus layer and bind to the receptors on the cilia. The binding triggers a signal cascade inside the neuron, and within milliseconds the neuron fires an action potential that travels to your brain, where it's interpreted as a smell. The cilia massively increase the surface area available for receptors. A single neuron with 20 cilia has about 20 times more receptors than it would with just a cell body. That means 20 times more opportunities to capture odour molecules. That's why your sense of smell is so sensitive. You can detect some chemicals at concentrations as low as parts per trillion. It's molecular detection, enabled by ciliary architecture. And when those cilia are damaged, by infections, by toxins, by head trauma, you lose your sense of smell, anosmia. It's more common than people realise. Upper respiratory infections, like the common cold, can damage the cilia temporarily, causing anosmia that lasts days or weeks. COVID-19 famously caused prolonged loss of smell in millions of people, likely by damaging the supporting cells around the olfactory neurons, disrupting the environment the cilia need to function. But it's not just smell. Your sense of taste also depends on microvilli, tiny projections on taste receptor cells that are structurally similar to cilia, though built from a different protein scaffold. Taste and smell are tightly linked, both relying on surface projections to maximize receptor density. And then there's your sense of balance and hearing. In your inner ear, there are specialized sensory cells called hair cells. Each hair cell has a bundle of stereocilia, not true cilia, but elongated microvilli, arranged in a specific pattern on its surface. The tallest ones are on one side, the shortest on the other, creating a gradient. And at the base of the tallest stereocilium is often a single true cilium, the kinocilium. When sound waves enter your ear, they vibrate the eardrum, which moves the bones of the middle ear, which push on the cochlea, a fluid-filled spiral in the inner ear. The fluid moves, bending the stereocilia on the hair cells, and when the stereocilia bend toward the tallest one, toward the kinocilium, mechanosensitive channels open. Ions flood in. The hair cell depolarizes. It releases neurotransmitter onto a sensory neuron, which fires an action potential that travels to your brain. You perceive sound. The precision is extraordinary. Hair cells can detect movements as small as a fraction of a nanometer, less than the width of an atom. They can respond in microseconds, and they can distinguish frequencies over a range of about 20 Hz to 20,000 Hz. Because different hair cells along the cochlea are tuned to different frequencies based on their position and the stiffness of the surrounding structures, and it's the same for balance. In the vestibular system, the part of the inner ear that detects head movements and orientation, hair cells are embedded in gel-like structures that shift when your head moves. The stereocilia bend, channels open, signals fire, and your brain knows which way you're tilted, whether you're accelerating, whether you're spinning. That's how you can walk without falling over, how you can track moving objects with your eyes, how you maintain your sense of up and down even when your eyes are closed. All of it depends on ciliary and ciliary-like structures. Projections from cells, packed with mechanosensors, converting physical motion into electrical signals. And again, when these systems fail, the consequences are severe. Genetic mutations in the proteins that form stereocilia cause hereditary hearing loss. Exposure to loud noise damages and destroys hair cells, causing permanent hearing loss. They don't regenerate in mammals, so once they're gone, they're gone. Infections can damage the vestibular hair cells, causing chronic dizziness and balance problems. So here's the picture. Every breath you take is cleared by cilia. Every odour you smell is detected by cilia. Every sound you hear is transduced by ciliary-like structures. Your kidneys sense their environment with cilia. Your blood vessels sense flow with cilia. Your brain circulates its fluid with cilia. Your embryonic development depended on cilia to tell your cells which side is left and which is right. You're alive because of molecular motors, not metaphorically, literally. Dynein walking along microtubules, powered by ATP, beating in rhythm, coordinated through mechanical coupling and chemical feedback. The same motors that evolved in single-celled organisms billions of years ago. And you didn't build them, you inherited them. They were passed down through every generation, from the first eukaryotes to the first animals to the first vertebrates to the first mammals to you. The dynein in your lungs is the descendant of dynein in ancient algae. The structure is conserved, the function is conserved, the mechanism is conserved. Evolution doesn't start over, it tinkers, it reuses, it adapts what already works, and molecular motors worked so well, so early, that life has been using them ever since. But there's one more place we need to look, one more system where these motors are absolutely critical, where their function determines whether life continues or stops. Reproduction. Let's talk about sex, cellular sex. Because reproduction, the ability to create the next generation, depends entirely on molecular motors. In almost every sexually reproducing organism, at least one gamete has to move. Usually it's the small one, the sperm, the pollen, the motile gamete that has to travel, sometimes vast distances on a cellular scale, to find and fuse with the egg. And that journey is powered by the same motors we've been talking about. Dynein, microtubules, the 9+2 axoneme, the beating flagellum. Human sperm are among the most specialised cells in your body. They're stripped down, streamlined, built for one purpose, to swim. The head contains the nucleus, tightly packed DNA, condensed to about 1/6 the volume of a normal cell nucleus. There's barely any cytoplasm, no ribosomes to speak of, no machinery for making new proteins. The sperm can't repair itself, it can't grow, it can't divide. It's a missile, launched with a finite fuel supply and a single mission. And the engine is the flagellum. It's about 50 micrometers long, roughly the length of the head. And it consists of the same 9+2 axoneme structure found in every motile cilium and flagellum across eukaryotes. 9 pairs of microtubules in a ring, 2 in the centre, dynein arms reaching between them, pulling and sliding, bending the flagellum in waves that propagate from base to tip. But sperm have an additional structure that cilia don't, the fibrous sheath. It's a protein scaffold that runs along the length of the flagellum, providing structural support and elasticity. It makes the flagellum stiffer, more spring-like, so that when the dynein motors pull on one side, the flagellum doesn't just collapse, it bends, stores elastic energy, and snaps back. The result is a smooth, undulating wave rather than an erratic flail. And wrapped around the base of the flagellum, just behind the head, is the midpiece, a tightly coiled spiral of mitochondria. About 75 mitochondria, arranged end to end like beads on a string, forming a helix around the axoneme. These mitochondria are the power plants. They produce ATP through oxidative phosphorylation, the same process happening in the mitochondria and every other cell in your body, but here it's happening at maximum capacity. Because sperm need a lot of energy. According to research published in the early 2000s by David Clapham and colleagues at Harvard Medical School, a swimming sperm consumes about 10.9 molecules of ATP per second. That's a billion ATP molecules every second. The flagellum is beating at about 10 Hz, and every beat requires thousands of dynein motors to step, slide, and reset. The ATP is consumed as fast as it's produced. The mitochondria are running at full output constantly, and the fuel is finite. Sperm carry some glycogen, a small reserve of glucose polymer, but it doesn't last long. Once it's depleted, the sperm slow down. The flagellum beats less vigorously, eventually it stops. The sperm die. They have a lifespan measured in hours to days depending on the environment. In the female reproductive tract, with access to nutrients secreted by the surrounding tissue, they can survive for up to 5 days. Outside the body, they die much faster, minutes to hours. So the clock is ticking from the moment they're ejaculated, and they have a long way to go. During ejaculation, about 200 to 300 million sperm are deposited in the vagina. The distance from the vagina to the site of fertilization, the fallopian tube, is about 15 to 18 centimeters. That doesn't sound far, but remember, a sperm is only about 60 micrometers long. Traveling 15 centimeters is the equivalent of a human swimming 2,500 kilometers, the distance from New York to Los Angeles, across the country. And sperm don't swim in a straight line, they spiral. The flagellum beats asymmetrically, generating thrust that pushes the head forward, but also rotates it. The sperm traces a helical path through the fluid, like a corkscrew drilling through space. This is partly due to the off-center attachment of the flagellum. It doesn't emerge from the exact center of the head, so the thrust is angled, causing rotation. And this is actually advantageous. The spiraling motion stabilizes the trajectory. It prevents the sperm from tumbling randomly due to Brownian motion and fluid turbulence. But even swimming at full speed, about 200 micrometers per second, it would take hours for a sperm to swim the full distance, and most of them don't make it. The vagina is acidic, hostile to sperm. Many die immediately, others get trapped in cervical mucus, which is thick and sticky, a barrier that filters out slow or malformed sperm. Only the fastest, strongest swimmers make it through the cervix into the uterus. Once in the uterus, the environment is better, closer to neutral pH, less hostile, but the journey is far from over. The uterus is a large cavity, relatively speaking, and sperm have to navigate across it to find the opening to the fallopian tube, and there are 2 fallopian tubes, one on each side. Only one of them on average contains an egg during any given menstrual cycle. Sperm that enter the wrong tube reach a dead end. Their journey ends in failure. But the ones that find the correct tube still have work to do. The fallopian tube is lined with ciliated cells, millions of them, and their cilia are beating, creating a current that flows downward toward the uterus. The egg is swept down this current after ovulation, drifting passively toward the uterus, but the sperm are swimming against it. They're fighting a current generated by the same type of motor proteins that power their own flagella. Dynein versus dynein, cellular motors on both sides of the interaction. And this is important. The ciliary current in the fallopian tube doesn't just transport the egg, it also selects for the strongest sperm. Only sperm that can swim powerfully enough to overcome the current reach the upper part of the tube, where fertilization typically occurs. It's a filter, a physical barrier that ensures only the most motile sperm succeed. When a sperm finally encounters an egg, it doesn't just bump into it and fuse. It has to get through layers of protection. The egg is surrounded by cumulus cells, follicle cells that stick to the outside of the egg, embedded in a matrix of glycoproteins. Sperm release enzymes from the acrosome, a specialized compartment in the head, that digest through this matrix, creating a path, and the flagellum keeps beating, driving the sperm forward through the dissolving barrier. Once through the cumulus, the sperm reaches the zona pellucida, a thick glycoprotein shell around the egg itself. The sperm binds to receptors on the zona, and this triggers the acrosome reaction. The acrosome ruptures, releasing more enzymes that digest a hole in the zona. The sperm burrows through, flagellum still beating, pushing forward until finally it reaches the egg membrane, and then fusion. The membranes merge, the sperm nucleus enters the egg. The flagellum is left outside, discarded. It's done its job. The motor that brought the sperm this far is no longer needed. The egg will degrade it, breaking down the microtubules, the dynein, the mitochondria. None of it passes to the next generation, but the information does. The genes encoding dynein, tubulin, the proteins that built the flagellum, they're in the nucleus, and they'll be passed on. The next generation will build its own motors, using the same instructions, and the egg, for its part, doesn't move on its own. It has no flagellum, no motile cilia. It's passive, but it's carried. The cilia lining the fallopian tube beat constantly, moving the egg toward the uterus. If the egg is fertilized along the way, it continues drifting, dividing as it goes, until it reaches the uterus and implants. If the cilia aren't beating, if there's a ciliary defect, an infection that damages the epithelium, or scarring from surgery, the egg doesn't move. It can get stuck. If it's fertilized while stuck in the fallopian tube, you get an ectopic pregnancy, a pregnancy in the wrong place. The embryo grows in the tube, which can't support it, and eventually the tube ruptures. It's a medical emergency. And it's fundamentally a failure of ciliary transport. So both gametes, sperm and egg, depend on molecular motors. The sperm's motor moves the sperm. The egg's transport is driven by motors in the cells around it. Reproduction is impossible without them, and once fertilization happens, motors become even more critical, because the fertilized egg has to divide, over and over, growing from 1 cell to 2 to 4 to 8 to trillions. And every division requires motors. During mitosis, cell division, the chromosomes in the nucleus condense, the nuclear envelope breaks down, and the chromosomes attach to a structure called the mitotic spindle. The spindle is made of microtubules, radiating from 2 poles on opposite sides of the cell. Motor proteins, kinesin and dynein, pull on the microtubules, positioning them, adjusting their length, sliding them past each other. Each chromosome has a kinetochore, a protein complex at its centromere that attaches to microtubules from both poles. The microtubules pull in opposite directions, and the chromosome is held in tension, balanced at the center of the cell. This is metaphase. The chromosomes line up in a plane halfway between the 2 poles. Then, when everything is aligned correctly, the cell triggers anaphase. The connection holding the 2 sister chromatids together is severed, and motor proteins pull the chromatids apart. Dynein at the poles reels in the microtubules, shortening them, pulling the chromatids toward opposite ends of the cell. At the same time, kinesin walks along the microtubules, pushing the poles apart. The cell elongates, the chromatids separate. By the end of anaphase, each daughter cell has a complete set of chromosomes, and then the cell divides, cytokinesis. In animal cells, a contractile ring of actin and myosin forms around the equator of the cell and tightens, pinching the cell in 2. Myosin pulls on actin filaments, sliding them past each other, constricting the membrane like a drawstring. The cell splits. 2 daughters, each with a nucleus, each with a complete genome, each ready to divide again. Without motor proteins, none of this works. Chromosomes don't separate, cells can't divide, development stops, the organism dies. This is why mutations in motor proteins are often lethal. If dynein is completely non-functional, sperm can't swim, cilia can't beat, chromosomes can't segregate. The organism doesn't survive. Even partial loss of function, like in primary ciliary dyskinesia, causes severe disease. Motor proteins aren't just useful, they're necessary. And during embryonic development, motors do even more than move chromosomes. They move entire cells. In the early embryo, cells migrate. They move from one part of the embryo to another, guided by chemical signals crawling over and around each other, rearranging themselves into the structures that will become organs. This migration is driven by the cytoskeleton, actin filaments and microtubules, and by motor proteins walking along those filaments, pulling the cell forward. Neural crest cells, for example, arise near the developing spinal cord and then migrate long distances to form structures all over the body: facial bones, peripheral nerves, pigment cells in the skin. They crawl through the embryo using myosin and actin, extending protrusions called lamellipodia at the leading edge, anchoring to the surrounding tissue and pulling themselves forward. If the motor proteins don't work, the cells don't migrate. The structures they're supposed to form never develop. You get severe congenital defects, and we've already talked about nodal cilia in the embryonic node that rotate in a specific direction, creating a leftward flow of fluid. That flow carries signaling molecules to the left side of the embryo, activating genes that specify left side identity. If the nodal cilia don't beat, or if they beat weakly, or if they rotate the wrong direction, the signaling is disrupted. You get randomized left-right patterning. 50% of the time, organs develop normally. 50% of the time, they're reversed. Situs inversus. So motors determine body plan. They establish axes. They move cells where they need to go. They transport molecules to the right places at the right times. Development is a choreography of motion, and molecular motors are the dancers. And even after development is complete, even in an adult organism, motors are essential for maintenance. Cells in your gut lining turn over every few days. Cells in your skin are constantly being replaced. Blood cells are produced continuously in the bone marrow. Every new cell comes from division of an existing cell. And every division requires motors to pull the chromosomes apart. Your neurons, on the other hand, don't divide. Most of them are as old as you are, but they still depend on motors. Kinesin and dynein are constantly transporting cargo up and down the axons. Neurotransmitter vesicles travel from the cell body to the synapse. Damaged organelles and misfolded proteins are transported back to the cell body for degradation. If the motors stop, the neuron starves at the synapse and accumulates garbage at the cell body. Axons degenerate. Neurons die. This is what happens in some neurodegenerative diseases. Mutations in motor proteins cause axonal transport to fail, leading to progressive loss of motor neurons or sensory neurons. Charcot-Marie-Tooth disease, hereditary spastic paraplegia, both caused by defects in kinesin or dynein. So motors aren't just for development, they're for life. Continuous, essential, irreplaceable, and all of it. Every sperm swim, every ciliary beat, every chromosome separation, every neuronal transport event traces back to the same molecular machinery. Dynein walking on microtubules, kinesin walking on microtubules, myosin pulling on actin, ATP broken down, energy released, conformational changes, force generated. Over and over and over, the motors that keep you alive are the same motors that kept your ancestors alive, and their ancestors, and theirs, an unbroken chain of motion, inherited across billions of years. So let's pull back, let's look at what we've traced. We started with a bacterium, spinning its flagellum at 100,000 RPM through water that behaves like molasses. We moved to protists, coordinating thousands of cilia without a brain. We followed sperm swimming through the human reproductive tract, powered by the same dynein motors that beat in algae. We saw how your lungs clear mucus, how your ears detect sound, how your cells divide, all through molecular motors that have been running continuously for over 3 billion years. And here's what stands out. None of this was designed, not in the way we design things. When humans build a motor, we start with a plan. We draw blueprints. We machine parts to precise specifications. We assemble them in a specific order, with tools and jigs and careful measurements. And we build in redundancy, fail-safes, maintenance schedules. We designed from the top down. But molecular motors didn't come from a blueprint. They emerged, bottom up, from chemistry that learned to move, from proteins that folded into shapes that could capture energy from ATP and convert it into force, from random mutations that occasionally produced a protein that worked slightly better than the one before, from billions of years of selection, filtering out the failures, amplifying the successes, refining the mechanisms one small change at a time, and the result is machinery that by almost every metric exceeds what we can build. Efficiency. The bacterial flagellum motor converts nearly 100% of its input energy into mechanical work. ATP synthase operates at close to perfect thermodynamic efficiency. Human-engineered motors, electric, combustion, hydraulic, typically operate at 70 to 90% efficiency at best. We lose energy as heat, friction, vibration. Molecular motors don't. They're operating at the physical limits of what's possible. Precision? Kinesin steps along microtubules with 8 nanometer precision, placing each foot exactly on the next binding site every time. Dynein coordinates thousands of motors across a cilium, producing smooth, rhythmic beats. The tolerances are atomic, there's no slop, no wobble, just perfect, repeatable motion. Durability? These cilia in your lungs beat 10 to 15 times per second, 24 hours a day, for your entire life. That's over 400 million beats per year. Multiply that by a 70-year lifespan, and you're looking at 30 billion beats. The motors don't wear out. They don't need oil changes. They don't need maintenance. They run until the cell dies, and when the cell is replaced, the new cell builds new motors and starts the cycle again. Self-repair. Molecular motors are constantly being degraded and rebuilt. Proteins are broken down by cellular machinery and recycled. New proteins are synthesized from amino acids, folded by chaperones, and assembled into functional motors. The motor you had yesterday isn't the same motor you have today at the molecular level, but the function is identical. The structure is preserved. It's ship of Theseus at the nanoscale. And self-assembly? Perhaps most remarkably, molecular motors build themselves. There's no external construction crew, no assembly line. The proteins are made in the cytoplasm, they diffuse through the crowded cellular environment, and they find each other. They recognize complementary surfaces, lock together, and form functional complexes. The bacterial flagellum assembles in about 20 minutes. The cilium assembles over a few hours. No instructions needed beyond the shapes of the proteins themselves. This is emergence. Complex, coordinated, functional structures arising from simple rules and local interactions. No top-down control, no global coordinator, just molecules, following the laws of chemistry and physics, and somehow producing motors that work. And this is what life does, not just with motors, but with everything. Metabolism, replication, sensing, responding. It's all emergent, all self-organized, all built from the bottom up, through interactions between molecules that have no knowledge of the larger structures they're building or the functions they're serving. The cell doesn't know it's alive. The motor protein doesn't know it's moving something. There's no intentionality at the molecular level, just chemistry. Bind ATP, change shape, release ADP, reset, repeat. Do these enough times, with enough proteins, and you get a flagellum that propels a bacterium toward food. You get cilia, that clear mucus from lungs. You get a heart that beats 100,000 times a day, pumping blood through 100,000 kilometers of vessels. Motion at every scale, built from motion at the molecular scale. And humans are trying to replicate this, to build synthetic molecular motors, to engineer molecules that can walk, rotate, pump, transport. There's been progress. In 2016, Jean-Pierre Sauvage, Fraser Stoddart, and Ben Feringa shared the Nobel Prize in Chemistry for their work on molecular machines. They built molecules with moving parts, rotors that spin when exposed to light, shuttles that slide along molecular tracks, rings that open and close. These are artificial molecular motors, chemically synthesized, designed by humans, but they're primitive compared to what biology has achieved. The synthetic motors are slower, less efficient. They require specific conditions, particular solvents, temperatures, light wavelengths. They don't self-assemble, they don't self-repair, and they can't coordinate with thousands of other motors to produce complex large-scale motion. We're learning, but we're far behind. Evolution had a 3 billion year head start. Still, the potential is enormous. If we could build molecular motors that rival biological ones, the applications would be transformative. Imagine drug delivery systems where molecular motors carry medicine directly to cancer cells, navigating through tissue, targeting with precision. Imagine nanoscale factories where motors assemble molecules one atom at a time, building materials with properties we can't achieve with bulk manufacturing. Imagine self-healing materials where motors repair damage as it occurs, reknitting broken bonds, restoring structure. Some of this is already happening. Scientists are using DNA nanotechnology to build molecular walkers, strands of DNA with legs that can step along DNA tracks, carrying cargo as they go. They're slow, and they only work in test tubes under carefully controlled conditions, but they work. They demonstrate the principle. and we're learning from biology. Researchers are studying dynein and kinesin, mapping their structures at atomic resolution, watching them step in real time, figuring out how the conformational changes translate into force. The goal is to understand the design principles, not to copy them exactly, but to extract the ideas and apply them to synthetic systems. Because molecular motors solve a problem that's fundamental to nanotechnology. How do you do work at the nanoscale? How do you move things, build things, manipulate things when everything is buffeted by Brownian motion and you can't use macroscopic tools? The answer is, you use the same strategy life uses. You harness energy from chemical reactions. You build structures that bias random motion. You create directional processes from reversible interactions. You let the system self-organize. And if we can do that, if we can build motors that self-assemble, self-repair and coordinate, we'll have crossed a threshold. We'll be able to build machines at the scale where biology operates. We'll be able to interface with living systems in ways that aren't possible with current technology. We'll be able to repair cells from the inside, to engineer tissues, to control biological processes at the molecular level. But even if we never achieve that, even if synthetic molecular motors remain a distant goal, what we've learned is profound. Because understanding how life moves, really understanding it, at the level of atoms and molecules and forces measured in piconewtons, reveals something fundamental about what life is. Life is not static. It's not passive. It's not an arrangement of molecules that happens to persist. Life is motion. Constant, directed, purposeful motion. From the bacterial flagellum spinning to the heart pumping, to neurons firing to muscles contracting. Motion is the signature of life. When motion stops, life ends. And that motion doesn't require complexity in the way we usually think about it. A bacterium has no brain, no organs, no differentiated tissues, but it moves with purpose, it navigates, it hunts, it escapes, all through molecular motors and chemical gradients and mechanical feedback. The intelligence, such as it is distributed, emergent, encoded in the structure and dynamics of the system, not in any central processor. And that's true at every scale. Your brain coordinates your movements, but your brain is itself built from cells that move, that transport, that communicate through motors and channels and signals. There's no homunculus, no ghost in the machine, just layers of motion, from molecules to cells to tissues to organs to organisms, motion all the way down, motion all the way up, and it's old. The motors in your cells are descended from motors that were spinning in ancient bacteria 3 billion years ago. They've been refined, adapted, duplicated, specialized, but they've never been replaced. Evolution found a solution that worked, and life has been building on it ever since. So when we ask, how did life learn to move before it had muscles? The answer is, it learned through trial and error over billions of years, through the same process that shapes everything in biology. Random mutation, natural selection, drift, occasional catastrophe, survival of whatever worked well enough to reproduce, and what worked was chemistry. Proteins that could change shape, membranes that could anchor rotating rings, filaments that could slide past each other, energy gradients that could be harnessed. Simple components interacting through simple rules, producing complex coordinated functional behavior. That's how life learned to move, not with blueprints or plans or foresight, but with molecules that stumbled randomly into configurations that generated motion. And once that motion existed, once cells could swim and hunt and escape, the rest followed. The evolutionary arms race, the diversification, the scaling up from single cells to multicellular organisms to animals with nervous systems and muscles and bones. But the motors never changed. They're still there, still running, in bacteria, in protists, in algae, in the cells of your lungs and your ears and your reproductive tract. Ancient, conserved, universal. And every time you breathe, every time you hear a sound, every time your heart beats, you're running on engines that are older than bones, older than eyes, older than brains, engines that existed before anything we'd recognize as an animal. Engines built from nothing but folded proteins and chemical energy. That's the legacy we inherited. Not just genes, not just DNA, but machinery. Working machinery, Machinery that moves us, heals us, senses for us, builds us. Machinery that connects us to the first cells that ever moved in ancient oceans, under a sky with no oxygen, on a planet that looked nothing like the one we live on now. The motors are still running, and they'll keep running, passed down through every generation until there are no more generations to pass them to. Motion is life, and life has been moving for 3 billion years.